हेलो 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 हेलो। अभी देखो, सुनो सुनो, आप लोग आपको तो डायरेक्ट आएगा इधर, सुनो ना, लेवल आ रहा होगा ना, ऑडियो लेवल बिल्कुल डायरेक्ट आएगा जिसमें वीडियो करते हैं। हेलो हेलो, ग्रीन हो रही हुआ ना, बस सर बचा के अपना। हेलो 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 हेलो, साउंड चेक 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 चेक, वन टू। हेलो हेलो, कुछ और बंद हो गया, कुछ और। नहीं, वो फ्लैश जो था, ये वाला, ये वाला झुमका आ रहा है, झुमका जा रहा है। एक बार देखना ही क्या हुआ? नहीं नहीं, ये तो हमारे ऐसे रहेगा तो इसको खोल एक काम कर।
Settings of video and audio inputs. As you can see, a lot of cables are connected. We will now set up the video.
check check 1 2 check 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 1 check check 1 check 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 1 PC1 है, PC1 है ना ये। चेक चेक वन, ठीक है। हेलो 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 चेक चेक वन टू, हेलो 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 चेक चेक वन, हेलो, ठीक है। hello, hello, hello. John, audio check करिए बोलिए. Audio check करें. Hello, 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 hello. Check, 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 check.
Check.
Check
Okay.
one, two. Audio. Audio. Hello, check, check, check one, two. Test. Hello, hello, check. Hello, hello, check, check one, two. Hello, hello, check, check, check one. Hello, 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 check, check one. Hello,
I can hear you.
hello, check, check one. Hello, hello, check, check, check. Hello, 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 hello. आ गया ना? Hello, hello. Hello, hello, hello. Check, check. Hello, hello, hello. Check, check one, two. Test. Hello, hello. Check, 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 check. Check. इसका बंद कर दियो, इसका ऑडियो. उसका इसका माइक ऑन का बंद कर दियो. ठीक है. ये मुझे वो मिलेगा क्या? उससे बात हो पाएगी? वो उस कैमरे से उसको थोड़ा सा बोलो जूम आउट करने के लिए, और इसका पोडियम पे हल्का सा नीचे करना. पोडियम से हल्का सा नीचे करना. हाँ, मतलब कितना आ सकता है नीचे. उसके पास कुछ है वो सेट कर देगा. एक मिनट में नहीं, उसको सेट करके नंबर 3. एक मिनट उसको बोलो कैमरे को देखो. हां ठीक है, ठीक है. तो फेस नहीं आएगा फिर. वो ये वाला कटेगा फिर. हेड ठीक तो आ रहा है. ये देख लो.
नहीं नहीं, अब चलते हुए नहीं चाहिए. पता चला उस दिन तेरा यूट्यूब बंद हो गया तो.
अच्छा, जूम पे साउंड नहीं आ रहा. नहीं
सर बंद है ना, यूट्यूब खुला. वो टेस्ट करेंगे तो आएगा.
नहीं तो अब
अभी आप होम पे भी टेस्ट
हेलो, हेलो, हेलो.
हम्म,
Check, check. Hello, check one, two. Hello,
क्लियरिटी नहीं है.
hello, check, check one. Hello, hello, check. Hello, one, two. Hello, hello, check. हाँ?
साउंड
हेलो. एक मिनट.
इनके नंबर बैठ जाए, बीएस में बैठ जाए, कोई दिक्कत नहीं है। 4 बनाए।
और क्या नाम है, जो वाईफाई का पासवर्ड है ओपन
वाला ओपन नहीं किया मैंने।
वो कर देंगे फिर बड़बड़ हो
वाईफाई
जाएगी।
ओपन कर
स्विच स्टेक आरसीबी के चल रहा है ना।
सारे गैस मुझसे मांगा, एक था गैस तो आरसीबी से पहले से
स्टे का वाईफाई ओपन कर।
हेलो 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 हेलो, चेक चेक 1। हेलो, ठीक है। ठीक है, नहीं नहीं कम करना है क्या? कोई बात नहीं, कोई बात। कम कर देख। हेलो हेलो, चेक 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 1। हेलो हेलो, चेक चेक। हेलो हेलो, चेक चेक।
और कम करो।
हेलो, चेक चेक 1 1 2। हेलो, ठीक है। हाँ ये, हाँ ठीक है।
हाँ सिर्फ एक का रह रहा है शायद, वो मैम अभी ले लेंगे। तुम लीक्स मत
Morning everyone. I request you all to please turn off your cellular devices, and for those joining online to please put your devices on mute as we will be starting shortly.
Hello. 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 Hello.
Good morning, everyone. On behalf of the entire organizing committee of Translational Health Science and Technology Institute, I, Shefali Tyagi, and my co-host, Jaskaran Kaur, extend a warm welcome to all the dignitaries, guests, faculties, and colleagues to the International Meet on Preparedness for Future Epidemics.
It is a matter of extreme honor and pleasure for us to be your hosts for the next 2 days, as we hope to have a healthy interactive exchange of ideas and information with the goal to develop roadmap for accelerating concept, design, delivery of vaccine development for future epidemics.
We would like to start the event by the auspicious lamp lighting ceremony. I would request the dignitaries, Dr. K. Vijay Raghavan, Dr. Sudhanshu Vati, along with Dr. Pramod Garg, to come on the stage for lighting the ceremonial lamp.
Yes, sir. వచ్చినాడు హాలిడేస్ కి వచ్చింది క్రిస్మస్ ఎగ్జామ్స్ అయిపోయినాయి యూ వాట్ టాకింగ్ మ్యాన్ you literally nowadays after IBM business ISB you are
lost in
ఏది ప్రోగ్రామ్ నడుస్తుందా
వీళ్ళు ఎప్పుడు వచ్చేది మా రేపా
రేపు నైట్ వస్తారు వెంకటరమణ ఎన్నిటికుందో చూస్తే యాక్చువల్ పోవాల్సిన జ్వరం వచ్చింది జ్వరం కాదు భయం వేసింది మన వాళ్ళు పిచ్చినా కొడుకు కదా ఒప్పేసుకుంటాం ఇంతమందిలో
Thank you, everyone.
To begin with the event, may I request our director, sir, Dr. Pramod Garg, to give the welcome address.
మెహర్ గడ్ పోయింటాడు తిరుపతి
अभी कर Dr. K. Vijay Raghavan, former Principal Scientific Advisor to the Prime Minister of India and former Secretary DBT, Dr. Vrati, all the dignitaries who have come from abroad and India, the faculty members, delegates, ladies and gentlemen. A very good morning to all of you and a warm welcome to THSTI. And it's my real pleasure that we are hosting this international meet on epidemic preparedness. Despite all the problems that we faced in the last 2 years, and often the situation was called a disaster, I think there are many lessons that we have learned during COVID. And one of the important lessons that we have learned is that we can really face the challenge. And whenever there is an adversity, it actually brings out the best in all of us. As a nation, India responded remarkably well. I mean, all over the world, people recognize that. Be it taking care of patients, educating the public, or development of a vaccine. And this has given us a lot of confidence as a country that we can do well, and sometimes even better than the developed world. And one of the reasons for that, to my mind, was that people were with us. There was a very strong leadership, and which took people along, and everyone understood what is supposed to be done, whether it was wearing masks or all kinds of COVID appropriate behavior that we were supposed to follow. Everyone did that. There wasn't actually any kind of vaccine hesitancy that we witnessed in many of the developed world. So overall, I think there are more lessons learned, although we had a huge loss of life also, as all over the world, because of this deadly virus. And that is why now I think we are in a better shape to face future challenges. Somehow or the other, the focus has shifted from non-communicable diseases, which actually form even today the largest share of the healthcare burden, the focus has shifted somewhat to infectious diseases. And we know that in future also there could be many other infectious diseases, apart from the fact that we still have a lot of burden of tuberculosis or dengue and other infectious disease. And this meet in particular will focus on how we can actually come up with new solutions for emerging problems. We had a delegation from CEPI last month, which talked about the CEPI's 100-day challenge. I mean, the COVID vaccine actually came up within 6 months of the start of the pandemic. Now the challenge is even, bring it down to 3 months or 100 days. Are we really prepared for that? And to answer that, I think if we have the pipeline set in, if we have the platform ready, we can do that. If you know what are the key components of such a strategy, then we can actually achieve that. For example, we have actually seen a lot of synergy and convergence between academia and industry. And that is why I'm happy that we have many representatives of the industry here during this meet, which all the key players in this country are here. I'm very happy to note that. And my special thanks to Dr. Shailja Gupta for organizing this meeting and bringing them all together. This is very, very critical. And I was talking to all my scientists here that, whatever you're doing in your lab will remain within the lab, or at the best, you will have a good publication. You will add to your CV, may get an award or something, but that's the end of it. Unless industry comes along, they think your product is good and take it further. And that is what has been shown in the case of Covaxin. Dr. Priya Abraham is here, and she's the one who was at the forefront of that development of that vaccine. So I'm very happy that THSTI has been involved in many such programs during last 2 years. And for the benefit of those who are here with us and may not know much about THSTI as an organization, can I have this presentation on, please? So what I'll do is to quickly run through some of these slides that I prepared for our guests from outside India and also within India who may not know much about our institution. So THSTI is a catalyst and enabler of translation in the research ecosystem in this country. We have many team-based research programs which prioritize public health problems unique to India. One of the programs which is major and today's focus is infection and immunology, where we are trying to understand the disease pathophysiology, developing novel vaccines and molecules. We have a large program on maternal and child health. We have a program on non-communicable diseases, particularly non-alcoholic fatty liver disease. And we have a multidisciplinary clinical and translation research program on diagnostic development, particularly point of care tests. And these are ably helped by many facilities that we have: a large BSL-3 facility, biorepository, data center, animal facility, and also something which is unique to this institution is Clinical Development Service Agency, which is kind of an academic CRO. Now, the infectious disease is the major focus here, and tuberculosis, as I mentioned earlier, is still a major problem in this country and also in many parts of the world. THSTI is involved in disease biology, therapeutics, and vaccines. In fact, there are many new molecules which are being investigated, and I'm happy to note there are at least 5 molecules which are at different stages of development and shown very good promise. 2 such molecules, SPR506, has shown very good results, and SPR113, which was developed earlier, is awaiting large tox studies, followed by hopefully phase 1 studies. So this molecule, we hope in the next couple of years, might be at a stage that we're looking at adding it to the anti-tuberculosis armamentarium. Dengue is another major focus area at HSTI. We are looking at multiple aspects of dengue, development of cohorts, antivirals, vaccine assay platforms, and antigen designing. Cohort study, Dr. Guru Prasad is involved in dengue studies. We have many cohorts through AIMS, collaboration with AIMS Delhi, Employees State Insurance Medical College Faridabad, Jamia Hamdard, and he has shown through multiple studies the prevalence of antibodies which are cross-reactive across serotypes. And dengue antiviral studies are ongoing, where we are repurposing some of the known drugs. In fact, 3 drugs have shown promise: remdesivir, niclosamide, and nelfinavir. In fact, we have a partnership with DNDi, the international organization, through which there is a large consortium now, which is looking at many antivirals. And these data have been also confirmed studies from Thailand and Brazil. Our scientist, Dr. Suti Samal, has also developed a preclinical animal model to support dengue R&D. And this is a model of severe dengue infection. So, you know, all of you know that severe dengue is the one which kills patients, and we have now developed a very good model with enhanced vascular permeability, where many of these antivirals and anti-inflammatory drugs can be tested. Vaccine is also another important area for dengue, and couple of antigens are being designed, mainly dimers and also domain 3 based putative vaccine constructs, which have shown broadly neutralizing antibodies. So I hope that some of these could be taken forward in partnership with industry. Influenza has also been a major focus of research area. And you know that every year in India, thousands of people die because of the common flu, and the vaccines which are available are, one, they're costly, and second, every year there is a new strain coming in. So we have 3 large grants through DBT and European Union, and through which many institutions are involved in these studies, and I hope that we can have a very good vaccine in the next two to 3 years. For the first time in this country, we have established a ferret facility at THSTI for influenza-based research. And I think this will come handy for the vaccine development. COVID, of course, most institutions in this country have been involved in COVID-19, which is so very encouraging that whether or not you are a cardiologist or a cardiothoracic surgeon or a diabetologist or a vaccinologist or immunologist, everyone contributed to COVID-19, which is so gratifying actually. And no wonder that THSTI was involved in multiple areas: cohort developments, vaccine effectiveness studies, animal models, vaccine design and development, immunological assays. In fact, we have more than 31,000 people enrolled in different cohorts. Either they were patients who were infected with the virus or they are vaccinated people, to follow and find out the long-term durability of immune responses, including hybrid immunity. In fact, in one of the editorials from THSTI, we first of all mentioned about this hybrid immunity concept of vaccine and natural infection, which is now so pervasive. During the height of Delta infection, THSTI carried out a vaccine effectiveness studies against Covishield and showed it to be 63% as against the Wuhan virus, which was over 80%, and this was published in the Lancet. THSTI is also a part of the INSACOG network. We have now sequenced more than 2,000 viral sequencing, and we also established a pan-India multi-site hospital network study, where we have 14 hospitals across the country, and each hospital is linked with a sequencing site to understand the emerging new variants and how that variant is causing what kind of illness, mild, moderate, or severe. And now we have around 5,000 patients' data prospectively collected across the country. When Omicron first emerged in December, THSTI was the first institution to culture the virus, and we showed that there is suboptimal neutralization of the Omicron by the vaccine-induced antibodies. And again, this paper was published in the Lancet eBiomedicine. When it came to development of vaccines, THSTI was involved very, deeply with the vaccine development, be it Sputnik 5, Zydus DNA vaccine, and biological E. And now we are also developing our own indigenous multi-epitope vaccine against SARS-CoV-2 in partnership with Panacea. I'm happy that we got the drug substance, and also the preliminary results are very, very encouraging. And based on this, our scientists led by Dr. Suti Samal and Amit Avasthi, we have a novel technology platform based on which we are developing a novel, highly immunogenic antigen for pan-beta coronavirus vaccine in partnership with Panacea. In fact, this consortium was awarded $12 million by CEPI to develop this vaccine. And again, the preliminary results are very encouraging. And this will be against SARS-CoV-1, COVID-2, and MERS. And multiple epitopes will be stitched together to come up with a broadly neutralizing antibodies. Mother and child health is another major program. In fact, this is a unique institution which is engaged both in basic biology, translational, and also clinical research. And the largest cohort of 12,000 pregnant women has been developed at HSTI, which is being followed up to understand what is happening to the babies, whether they are preterm, if they are preterm, what are the risk factors for that. And there is a multi-omics approach to understand the outcomes of pregnancy, microbiome, genomics, proteomics, and metabolomics. And there are multiple randomized trials also being conducted in newborns to understand the disease biology and also interventions to decrease mortality. In fact, I want to highlight this study on immediate continuous kangaroo mother care that you might have heard. And HSTI was involved in coordinating this study and also participated in it through our collaborating partners, and there was a 25% reduction in newborn mortality following this. And this month, in November, WHO updated its guideline to include kangaroo mother care as a standard of care for all preterm babies. And following the success of this, we have now a second phase of birth cohort where newborns will be followed up at least for 5 years and hopefully for 10 years to understand their postnatal development and the influence of prenatal and natal exposures to different factors. And this gardening cohort has now, there are many partners across the world in WHO and JIPA and MOMI consortium. We have existing partnership with many hospitals too in our close proximity in Gurgaon and Faridabad, where all these pregnant ladies are being recruited and followed up. And I'm very happy to tell you that we are currently establishing a medical research center in this campus, the DBT National Science Cluster, Bioscience Cluster. And this hopefully within a year or so, we will have a research center on campus here, which will be involved in very, vaccine trials, phase 1, phase 2 studies, cohort studies, and other things. Human microbiome is another major program at HSTI. And so is immunology division, which is engaged in both basic T-cell biology and also fundamental applied research to various diseases. Clinical Development Service Agency, I mentioned at the beginning, is something which is unique in this country. This is the only institution which has an academic CRO, which supports and nurtures clinical product development. There are many objectives. The main is to conduct, supervise, and monitor multi-center trials, and also involved in training and capacity building in the country. It has many verticals. We provide support for clinical regulatory science, clinical portfolio management, IT data science, biostats, and training. And we have totally completed 25 trials at different sites, and 10 are ongoing where HSTI CDSA is involved. We also provide training for GCP and good clinical lab practices, ethics, and clinical research methodology. And this year we have started MSc in clinical research at HSTI. The first batch has been taken of 14 students. This is a 2-year course, and this will, I think, help a lot in building capacity in clinical research, particularly clinical trials in the country. All these activities are supported ably by many translational laboratories. One of the important labs that we have is a bioassay lab, which is a CEPI centralized lab, part of 9 networked labs in the world. We also have a large biorepository. In fact, during COVID also, this biorepository helped a lot. Not only the academia, but also industry. We supplied biospecimen to so many companies also for their vaccine development and diagnostics. We also have a large BSL-3. We had a small BSL-3, but now a large new BSL-3, which is a modular BSL-3, is functional. And I would suggest and request all the partners here, be it from academia or industry, utilize these facilities, which are state-of-the-art. We also have a very large animal facility. We have transgenic mouse and other things, and this is also being upgraded. This is a shared, all these facilities are shared between THSTA and RCB, and I am happy that Dr. Vrati is here. THSTA and RCB work very closely So for example, small animal facility is now being upgraded to a world-class facility, which should be available over the next 1 year or so. We have all the latest equipments, including complete proteomics and metabolomics, next-gen sequencing, Schrodinger for drug discovery. This is something we acquired last year and is coming handy for new molecules and also for vaccine designing, so which is very important. One of the new things that we are setting up now, currently in progress, is a vaccine design and development center. It will consist of 3 major components: upstream processing, downstream processing, and analytical lab. And this is basically from a lab. If you have an antigen, how do you convert it into a drug substance and drug product before it goes to the industry for final development of a vaccine? So this will be very important to bridge the gap between academic labs and industry partners for early translation. So in summary, the major focus of THSTI is on virology and vaccines, mother and child health, tuberculosis, immunology, microbiome diagnostics, and CDSA. And we have, obviously all thanks to my faculty and scientists, that we have many achievements, including publications, some of the top journals, and many patents. I'm just like a caveat or a boatman. I'm just, facilitating their research. But I must say that the team here is excellent in terms of their dedication to science and research and capabilities and, intelligence and everything. And I'm really, really happy to be part of this large team. Thank you very much for kind attention. I've taken a little longer, but I just want to tell people about this institute, which is relatively young. Thank you for your attention.
Thank you, sir, for delivering the welcome address. We have the proud privilege to welcome Professor Vijay Raghavan, Padma Shri awardee and former principal scientific advisor to the Government of India, to deliver the keynote address. Professor Raghavan is currently Homi Bhabha Chair at the National Center for Biological Sciences, Tata Institute of Fundamental Research, India. He is also a fellow of the Royal Society and Foreign Associate of the US National Academy of Sciences. Sir, I request you to please come on the podium.
Dr. Pramod Garg, Dr. Sudhanshu Rathi, and all our colleagues over here, very good morning, and it's a great pleasure to be here. Is this the right one? Yeah. Yeah. Thank you. It's a great pleasure to be here, and it was really very nice to listen to Pramod's exposition about all what is happening at THSTI and at the RCB. This campus has really come up extraordinarily well in terms of not only its facilities, but also importantly, the people and the questions they address. I'll come back to that later on, but I'd like to add a little bit on that front to say that if you start from the South Campus of Delhi University, go on to JNU, IIT, ICGB, NII, NIPGR, All India Institute of Medical Sciences, the Indian Statistical Institute, the National Brain Research Institute, and here RCB and THSTI. There's an extraordinary powerhouse of capability, not just in life sciences, but in any area of research. And it's really through this network of institutions, if they work together, that many of these issues which we are going to address over today and tomorrow can actually result in fruition. So if you go to the CEPI website, and I think you have this on your notes also, about a 100-day challenge, it's really about a disease X and whether one can respond fast enough to that disease X as it becomes an epidemic or a pandemic to make a vaccine. And CEPI's point of view is that this is feasible. So what I will do very briefly now, and I assure you that we'll be back on time and we'll be able to have tea on time, is to go through CEPI's points and ask whether the world is ready, and then ask what we can do to perhaps address this challenge if it's meaningful. So first of all, the first point which CEPI makes is that it asks, is it possible to design a vaccine candidate within 2 days of the genetic sequence of a new virus being published? And it says we know that that's possible because that's what the NIH Vaccine Research Center did in the case of SARS coronavirus 2. Now, that's very well. In this case, the vaccine design was a genetic sequence resulting in the design work being done at NIAID, which has, got several decades of extraordinary work on vaccine design and development. And indeed, that work went very rapidly. But if you look in other examples, let's take Zika, whose viral sequence is already known. The progress in vaccine design and taking it forward is very, very comparatively much slower for a variety of reasons. Partly, all of that we can guess, but partly because it didn't become a pandemic-like situation or wasn't anticipated to become such as SARS-CoV-2. So what I'd like to point out in this and the subsequent points is that if we take the example of response during a very major crisis for a focused response to that specific problem, you will find extraordinary examples of heroism and speed, which are not normally likely to be seen in other situations, even though they are equally or maybe very important by itself. Let's look at the next feasibility argument of CEPI's. CEPI asks whether it's possible to move into first human trials in 66 days from the release of the genetic sequence, and says that's what Moderna did. But if you look at, the situation in other diseases, there can be many complications, and it fortunately turned out that SARS-CoV-2 was simpler, that the vaccine design and human trials can be much more difficult given the level of exposure, the complications which arise from previous exposure, and the design, the presence of different variants of the virus, the responses, the immune responses to those variants in the context of previous exposure to other variants. So all of this can complicate this point about human trials. Much greater care would be taken in quote unquote normal situations because vaccines have to be administered to people who have no other disease typically. The third argument is it possible to publish the first safety data 63 days after a phase 1 clinical trial starts? And CEPI says that's what Moderna did, therefore it's feasible to do that. But let's take another example, the cholera vaccine, which was developed, one version was developed in Sweden in the 1980s and contained a native B subunit purified from cholera toxin produced by a wild type strain. And the immunogenicity and safety studies really took place effectively 4 years later, or more years later, 1984 and then in 1987. So again, certain contexts which are not seen as urgent or which have biological complications can take a long time for this deadline to be met. It may not always be met in normal times. So of course, the point CEPI makes is that this is disease X, and that's a very dangerous new disease, and therefore the response will be much better coordinated. CEPI's fourth argument is that it is possible to go from first human clinical trials to vaccine registration in about 7 months, and because that's what Pfizer-BioNTech did. Now, again, this is a situation which is an unusual situation during the pandemic. And there are other examples. Many of you are very familiar. Those of you who have been at ICGEB, for example, about the malaria vaccine, about the way tuberculosis vaccine research has had major challenges, that the vaccine registration path can be very, very long haul and can be very complicated. So it's not at all evident that this speed is something which is predictable no matter what the disease is. Finally, the last point is it possible to get emergency use approval within 1 day of filing all the required data with regulators, within 1 day. And CEPI says that we know this is possible because that's what China's CanSino did for their adenovirus vector vaccine, which subsequently has got emergency use approval in many countries. Here again, the situation depends illustratively on the context. If there is a pandemic going on and there's an emergency, regulators move faster. They have to contend with what is feasible in the context of what they would normally do versus what the dangers of not moving fast enough are. And those kinds of challenges vary a lot depending on the context. And that's a very big challenge. So here are 4 or 5 different components of the CEPI challenge, which are articulated against responding against a disease X, but seems to ignore, and rightly because it's focused, I suppose, on one thing, seems to ignore the broader context in which these are, these are stated. If you were to go outside this campus or go to anywhere in Delhi or anywhere in the country and ask what are the top 10 challenges which people face, getting a vaccine ready against disease X is not something which is on most people's radar. They have a huge number of other problems, and this will be in the top 10 problems, this will rank about 1,000. So how is it that public imagination and public interest can also come in onto such a big challenge? Because that's necessary, because resources are necessary, and many other components are necessary. So unless all of that happens, this is not going to be a priority. Even from a public health perspective, if you list out the public health challenges today, there are many pressing demands on resources and capabilities of people and so on. So that's a very big challenge. And therefore, to apportion intellectual resources into these kinds of things remain to be they remain a big concern. So what exactly is the way out, or is there a way out? So let me end by saying that to this question about can India meet the CEPI challenge, the first question we should ask is the question correctly posed? Because if the question is not correctly posed, we will enter into a discussion about whether and how to do it, when actually our answer should be, well, it's a bit more complex and the question should be posed differently. And I state that the question perhaps could be posed differently because we know little about the nature of disease X. Is it going to be something like malaria or HIV/AIDS, where vaccines have been extraordinarily difficult, or tuberculosis? Is it going to be something else where vaccines have been, we've been very fortunate and vaccine development has been very rapid? So unless we know about all of that, it's very difficult to say that a 100-day challenge as a generic response is something which one can be prepared for. The other matter relates to public health information and data flow systems. Are they up to speed, both nationally and globally? Typically, the world over, those who report on public health emergencies or potential emergencies are also the people who play a role in making sure that they are effective in responding. In other words, those who report are also effectively the responders. This poses a big challenge in every country. Do you react to any potential emergency by stating that it's a potential emergency and nothing happens and you're likely crying wolf? Or you would be accused of not taking enough care and therefore that emergency arose in the first place? Or do you just hope that the potential emergency goes away and doesn't spread, or take local measures and then see what happens? So it's all very well to say that one can get sequence information fast, but the question is, there's a long early lag and a late lag period in many contexts before one is ready to get that sequence information, right? So there are other ways by which one can get potential sequence information across the context in a generic manner, but in a specific instance of potential danger, which one has to be extremely watchful, there's a lag in public health systems all over the world. And we saw that's what happened in China too. So that lag is a big challenge, and the public health information system is something which needs to be addressed. Even after an emergency has been identified, data flow systems tend to contend with different agencies at the city level, the state level, the national level, the international level, all working in sync. Remember that while the sequence in China, the SARS-CoV-2 sequence was made available very rapidly, it was not done through an official channel. It was done and sent out, and then, the official channels responded. So one must be, again, wary about relying on the speed of this information in multiple contexts, and are those global information systems in place. This is really the key. There is the traditional 20th century approach to public health has been sharing public health data and information in a very slow postal mail kind of manner. That has not yet even fully transcended to using IT kinds of systems, let alone having an integrated way by which immediate information is shared globally. This is something which is very important and necessary for any of the subsequent steps to take place. And this is an area where India can actually push and take a significant role here, particularly as the G20 presidency comes up. So this information system flow is going to be very important. Our vaccine platforms in place. We saw the extraordinary efforts ramping up at THSTI. There have been similar efforts at the ICMR National Institute of Virology, the defense labs, and in other places where platform technologies are being put in place, both for early stage culturing of viruses and also various kinds of platforms. No particular platform is a magical one. It's not that mRNA platforms are going to solve everything, or traditional platforms are not effective. There's going to be a lot of trial going on, so multiple platforms need to be nimble. They are very expensive to, at early stages, to run and maintain. They need to be done in partnership with industry. That means our institutional networks need to be feasible to do that, and also encourage startups and facilitate them in using these kinds of platforms. Our shared preclinical facilities in place, and that relates to the vaccine platforms. If at all one needs to be competitive, and I don't mean competitive only in a business sense, if one has to get the job done from an Indian perspective for many diseases which will be addressed only by India, then you need to have an ease of access to animal facilities, preclinical facilities, and other trial facilities, which I'll come to, for our small startups, which are adjacent or in or budding off from our university system. Unless this happens on a large scale easily, we're not going to get anywhere in terms of being nationally prepared. There are some very exciting startups which came up successfully during these very difficult times. Their stories will be very important to listen to and see how one can facilitate them. Again, these shared preclinical facilities shouldn't be just amongst institutions or within small institutions with startups, but also with bigger industry. That's also necessary. And they include large animal trials for large animal facilities for large animal trials also. Are animal experiments feasible with speed? We have seen some heroic efforts during the pandemic, but if you have to go forward, we shouldn't rely on heroic efforts. We should have these kinds of situations relatively routine. There have been instances where, a irrelevant minor complaint of some sort can shut down an animal facility for months, and no one seems to think twice about it. Well, if we really want to meet this kind of a 100-day challenge, then there must be a robust green signal from institutions, their nodal ministries and everyone else. So this is not an area you can mess around with, because this is critical for defense and for public health, for defense of the people. Can our regulatory system cope? Our regulatory system, we must remember, is something which has an enormous set of constant challenges, and its revamping will naturally be a never-ending process. Science changes, expertise changes, and response changes, and most important, response depends on context. There are three different pushes which regulatory systems need to contend with. One is from industry. They need to get their products through the system. and they will, keep pushing and asking for approvals to be done. Is there rigor in responding to that? The second is from public health people who are coming with novel compounds. Each one will claim that they've done all due diligence thoroughly and their compound or their vaccine needs to be gone through. And thirdly, as technology changes, as disease changes and disease prevalence changes, their responses to each of these other two will have to be calibrated because it's the context which matters. There are situations where going slow is not a problem. There are other situations where going fast is important. So one must really work constantly, even when one is, with industry or academia or in public health, to strengthen regulatory processes, agnostic to whether one is specifically involved oneself or not, because if you don't do that, if the systems are not strong, we are the ones who will be in trouble. Can the implementation system cope once vaccines go through our regulatory system? Is our rollout of vaccines, and because this will effectively be a trial with millions of people involved, can one look at adverse events? Can one look at effectiveness and efficacy in this context and understand that? And there, during the pandemic, India has had extraordinary experience in doing that. And the question is, can we have a system to have information transfer at that stage, have clinical trials going on at that stage with different patients who are hospitalized and respond very rapidly, both to vaccine effectiveness, but also other kinds of therapeutics. Finally, let's take the case of tuberculosis. If one wants to take available vaccine candidates through trials and fruition now, that will itself cost, globally, in the range of few $100 million, if not $1 billion. There are extraordinary tuberculosis research work going on in India. And if they come together and if they have multiple vaccine candidates, they will all require huge funds. And tuberculosis is very important in India. Where are those monies going to come from? So that's again a very big challenge. Both national and global agencies will be very stretched in the context of a disease X, or even otherwise in choosing between, let's say, tuberculosis and dengue and chikungunya and malaria and so on and so forth. Each of this is very, very expensive. So resources are not a small matter. So let me end by saying, here is a way out, and sorry for the typos in the second paragraph. So instead of asking, whether the CEPI 100-day challenge can be met, can we have the THSTI meeting have a challenge on the CEPI challenge? Can we, in 365 days from now, be ready to meet the CEPI 100-day challenge? For that, we need to go over each of the points which CEPI has listed out and see whether we have enough net practice and make sure that we're up to speed in each of them. How is that feasible? And the answer came from, in part, from Dr. Pramod Garg's talk. If you look at the extraordinary capabilities of THSTI and RCB, and if you look at what I said about the National Capital Region, there is in principle no problem in this whole region collaborating, choosing, to get certain kinds of platforms ready, or choosing one disease or many diseases and addressing that. And what is true here is true for Pune, for Bangalore, for Hyderabad, for the Chandigarh region, and many other places. So it is critical, if these kinds of challenges are to be addressed, that our national laboratories and our universities work together. If you have a map of the ICMR labs, the CSIR labs, the DBT, the DST, and the Defense Lab all over the country, they cover every part of the country. If you have a different map of the central universities in India, they cover all the country, and the ISAs and the AIMS. And if you have a third map of the state university, it's just packed. The entire country is packed and there's no space. But each of these layers don't talk amongst themselves and don't talk to the others. In that situation, if we then are forced to adopt a Western model of the costs required to do anything for any one disease, we are doomed to failure. On the other hand, if we collaborate with far less resources, we can address far more complex problems. Keep in mind that the entire research budget of one pharma company exceeds the entire US National Science Foundation budget, right? The entire research budget of one big American universities exceeds India's entire science budget. Now, these are big mountains, and if you try to climb these mountains, we will never be successful if you try to imitate what needs to be done. But if you know what the outcomes and goals are, there are easier ways. They're still very difficult, but they're much easier than climbing the mountain. There are easier ways to negotiate this for far less resources and go ahead. And for that, we have to work together. Thank you very much.
Thank you, sir, for honoring us with your presence and for delivering the keynote address. May I request our director, sir, Dr. Pramod Garg, to felicitate Dr. Vijay Raghavan. May I also request Dr. Pramod Garg to felicitate Dr. Sudhanshu Vrati. Thank you, sir. We have a quick tea break here, and I request everyone to be back in 30 minutes. In the meanwhile, I would request all the speakers for the first session. Okay, so let's be back in 15 minutes. So in the meanwhile, I would request all the speakers of the first session to hand over their presentations to Dr. Shankar, Dr. Priyanka, or the technical staff on the dais. Thank you so much. We will now begin with the first session on accelerating vaccine development. To chair this session, I would like to invite Dr. Narendra Kumar Arora, the Executive Director at the Inclin Trust International and Chair of NTAGI, National Technical Advisory Group on Immunization, to chair this session. Professor Arora is a well-known public health expert in India, and he is extensively involved in policy making, program planning, implementation, and evaluation of public health programs at both national and international level. I would like to request our director, sir, to welcome our session chair with a planter. Thank you, sir.
A gentle reminder that each talk will be of 15 minutes, followed by 5 minutes of question and answer session. And at the end of each session, there will be question and answer session of approximately 20 minutes.
Welcome, friends. Good morning, and it's a very timely meeting. Is it working? Yes. No. Yeah. Good morning, friends. And in fact, some of the challenges Professor Vijay Raghavan set for and the challenges to achieve 100-day benchmark by Professor Vijay Raghavan, I think we will try to get some answers in this session. And how things can be made feasible and what are the still lingering barriers. In fact, I was just mentioning to Professor Raghavan, is 365 days the right, or can't we reduce it further? So we will begin with this session, and I will request all the presenters to restrict themselves for about 15 minutes. And then we will have discussion, because discussion and interaction will be a key part of this session. May I invite Professor Bernard van der Ziest. I'm sorry for not pronouncing it properly. Leiden University Medical Center, the Netherlands. Professor has been working in the field of vaccine development and production for last 4 decades, and he played instrumental role in advising the Dutch government on vaccine purchase, communicating the importance of vaccination to general public during the pandemic. And from 1997 to 2009, he was director of vaccines of the National Institute of Public Health and Environment of the Netherlands and scientific director of the Netherlands Vaccine Institute. Professor Bernard Ziest. I understand he is online. Welcome, sir. The floor is yours.
Thank you. Can you properly hear me?
Yes, we can hear you.
Okay, then I will start to share my screen. Okay, well, thank you very much for the invitation. And I'm looking forward to hear more about the many interesting research projects going on in India in the coming days. What I would like to do is a little bit extension of the keynote, looking back and then taking lessons to speed up vaccine development. So we now, hopefully after 3 years at the end of the COVID pandemic, and this was a very heavy period, about 7 million deaths and 13 trillion economic damage, which is has many zeros, as you can see. Vaccine development, to start with, was very fast. Already in December 2020, the first vaccines were approved, and that was unprecedented if you look back at the average development time for vaccine, which is several decades in many cases. In the end, there were 3 vaccine classes that were used most: the classical inactivated viruses, a completely new platform, the messenger RNA vaccines, and vaccines based on the adenovirus platform. There was already some experience with that, but that was still limited. And these messenger RNA vaccines and also the adenovirus vaccines contained the spike protein of the virus, and it was very important that it was frozen in what we call the pre-fusion conformation, which is different from the post-fusion conformation. We have presently 11 vaccines which are widely used. You can see them here on the website from WHO. And the global vaccination rate is about 70% at this moment. Having a vaccine does not mean that it is immediately rolled out, and that is one of the problems. I have here 2 pictures of rollout in European Union on the left, and in Argentina, and in most countries, the rollout after the vaccine became available was between 3 and 6 months, and in developing countries even longer. And that was due to 2 factors. The suppliers had to gear up their production facilities, their capacity, and also the governments were not ready. The infrastructure was lacking to administer the vaccines. And finally, the messenger RNA vaccines had to be used at a very low temperature. They had to be stored at a very low temperature. And of course, when the high-income countries had made all kinds of contracts, but that meant that the lower middle and income countries lagged behind. COVAX was established to solve this lack of access, but it was not completely solved. And also, in some countries, as you can see on this map, the darker, the more people are vaccinated. In some countries, the vaccination rate, even at this moment, is still low. And the factors are not completely known for that. So if we look back, what can we do to speed up vaccine development, maybe to 100 days, and then development is one thing, but a rollout is another thing. So that is a matter of definition. This meeting is very timely. Worldwide, many countries are now looking back and are preparing for possible new outbreaks. And as I will discuss later on, there is a real chance that there will be another outbreak. It's probably another viral outbreak, but it may be not a coronavirus, and it makes a big difference in the possibilities to speed up vaccine rollout. I have identified 8 areas where improvements are possible, and I will very shortly, because of the lack of time, discuss those. And in addition, it's very important, it was already mentioned in the keynote, that we look out at the economic costs of all these preparedness measures and see what is possible and what should be done. The first point is that we have to understand the correlates of protection, how vaccines protect better. We know now that antibodies are made, and the antibodies prevent vaccines entering the cells. And that's and we know very well what level of antibodies are required for protection. But another very important contribution is made by T cells. They kill infected cells, and these are supposed to contribute mostly to the more long-lived protection against the severe disease. And there's still a lot of work to do to understand these T cells and to measure the contribution of the T cells. And then also we saw some very rare adverse effects in the messenger RNA and adenovirus vaccines, which were not detected in the phase 3 clinical trials because the numbers were too small. And these were acceptable at that time, but preferentially we should solve them. The next thing is that we have to harmonize laboratory procedures to measure the immune responses. And if we want to approve in the future vaccines on the basis of immune correlates, we have to have reliable methods, and they should be all give the same result in different laboratories. And so this requires harmonization between laboratories. Then how can we develop the improved vaccines, the licensed vaccines, sorry. The problem we met that was that the variants escaped immune responses by the vaccines. So we had to adapt the vaccines. So one approach would be to make broader protected the vaccines. And there are several possibilities. We could improve the conformation in the prefusion states. We could also take up non-spike T-cell antigens in the vaccines. And I know that vaccine companies are already working in this field, and also CEPI is funding many projects on this topic. Then there are some new developments on the horizon. One of them is live attenuated vaccines. And these are also much easier to produce. The problem of course is reversion to virulence. And the other development is nasal administration, which already has shown to be better for immune responses. And even two vaccines have already been approved, but more research is needed. Then there's still a lot to gain in shortening the clinical development, because that takes up most of the development time. If you look at the example of the BioNTech Pfizer vaccine, the preclinical development time was 3.5 months, and the clinical development time was 8 months. And if we can use immune correlates of protection, then this would be shortened considerably. And alternatively, a proposal has been made to use human children's trials, but this of course has some ethical implications. Then regulatory improvement, there's still a lot to gain also there. We already had the rolling reviews, and that accelerated the approval of vaccines considerably, but further streamlining is possible. The EMA has taken a step by establishing a special task force, and more international collaboration is also possible. The International Coalition of Medicines Regulatory Authorities is already working in this field, and this will bring top experts together from countries and save time and work. Production capacity is, of course, also key. At this moment, there's a lot of capacity to produce the vaccines, but that will probably stop. And the only way to solve this is to make agreements between public health entities, governments, and vaccine producers, and to have access when needed to production capacity. And what we see already is that the USA and the European Union are taking initiatives on this field, and this should also happen elsewhere. Rollout, as already mentioned, that is still a problem. The infrastructure, but also a lack of confidence, which is currently not the case in India, is a problem. But the problem is mainly occurring in Africa, where only 24% is now vaccinated compared to about 70% worldwide. So I don't have to argue that there is some need for action. There is a chance of new pandemics. We were very lucky that this was a mild virus, but we don't know the R0, the case fatality rate, and incubation times of a new virus when it would have occurred. And then, of course, it has been shown that vaccination is key to stop a pandemic. Antivirals are on the horizon, but they take longer to develop. Of course, who's paying for this preparedness? And that depends on two topics. The chance that we have a new pandemic, that's of course almost impossible to predict. But some attempts have been made, looking back at many other pandemics in the past, and this prediction comes to 38% in one's lifetime, but that may go up. It probably will go up because of the increased mobility, population densities. So it may go up to once in a lifetime, and that means that you cannot ignore it. There's little bit of economic evaluations, and these indicate that putting money in preparedness is cost saving, but that has to be looked at better. So what I show here is now a summary, say a kind of to-do list on these 8 points. And I won't go through it again. But of course, it's very efficient, as already was mentioned in the keynote, that we look at the cost benefit of the measures. So I'm very optimistic in reducing the development time of another coronavirus. But of course, it would be a different story when it would be a completely other virus. And then it is more complicated, would require a lot of out-of-the-box thinking. But some work can be done in advance, and already WHO has taken the initiative by making a blueprint, and there are already some roadmaps developed for some viruses and for some other viruses, among which is pathogen X, the most difficult culprit. It's under development. So I wanted to share this with you, and the last slide is only the literature references that were given in the previous slides. Thank you for your attention.
And thank you very much for that very nice overview of how things were done during the COVID pandemic and what are the remaining challenges, and developing a roadmap for pathogen X. We are open for we have about 7 minutes for question answers. Any queries, clarifications, questions from Professor Bernard? Yes, Shelja. Mike.
Good afternoon, good morning, Professor Bernard, and we really are sorry that you couldn't be here with us, but we'll be in touch. My question is, you talked about there are already roadmaps for a few diseases which WHO has made, and some are under preparation. Do you think these sort of roadmaps would help in diseases which concern India? And should we be looking at creating these roadmaps for India? Whether it's TB, malaria, sir talked about TB, malaria, dengue, chikungunya, would that be a way forward?
Yes, I think so. Of course, it will be very important to know how protective immunity develops and what the levels of antibodies or whatever of T cells are for protection, because then you know when you test vaccines whether they probably will be protect without carrying out large clinical trials. Clinical trials may be needed still, but during the development road, this will be important. And also, of course, you can put say diagnostic methods in place. So there is a lot of work already been done. So preparation is key. And so you don't have to do that when it's something new happens. Is that an answer to your question?
Yeah. Any other? Professor Bernard, in fact, you highlighted one of the highlight of your presentation was collaboration between researchers and industry, between researchers, and then between industry and government and regulators. Can you help us identify where these barriers created delays or some of the inequities you talked about?
Well, of course, somebody has to be in the lead, has to coordinate, because if it's a loose collaborative network, it's not as less efficient. And what we see now in Europe, because we had many different in the Mexican flu, you may still remember that the countries acted on their own, but now the European Union has taken the initiative, and people and countries are working together. So also India could take the government of India could take the lead to collaborate with countries in the region and establish a network to plan, to carry out a plan, a predefined plan.
Thank you. Yes, Professor Raghavan. Yeah.
Thank you very much for that very nice talk. You mentioned the EU starting a BARDA-like structure. What is the rough time scale for that to start? And what's the kind of thing which will be there for this? And secondly, would that kind of a structure along with BARDA be sufficient for the world? Do you think yet another structure, say anchored in Asia or Africa, is needed?
Yeah, I think the more regional, well, the European Union is already working together with BARDA, say, looking at best practices. But I think that there are more regional networks necessary. We already see that Japan is taking an initiative, Australia is taking initiative, but Asia, with its very strong pharmaceutical industry, could be in India particularly, could be an excellent candidate to do that. So that would be my advice, but I'm of course not an expert in the region.
Okay. Yes, please. Please identify yourself also.
Thank you, sir. I'm Dr. Hans, Bangalore, from the Department of Neurovirology. My question is, does the WHO blueprint address the issue of vaccine hesitancy?
Vaccine what?
Address the issue of vaccine hesitancy, or like improve confidence in vaccines before the next pandemic hits us?
No, not in that sense, because vaccine hesitancy is very local, and it's a culture. It has to do with the confidence of people in the government mainly, and so this is something that you cannot do globally. It has to be done locally. But there are some experts in that work on vaccine hesitancy, especially in the London School of Hygiene and tropical diseases.
Thank you very much, Professor Bernard. We will request you to stay on as we will have a kind of a question answer session towards the end of the presentations also. Thank you.
Yeah, okay, thank you.
May I now invite Professor Ramarao Amra from Emory University, USA. He will speak on accelerating the development of viral vector-based vaccines. Professor Amara is Charles Howard Candler Professor in the Department of Microbiology and Immunology in Emory Vaccine Center. His research is focused on the development of vaccines for infectious disease such as HIV, SARS-CoV-2, HCV, and tuberculosis. And his lab has pioneered the heterologous prime boost vaccination approaches using DNA modified vaccinia ankara and novel protein immunogens for inducing robust immunity. Professor Amara.
Good morning. I'd like to thank the organizers for giving me this opportunity to come and speak to you all. It's always a pleasure to come back home and see friends and talk to all the young scientists. Before I go into my talk, I'd like to just add one more point to what Professor Vijayaraghavan said. I completely agree with the points that he was raising. One other thing I want to say is that we were so fortunate that the spike protein of the SARS-CoV-2 was so immunogenic and was able to induce these high titers of neutralizing antibodies. So that's not the case for many viruses. So that one other thing that we need to keep in mind, that we don't know what is going to be like the new pandemic virus that is going to come across. And if we are not that lucky again, so we'll be in big trouble. Okay, so and also I'd like to say that my laboratory made multiple vaccines, but I don't have any manufacturing experience. Since the crowd here is going to be a mix of both research scientists as well as the industry participants, I kept my slides to very with the minimal data. So I hope that would be okay. And the goal of our laboratory for the long time has been to understand the pathogenesis and immune correlates for protection against HIV, with a focus on developing a vaccine to prevent HIV infection, as well as immunotherapy to cure HIV. And more recently, we started going into vaccines against COVID-19, influenza, hepatitis C, as well as TB. So TB is my kind of first love that I did my PhD on tuberculosis. So I'd like to get back to TB soon. When we are talking about protective immunity at the portal of viral entry in the mucosa, we will need neutralizing antibody responses. If these are essential, so if you look at any vaccine that has been successful so far is, the most of them you would see it is protection is mediated by neutralizing antibody responses. It doesn't mean the T cells are not important, they are equally important. And the one the virus gets across and establishes an infection, then you have various other functions of the antibody like phagocytosis, complement-mediated lysis, or ADCC. All these functions also contribute to protection. So now there are more studies coming out showing that the importance of these kind of activities for protection against SARS-CoV-2. So although the virus is evolving away from the neutralizing antibody responses, these other functions that are quite conserved, and they're actually helping with vaccine-mediated protection. And then you also need tissue-resident CTLs, which are also important. And then accordingly, the vaccine protection, to induce vaccine protection, you need to induce these highly cross-reactive neutralizing antibodies. You need these cell-targeting antibodies with effective functions, as well as TRMs. And then there is a great need for, there is a great synergy between these various functions. And to get a strong antibody response, you need these high T follicular helper CD4 T cells. And then importantly, these responses should be homing to the mucosal site. And for some vaccines, you need Th1 biased CD4 T cells, for example, TB, whereas for vaccines like HIV, those can be detrimental for efficacy. So you need to tailor your vaccine to see which kind of responses you need. So as we know that the antigen can be delivered in using multiple platforms, recombinant viral or bacterial vectors, live attenuated vaccines, killed whole vaccines. You can have purified proteins, polysaccharides, DNA, messenger RNA, and a combination of any of these. These are called heterologous prime boost. So why do we? So when we have these many choices, how do they differ? They really differ in their ability to induce these antibodies, CD8 and CD4 T cells. For example, live attenuated vaccines give you a balanced response. They give you antibodies, CD8s, and Th1 biased CD4s. Whereas if you use the killed whole vaccines, you predominantly get the antibody and the Th2 CD4s. Like this, that they really differ. And for example, DNA vaccines give you low levels of all of these responses, the same, and the mRNA vaccines give you very high titers of the antibody and the low Th1 responses, not much CD8 T cell responses. If you use a combination of these, you can get everything. And then each of these platforms are associated with their own limitations. With the live attenuated vaccines, there is always a danger of reversion to virulence. There could be contaminating live agents. The same is with the killed whole vaccines. If there is an incomplete killing, there is a problem. And then the proteins, they need adjuvants, they need to be purified. And the recombinant viral vectors, some vectors, there is a pre-existing immunity issue. And then mRNA vaccines, that LNP acts as an adjuvant. So you need to pay attention to what kind of LNPs that you need to use. And then with the heterologous prime boost, you need 2 agents to vaccinate. So focusing on the live attenuated viral vectors, there are a number of viral vectors have been used for vaccines, and including pox viruses, as well as adenoviruses and the VSV, influenza and polio. And then today I'll focus my talk on the pox viruses, but how do you choose which vector to use? So you need to pay attention to the safety record, immunogenicity. The vector should induce the high magnitude, quality, and longevity of the immune responses. The vector should be able to carry large genetic material so that you can put more genes into it, and it should be easy to manipulate. It should not have pre-existing immunity issue, can be grown to high titer growth in vitro, and then it should be easy to manufacture. So we worked on pox viruses for a number of years, and then I thought I'll briefly talk to you about that. And then one of the important features of the pox viruses is that a single dose of vaccinia virus, for example, a vaccine can induce antibody and T cell responses that persist more than 70 years. So here are these 3 papers that were published around early 2003. One from Mark Slifka's lab, and one from Rafi's lab, and one from my lab. This is the data from Mark Slifka's lab. Here you're looking at the years after vaccination, the antibody responses. You can see after single vaccination with vaccinia, you can still detect antibody responses after 70 years. It's the same is true even for interferon gamma positive, TNF positive CD4 T cells. These cells can easily be detected. So this is the kind of vaccines that we basically need. So a single dose, if it can do that, it's really great. So one of the pox viruses that we are working with is the modified vaccinia ankara, which was actually derived from the vaccinia virus, which was developed as a vaccine for smallpox by passaging serially in chicken embryo fibroblasts. It lost parts of its genome, including the immune evasion genes, and it has an excellent safety record. And then there are so many advantages for the MVA vaccines. They're safe, well tolerated. They induce strong antibody response after single vaccination, and the immune responses are durable. In humans, we have durability data up to 6 months. In non-human primates, we have up to 3 years. I'll show you that data. The MVA can be delivered through multiple routes. In the recent study from Bernie's lab, that they showed that with the intranasal vaccine, you actually need 100 times lower dose than the intramuscular vaccine that you use. And then MVA can accommodate large inserts up to 10 kb size, so you can put more genes into it. The recombinants are quite stable, and then they induce both CD4 and CD8 T cell responses. And then importantly, a single dose of MVA has been shown to protect against multiple viruses, including SARS-CoV-1 and SARS-CoV-2 and Ebola, Zika, and MERS. So we have been working with a DNA MVA-based vaccine for many years, way back, starting from my postdoc days. And it also highlights that how hard it is to really develop a vaccine for HIV. And then this vaccine finished phase 2A studies in humans, including HIV-infected people, and showed excellent safety and immunogenicity. And then more recently, we started developing a new approach where we prime with the MVA and boost with the protein. This is done in collaboration with Raghavan Varadarajan's lab at the University of Science, who is also here. In this vaccine platform that we showed now in 3 different monkey studies, where we prime with 2 doses of the MVA and the single protein boost, and then showed a heterologous protection in non-human primates where we protect anywhere from 40 to 60% of the animals after multiple repeat exposures intrarectally. And then this is another study that we, as a continuation of the clade B study, we also developed a clade C vaccine, and that also showed great promise in the non-human primates. So when the COVID pandemic emerged, with all the experience that we had, we, like everybody else, we also developed MVA-based SARS-CoV-2 vaccine. We did this in 2 phases. In the early phase, that we used the stabilized spike and non-stabilized spike, and then more recently we used a stabilized spike as well as the nucleocapsid. So this could be one of the very few vaccines that where people have used the nucleocapsid in addition to the spike. And then furin cleavage we showed in this study, furin cleavage inactivation can actually improve the antibody titers by 50-fold. So here is a comparison of the vaccine that is expressing the regular spike followed versus the furin cleavage site inactivated spike. And we see much higher, just after priming, we see 50-fold higher binding antibody. After the boost, we see similar results. More importantly, now a single dose of this improved vaccine induces titers that are higher or comparable or higher than the 2 doses of the first version of the vaccine. That also reflects in higher neutralization titers. So now this vaccine also has the nucleocapsid, and now we are putting more genes into it, including the non-structural proteins, so that we can actually broaden the protection, if at all, that we really need to vaccinate people against COVID-19. And it so happens that this same vaccine also works against dual vaccine for monkeypox, because the backbone of the vaccine, MVA, is the pox virus. So the vaccine induces immunity against both COVID-19 as well as the monkeypox or smallpox. And then in 2007, we showed this, that a single dose of MVA can actually provide protection against monkeypox in macaques. So this was done by various labs those days, and this is our publication. And then in that study, what we saw was the vaccine-induced responses were actually durable for over 3 years. So here you're looking at the antibody responses induced by our DNA MVA HIV vaccine. These are the cross-reactive neutralizing antibodies to MVA. So you can see after the second MVA, they come up. When you look at the responses after 3 years, they're still persistent. The same is true for the CD8 T cell responses. You can even after 3 years, these are persistent. So again, it shows that the MVA-based vaccines, just like the vaccinia, that they can induce durable immune responses that can persist very long. And then more recently, we also developed an improved vaccine for the MVA, so whereby we introduced the anti-apoptotic gene B13R, and then that also further enhances the humoral immune responses induced by MVA. So now we are working towards taking this vaccine into India and for manufacture, and then set up the manufacturing process here. So like everybody, we learned a lot of things during this pandemic process. And then I wanted to share a couple of points that what we learned. So one thing we did was that we shortened the time for selection of these recombinants. Normally it takes 3 months. We shortened it to 3 weeks. So what we did, instead of using the conventional plaque picking protocol, we moved to a flow cytometric based sorting process. So what this allowed was to purify the plaque every day instead of every week. So that really accelerated the development of the vaccine development. And then we also developed a new method to stain for these intracellular proteins. If you're going to pick for the recombinants that are intracellular proteins like nucleocapsid, you need to fix the cells. And when you fix the cells, that the virus is also going to die inside the cell. So you can't select these based on the flow cytometric method. So we actually came up with a protocol where you don't kill the virus, but yet you can still stain for the intracellular proteins. And then we started our vaccine development towards the end of March 2020. We actually started very late compared to everybody. And then we completed our efficacy in non-human primates by the end of August 2020. So that's how fast we could make the vaccine, we could test and show that it was protective. So we could do it that rapidly, and we actually licensed it to a company in India in September of 2020. So not in India, in the US. So we can make these vaccines pretty fast. And then one thing that we realized during that process was that we needed a cell line to really grow the MVA to high titer. Conventionally, the chicken embryo fibroblast cells are used to production of MVA. However, if in a situation like pandemic, that you can't rely on CEFs. So we screen now, so that's why we invested a lot of time in the last 2 years. And then we screened multiple adherent cell lines and identified one that really supports high growth of the MVA. And then we are currently characterizing the cell line, also a suspension version of the cell line for high titer growth. So with that, I'd like to thank people that did the work, and today, whatever work I presented done by my former grad student Andrew Jones and Shailaja Gangadhara in the lab, and Andakishore Rautu and Anusmita Sahu. And then our HIV work is done in collaboration with Raghavan Varadarajan's labs. With that, I'll take questions.
Thank you very much. Yeah. Questions or clarifications? Yes. At the back, I see two hands.
Very nice presentation. So you have shown the live virus neutralization data for the COVID-19. Is it against Wuhan, or have you checked for the variants of concern? Because that is one of the major problems for RNA viruses, because the virus is evolving.
So the publication that the recent publication where we show that the Wuhan spike can induce very strong cross-reactive neutralization against the Delta variant. So we did not test against the other variants yet.
Thank you, sir.
Yes, at the back, please.
So this is regarding the TB vaccine with antigen 85 in MVA. So this was somewhere in early 2010s and 2020s when a huge trial went on. And basically, while the preclinical data was excellent, the human trial data turned out to be a disaster. What do you think is the problem with respect to the MVAs?
So I'm not sure that how strong is the preclinical data with MVA by itself. For TB, what I would recommend is to use a heterologous prime boost with the DNA and come in with the MVA. That's when you get the highest levels of your CD4 T cells and CD8 T cells. MVA by itself does induce good T cell responses, but they are not as high as I would think that the TB vaccine would need. So you need a DNA prime and MVA boost, or MVA combined with another viral vector.
Yes. Second row in the center. Yeah.
Yeah, my question is, heterologous prime boost. It's really very good. I mean, you get the best of the two worlds. But what do you feel? I mean, practically, you have to have two different pipelines to produce the vaccine and the logistics to immunize. How practical this situation can be?
Yeah, there are challenges in terms of the economics, but I think when you are trying to develop vaccines, you're not going to rely on one particular platform anyway. So for example, in India, people used the adeno-based vaccination for COVID. You could come with MVA-based vaccines for boosters if you need to, if you're limited in being able to go back and boosting those same individuals with the adeno-based vectors. And the MVA-based vaccines can have been used very well with pretty much everything. So you could use with the DNA prime or RNA prime or adeno prime, or any combination that people have used, that the MVA vector is an excellent booster as well. So I think that ultimately you might have to use the heterologous prime boost protocols.
Hi, I'm Dr. Shomodip. I work at the Tata Medical Center, Kolkata. So it was a nice talk, but I would just have a few questions on what we were discussing just now. So you told us that the MVA...
Where is the... Okay, sorry.
So you told us that this MVA-based vaccine, the Wuhan strain and the the spike of both the Wuhan and the Delta had produced similar neutralizing antibody response. So my question is, if that was so, why did Delta create havoc? Where were we? Number one. Number two is that what do you think is the way forward? Because with Omicron, we know that it's mutating very fast. And we all accept that the vaccine needs to be updated. Probably, just like influenza vaccine, we need multivalent vaccines against the circulating strains. So is that possible with an MVA platform?
Yeah, I didn't get your first question. So so we induced.
Yeah, if the response was similar in the preclinical trials, why did that not translate into human? I mean, why couldn't it protect our masses when it came to the same vaccine being used during the Delta pandemic across the globe?
No, so the MVA has not been used in India yet, right? So, but I'm talking.
So what was the barrier to, because the other vaccines have been shown, because with the Covaxin, we know that from my alma mater AIMS, the Covaxin data came out. And we know that then there was a lot of discussions regarding spacing of the introduction of vaccine, whether it gave immunity. There was a huge debate of how the papers got supported and all. I'm not going into it, but why did the MVA vaccine not come into market when the Delta pandemic was raging across the globe and we could not address it? Because in future also, we'll have to, if we are thinking of removing the roadblocks of what actually prevented us from translating it into clinical practice or from protecting the mass, we need to know what was the roadblock to remove that roadblock.
So I think the roadblock was the manufacturer having the right cell line available for the manufacturer. So that's where we were investing more time.
Okay, I think that answers, because that was one of the limitations, the replication of the virus and the cell culture system. And I think that would be the limiting thing. Can we move on to the next one? Thank you very much. And there are a few other questions, and I think we'll take it up in the common session at the end. Thank you very much.
Thank you.
Next, we have Dr. Umesh Shaligram from Serum Institute of India, Pune. And he is Executive Director and Board Member of Serum Institute of India Private Limited. And he spearheads the scientific development of vaccines and has been particularly playing a very important role in the COVID-19 vaccines. And he's also associated with generation of next category of biologicals, including biosimilars and biobetters. Umesh Shaligram.
Thank you very much, sir. Am I loud enough? Are you able to listen to me very properly, or?
Yes.
Yeah. Okay, great. Thank you. My presentation, you want me to upload, or it will be uploaded by your team? I can do either way. Just wanted to know.
Yeah, Shailja, so you have the permission, you can upload.
Yeah, I'll just give me a second. I'll upload my presentation. Can you see my slides? Is it visible? Sir, are my slides visible?
There is something, somebody is working. Yeah, there it is. Thank you.
Great, thank you. So thank you very much. First of all, my sincerest thanks to Honorable Chair, Professor Rajesh Gokhale, Co-Chair, Dr. N. Karora, and Professor Garg and Dr. Shailaja for inviting us to such a prestigious international meet and allowing us to give an idea about what we are trying to do as a Serum in terms of our future pandemic preparedness and what up till now we have contributed. Before I start my talk, I just want to say my sincerest apology. How much ever I wish to travel to Delhi and personally come, not only about this meet, but I wanted to meet Professor Garg and look for future collaborations, but I couldn't do due to health issues. So I'm really sorry. And I also wanted to meet Professor K. Vijayaraghavan, who played a pioneering role in our early days of COVID-19, especially getting this stockpiling, manufacturing stockpiling, filing license ideas and getting that through the government system. It was a key role without which probably we would have struggled hard to vaccinate whole of the country. So, it's really wonderful to see him personally there. I'm really sorry I'm missing him. It was quite an interactive early days in COVID-19. We interacted with him and he supported all of us pretty heavily on that. So with that, I probably will give a bit of overview about Serum. I'll just give a little historical perspective about Serum. So, and then we will take how we are trying to really see how we are preparing ourselves for future pandemic actually. And my previous two speakers, Rama Rao and Professor Bernard, has given an excellent talk and has really set a right forum for me to really give this talk. Thank you very much. So next slide, please. Yeah. So Serum is in existence in Pune since last 5+ decade actually, and we are known to be LMIC supplier, and we have supplied huge amount of vaccine globally, and we have really contributed substantially in supplying several vaccines cost effectively. That's the entire goal of Serum, is to supply cost effective vaccine and really address the disease and eliminate the disease. When we check back our past contribution, we have found out that we have almost given more than 25 billion doses up till now, got manufactured here in India and got injected into majority LMIC countries and now in a developed world also, including COVID-19. And these 25 billion doses of several vaccine has translated into saving millions of life actually, especially Menafrivac, complete kind of elimination of NA in sub-Saharan African countries, measles, saving more than 24 million deaths due to the measles, rubella, rubella eradication in Pan-American countries, and uncountable lives probably has saved because of our diphtheria, tetanus, pertussis, Hib, anti-snake venom, anti-seras, and miliary TB due to BCG. So clearly, everyone knows vaccines saves millions of lives, and we have been able to contribute by supplying billions of doses in a short time, cost effectively a vaccine. That was a major challenge during COVID-19 pandemic. In COVID-19, we have manufactured almost close to about 2.2 billion doses and supplied almost about 2 billion doses of 2 vaccines, Covishield, which contributed quite a significant supplies to lot of countries' vaccination and our country's vaccination itself. More than 88% of Indian adult population have got the COVID-19 Covishield vaccinated. And Covovax is the one vaccine which is manufactured in India and supplied to all developed world like US, Europe, Australia, New Zealand, except Japan. Everywhere Covovax, which is our Novavax collaboration vaccine, has been supplied. We also developed an anti-sera, which is yet not got license, but it's a very high titer anti-sera, which we found that it's a unique idea to really make some therapeutic, cost-effective therapeutic available for LMIC. And we also developed a recombinant BCG VPM1002 as an immune boost, which we are getting into a next level of marketing authorization. We are compiling up the data for that. So we put a lot of efforts around COVID-19 and really supplied, translated, manufactured a huge amount and supplied to countries across the globe. Actually, we are the only one who has supplied, except Japan, almost every other country in the world a vaccine. Our leadership has contributed this by really supplying these vaccines cost effectively, and it is estimated by Stephen Kaufman and all, which is published in Human Vaccines, that more than 30 million lives have otherwise would have perished has got saved due to such a cost-effective large volume vaccine availability. So this is a visionary efforts of translation by our leadership. We are located in India, Pune. Near Pune, we have 2 sites. One is Hadapsar and Manjri sites. And this is an overview of our Hadapsar site, overview of Manjri sites. And that's where, our next part of the talks comes into. We're the only one in the world who has already made a future pandemic preparedness facility. So there is a large facility which is ready. There are about 70,000 square feet, 5 floors, which are already built. And you can see that building ready, actually. And these 5 floors, each of 70,000 square feet, And I'm very happy to see that some of you have already visited these facilities, have an ability to really adapt to different technologies and quickly translate. The major challenge which we saw in COVID-19 pandemic was one thing, and that was we had to sacrifice a lot of our existing manufacturing area. We had to put down a lot of our existing product and delay the development of existing product and focus on COVID-19 supply. Now, learning from that, we already created an infrastructure, which is probably first time in the world and has an ability to really manufacture about 5 to 10 billion doses in less than a year's time with every technology which you can have in future coming up, like mRNA, viral vector, live attenuated viruses, inactivated viruses, as well as recombinant proteins and novel adjuvants actually. So we have been able to create an infrastructure which can really translate quickly and really make this huge amount of doses available. And time being, this infrastructure is utilized for existing vaccine, but the agility and ability to change over is built in such a way that a quick translation, quick scale up is possible in the existing future pandemic facility, which is ready under qualification. Also, in order to address this future pandemic preparedness, what we are thinking is we are trying to access all, we are really well versed and very well skilled into manufacturing of a lot of vaccines. So we have decided to stockpile and standardize methods of manufacturing. Like right now, Ebola outbreak exists in Africa. We have immediately quickly translated this, made a vaccine, and now shipping in a few days. You will listen to news that we have shipped the Ebola vaccine. A very fast translation, India contributing to the LMIC countries outbreak very short, in a shortest possible time actually. So we are shipping our vaccine to WHO and it will be utilized mostly in Sudan and Uganda, wherever there is outbreak. And we have decided to stockpile all these different vaccines like Ebola, yellow fever, Lassa, Rift Valley fever, West Nile disease, Nipah, monkeypox. All these things are, we are standardizing the technologies and qualifying these technologies so that we have some stockpile and we can then scale these up anytime whenever we needed, if there is a chance of a future pandemic. So whole idea is to really prepare ourselves with already known disease causing viruses, bacteria, and try to really be well prepared about it and take the things forward very properly. We are also translating into mRNA-based technologies, which is little unique with the thermostable mRNA technologies, and we are keeping ourselves ready with that also. So in case if anything needed, we can really quickly scale up and supply through that technology also. So every other technology, every other disease, we are trying to really understand an ecosystem of that entire disease and trying to really provide a solution around that ecosystem. So that ecosystem, what we are trying to come out with an innovative strategy in order to really come out with a solution of disease elimination strategy, what we are calling it as a detect, treat, and prevent strategy. So in terms of detect, treat, and prevent strategy, we are really trying to address this. And we have really put a lot of efforts around TB, first to start with, and then subsequently malaria. In TB, we have come out with a unique point of care detect strategy, then a treatment, which we will access through a classical rifampicin isoniazid as a treatment, and then prevent through a prevention of infection vaccine like recombinant BCG or BCG we are coming out with. So what we felt that with a unique track and trace tool, which is now available, COVID-19 pandemics has really brought in that. With the COVID app, Government of India has really done an excellent work to really show how the track and trace tool can really make a big impact in health. And with the detect, treat, and prevent strategy, every time we have a point of care detect, then treat, and then prevent, it would really bring a unique strategy in disease elimination ecosystem of diseases. So that's a strategy which we are driving. We have already put a lot of efforts on the TB and now have a unique complete solution around ecosystem of that. We and along with MyLab, our partners. Same thing we are trying to really, I already taken you through this, a unique strategy for the TB. Malaria also, we are trying to come out with a, certainly now we already have R21. We have completed a large phase 3 trial, and we are publishing the data shortly. But we have got a very good follow-up data with our R21 vaccine, but we are not stopping over there. We are coming out with a multivalent, multi-stage vaccine so that we can achieve a elimination strategy. So these are the efforts which are happening in each disease, also dengue, also chikungunya. So all these unique strategies which we are trying to really implement and create these strategies to really understand a complete ecosystem and make the solutions around it by not only providing the vaccine cost effectively, but give, detect, treat, and prevent solutions to the people with the track and trace tool. So this is what we are trying to come out in order to address our future pandemic preparedness, not only scale up the technology, not only supply to the globally supply the vaccines, but also come out with the unique strategy. This is what is the ideas which we are driving. And with that, probably my talk will not get completed unless I acknowledge all the efforts. But before that, with these strategies, we are also trying to really come out with newer solutions on cancers and HIV also. And my talk is not completed unless until there's sincere acknowledgements of all the people. This is impossible without millions of hands and millions of brains and millions of minds who have really supported us, and many of you are already over there who are listening to this talk, who have supported directly, indirectly. So our sincere thanks to all of you all. And with that, I pause my talk and I'm open for the question and answer.
Thank you very much, Dr. Saligram, for for that the futuristic component of what Serum is doing. And it is really heartening that Serum has played a stellar role in India's fight against COVID, as well as for future also there are on.
Hello. Yeah. So there was a pause. Yeah. Hello.
Please go ahead. Yes, please.
Hi, am I audible?
Yes, you are.
Hi, I'm Rama. Actually, I was interested, this is just a knowledge question. You mentioned that Serum Institute of India is trying to understand the disease ecosystem. I was wondering if you could list a couple of specific efforts, as in, are you actually engaged in collecting data in India? Because I think that's very important. Epidemiological, or is it more data mining and things? What are the specific efforts you're making in that direction? Thank you. Thank you for a nice talk, by the way.
Thank you. So I'll give an example. For example, what we are trying to do is, let's take a TB. So what in TB, we realize that, a lot of people are putting a lot of efforts, but there is no solution for the TB in terms of vaccine and elimination. And we realize that wherever there is a disease which has probably persistence of infection as a major cause of translating into a disease and a long duration exists, it's very difficult to, COVID-19, we were probably, I won't say lucky, but probably, as Dr. Ramarao said, that at least we had a spike protein and antibodies against that was protective and the disease was short and we could quickly see an efficacy, where efficacy trials are difficult. So what we felt as probably, you need to have a latency, you need to identify latency very properly. So we thought that what's a unique solution for LMIC is having a point of care latency identification. So instead of trying to depend on IGRA, we have come out with a point of care like there is a PPD test or Montague's test, but with a very specific recombinantly produced ESAT6 and CFB10, which is CEPI. And this CEPI is a point of care. So it gives an accuracy of IGRA, but cost effectiveness of a PPD actually. So, or TST. So you bring this solution first. So then you understand latency very properly. Then what you do is you create a track and trace tool with a, you detect. Then there is a in order to kill the latency, you have rifampicin isoniazid. So you try to create an access to rifampicin isoniazid. Luckily, in India, there are a lot of manufacturers who are making that actually. So then you have this solution, and then come out with a prevent solution with our recombinant BCG or regular BCG, or some other newer vaccine, which we think we can co-develop along with a lot of new efforts which are happening. So try to bring this understanding all these things. Similar way, in dengue, what you are coming out is same thing. In dengue, we are trying to really do a lot of research on the fronts which are related to what's a ground reality in terms of, and what's a quick way of detect, and then come out with a solution. So that's kind of effort we are putting.
That's great to know. Thanks so much.
Yes, Pramod.
Dr. Saligram, thank you very much for this excellent talk. I'm Pramod Garg this side. So you talked about the future preparedness from the industry point of view. I mean, you have created a wonderful facility, I believe. What do you expect the Indian academic institutions to come up with to shorten this development of new vaccines? And because, for example, in COVID also, for most of the vaccines, there was some component in terms of innovation from foreign universities and academic institutions. But in future, I think the innovation should come from Indian academic institutions. And what is it that you are looking for? What is it that you want?
So I must say that though it is seen that in Covishield and Covax, the technologies have come from foreign, but I must say that, without a lot of Indian institutes like ICER, THDSI, and ICMR Institute, it would not have been possible to translate this, because they all played a role. Probably the core technology may have come from others, but they played their core roles in, doing a neutralization assays, characterizations, and all that data has gone internationally. In fact, DBT supported Shantini played a key role, which is just startup out of Pune near NCL Venture Center. So all these BIRAC supported institutions, small organizations have played a role. I don't have time to, I wish I would have been face-to-face over there, would have explained you all, but it is impossible. There are many actors into it. So we are seeing one or two examples, but really there's a lot of things which has happened, and the huge thoroughness of all these data generated by Indian Institute have gone through a lens of USFDA, EMA, MHRA, all international agencies have checked this actually. And then the very fact that we are approved by all these international agencies, supplied almost 200 million doses to all these countries. So put together clearly shows that all the data generated is really an excellent data. So these contributions are not small, first of all. So I must thank all of them. The second thing is that in order to really come out with this more innovations and all these things for an Indian institute, I think we are already in talk with you, and we are trying to really come out with a solution. Unfortunately, I couldn't come. This was a time when I wanted to really come out. But basically it's all about putting a translational more with a private idea, with the ability of flexibility, and also we need to really do some changes in our thought process of how bring the innovation quickly into proof of concept human trial. That is where, like mRNA could happen very quickly in a Western world, whereas if India would have had that idea, how quickly we would have been able to translate. Whether we would have been, as a regulatory system, would have been very conservative about it or not is a questionable. Though we have done that, DBT played a key role, and now we have a self-replicating world's first mRNA available for people. So that this COVID-19 pandemic has channelized these energies. So now bringing this ecosystem actively rolling on with some more private part into it will probably help a lot with a more kind of idea of taking the product and bring it to a proof of concept human trial, and then allowing it to take the industry to take it forward would be a real good idea. So certainly that's the next phase, which in fact we are starting with you and DBT and trying to see how we can do that. No doubt about it. Yeah.
Thank you. Thank you. Should we move on because we are a little late? But the key message with Dr. Pramod Garg and Umesh are giving, that there needs to be now an ecosystem in India has transformed during last 3 years, and a more closer and a better collaboration between Indian academia and industry is required, so that some of the innovations right from scratch are part of the whole Indian ecosystem. Thank you very much, Umesh. Please stay on, and we'll move on to the next presentation. Dr. Guru Prasad, who is a professor at TSTI, he is going to talk to us, bridging the gap through capacity building and collaboration. Guru has spent a significant amount of his time in establishing flavivirus research program at TSTI, and encompassing host pathogen interactions, antiviral discovery, and immune responses to infection, with particular emphasis on dengue. In dengue, he has set up standardized assays which are being used for various purposes, both for regulatory purposes as well as scientific purposes. Guru.
Thank you, sir. Fine. I'm not sure if I should start by thanking the organizers, because I am one of them and I'm batting on a home pitch. In any case, I would thank THSTI organizing committee for having me as one of the speakers. So THSTI, actually capacity building is a genetic component of THSTI, I would say. For most of us who joined THSTI in 2010, capacity building at that time meant really, buying my first centrifuge, buying my first biosafety cabinet, having my first student, and how do I really start my program? So that was the capacity building in Gurgaon, in small buildings when we started. Most of my colleagues would agree with me. Then it was 2015 when we moved here. So that was capacity building again, in terms of occupying our labs, making sure generator doesn't trip, making sure we have, our first batch of animals that survive and we can perform our experiments. So having gone through these, first 5-6 years of capacity building to survive, that was the time when we had Dr. Kang and then Dr. Bhan, who anyway was a visionary and brain behind THSTI, who then started pushing us, to think beyond your own capacity, beyond your own lab, beyond your own science. So how do you really respond to what the country needs and what the world needs in terms of capacity for RNA virus research? So in 2018, we had our first RNA virus meeting organized by THSTI, funded by EMBO. So this was the first exposure that at least I as a virologist had with the global community, to understand really what is the trajectory for RNA virus research in the world, and what are the opportunities for translation. How do we really focus both on fundamental research and translational aspects? So after this meeting, multiple rounds of discussion with Dr. Bhan about what are the areas actually that India should invest in, resulted in this list of priorities that he thought should be the core essence of our infrastructure building and capacity building in India, and how THST actually should heavily invest on these aspects. I'm glad to say, and you have seen that from today, Dr. Garg's presentation, that we have been active in pretty much each one of these, except manufacturing ability, for which of course we have to depend on our industrial partners. We are very active in immunogen design and development. We have a very strong group now which has expertise in immunogen design and development. We are very active in predictors of protective immune responses, both T cell responses and humoral immunity. We have established now a platform with validated assays and harmonized protocols, which Professor Bernard really highlighted in his talk, that how essential it is to have validated assays and harmonized protocols. We are now investing in understanding disease burden data with cohort studies. We have extensive expertise and we have initiated programs on delivery platforms for vaccines. And of course, our core strength has always been clinical trial partnerships. Our expertise in clinical trial is what makes THSTI unique amongst DBT institutions. So where do I play a role in this? And that's when I really, after our discussions, I thought perhaps my strength should be focused on understanding predictors of protective immune response in RNA virus infections, establish capacity for vaccine development in the country, and also focus on generating disease burden data, which so far as was actually the domain of ICMR. So we are not competing with ICMR. We wanted to complement ICMR by generating data on pathogens of interest to us. So a year later, the sustained interest of DBT in capacity building, again, there was a meeting on which are the platforms that we need to invest on. And there again, the conclusion from that meeting was that we should invest on new generation viral vectors, which should be probably CMV, VSV, and measles virus-based platforms. After hearing Professor Ramarao's talk, I think MVA can be added to this list. I think that's probably another exciting viral vector platform. And at that time, there was a consensus that actually the country should invest heavily on mRNA-based vaccine platforms. Little did we know at that time that 6 months later, we would be really fighting a pandemic, and mRNA vaccines would serve as a boon to save the humanity. So our journey in capacity building at THSTI started with interactions with the industry. At that time, we were talking to Serum Institute, we were talking to Indian Immunologicals, we were talking to Panacea to understand what is the capacity that they would need in an academic setup. So the consensus was we need a place where we can go and perform assays which are validated, where there's accreditation for these assays, and then the regulators would really take this data as reliable and help in vaccine licensure. So we started this work, and it took us about 1.5 years. In 2018, we initiated setting up this laboratory. And by 2020 March, we were ready with our enable accreditation application. And that's when COVID-19 hit us. So this became the first GLP lab in a DBT setup. And we got enable accreditation, but it looked like actually we were preparing for pathogen X even at that time. So this is an example for how capacity building actually helps any pathogen. It can be pathogen agnostic. So the capacity that we built was never for COVID-19, but it became a COVID-19 facility at that time when the need was really to ramp up our RT-PCR testing, support vaccine development, support diagnostic kit development. So over the last 2 years, this lab has supported extensive diagnostic testing, more than 2 lakh samples. We also supported a mobile diagnostic lab. We partnered with ICMR, with FIND in supporting a number of diagnostic companies in validating their products. We partnered with FIND in training a number of people in performing RT-PCR. So almost every RT-PCR laboratory in Haryana actually came to us. Their staff got trained, and they were able to actually meet the needs of diagnostic testing at that time. Besides that, we also were busy with establishing in-house methods for quantitative ELISAs, neutralization assays for various SARS-CoV-2 variants and also other RNA viruses. So when we were doing all this, again, the question is, we are building capacity for India, and we were relatively doing well compared to other countries in terms of how we were fighting the COVID-19 pandemic. But if you look at the numbers as a global citizen, have we really conquered the pandemic even today? Because I think only 25 to 30% of the low and middle income countries have had access to COVID-19 vaccines. I mean, the problems are multiple. It could be manufacturing, it could be access, it could be vaccine hesitancy. But is there anything that we could do to really address these issues? Because this is the graph which clearly shows the disparity in vaccine equity. So how do we really play a role when we are okay, and how can we really make others also reach to this stage? So COVAX initiative is an initiative which was taken up by partnership with CEPI, Gavi, UNICEF and WHO. So the idea was to really make everyone safe because the pandemic doesn't know boundaries. And although we might be doing well as a country, we might always be at risk because there are other places where the pathogen could be evolving and could enter our borders anytime. So then there was a call for establishing a CEPI centralized network laboratories, which would provide validated assays and harmonized protocols for COVID-19 vaccine development for the world. So we responded to this call. We were selected as one of the 7 labs at that time. Now the network is growing and we are 11 laboratories. So we were the first lab in this network to complete all the assay validations for virus neutralization, pseudovirus assays, and quantitative ELISAs. So through these validated assays, we were able to support vaccine development for multiple manufacturers. Last year alone, we have processed over 25,000 samples. So this is our current capacity, and this can easily be doubled as and when the need arises and if there is demand for increasing the capacity. So once we had this capacity, then we could build on our collaborations. So WHO and NIBSC approached us to be part of their panel validation and panel preparation processes. So we contributed to the first and second international standard for SARS-CoV-2 IgG, the reference panel for various variants of concerns of SARS-CoV-2. We also were part of preparing the first WHO standard for SARS-CoV-2 antigen. We have supported the WHO Unity trial with AIMS in Balabgad, and we are also now supporting the solidarity trial of WHO, where we are partnering with Philippines to process these samples. So through the capacity building for pathogen X to capacity for COVID-19, we now have, we are very proud to say that the first subunit vaccine for India came out of THSTI. THSTI supported from preclinical till phase 3 for biological E, and the vaccine got emergency licensure last year, and now is one of the I think it's the only subunit vaccine in India, and is also being exported to many countries. And we have supported vaccine licensure for South Korea, for Netherlands, Taiwan, Vietnam. We are continuing to work with GPO Thailand and also Aurum Institute for South Africa. So this also side by side helped us to actually partner with ICMR, as I said earlier, to support many vaccine, many diagnostic kit development and also other accessories for diagnostics of COVID-19. So we now have a platform which can be used for many pathogens. This is one classical example where we have now validated assays for dengue vaccine development. Dr. Umesh Saligram was just mentioning about how they are preparing for dengue. And I hope that we can be a valuable partner and add a value addition to their dengue programs by providing these, partnering with them for any of these assays that they need. And as I said, now the capacity can be used for many pathogens. So now we are expanding, and the capacity is now supporting vaccine development for RSV, for dengue, for West Nile virus, and also we are ready for chikungunya virus vaccine development. We have an Indo-US vaccine action program through which we are now trying various adjuvant combinations, and we have interesting leads to show that the RSV vaccine that we have generated and the adjuvant combination that we have can be better than many of the other vaccines that are under development. And we are now venturing into understanding the population-based cross-reactive immunity to flaviviruses. We are seeing that majority of our adult population are already exposed to dengue, and they have antibodies to all 4 serotypes, but they do not have cross-reactive antibodies to Japanese encephalitis virus or West Nile virus, which again is not the same as what Western countries have reported. So there is an opportunity here for us to utilize this capacity and to build larger programs to understand, disease burden and to understand population level immunity to various pathogens. So in a nutshell, this capacity at THSTI has enabled both vaccine development and academic research. We have now one of the largest virus repositories for dengue viruses. We have a repository for SARS-CoV-2 isolates. We have supported serosurveillance studies at Pune, Mumbai, and also for INCLEN, where Dr. Arora is here, for their INCLEN serosurveillance studies supported by BIRAC, and also for AIMS at Balabgad. We have ourselves taken up studies to measure vaccine efficacy against Delta variant and Omicron, as Dr. Garg pointed out in his presentation. And we are extensively supporting people in THSTI, outside THSTI, in fundamental research. These are some of the outcomes of some of the papers that we were part of, where we could support both academic research and also product development. The latest being the DNA vaccine, which Dr. Arun is here from NCBS, for dengue, where we also provided all the support that they needed to show that this vaccine is actually efficacious in preclinical model. Now this is moving ahead with a mRNA platform, and we are partnering with NCBS to show that the mRNA platform also would be reliable. So essentially, we have now built this capacity to partner with many industries, with many academic collaborators, and we look forward for more partnerships with industry partners here, with ICMR, and also NTAGI probably should consider this as a national laboratory and encourage vaccine developers to come to THSTI and utilize these facilities and partner with THSTI in vaccine development program. So in a nutshell, we have established a GLP laboratory for supporting vaccine development with validated assays. We are also supporting academic research and product development. We are extensively involved in phase 1 through phase 3 trials for SARS-CoV-2 vaccines as part of CEPI network. We are supporting serosurveillance studies, and we are now expanding this capacity for assay development for other viruses such as flaviviruses and RSV, and pathogen X could any day be part of this capacity. So how do we see now what is the future for this capacity? As we all know that capacity building is a continuous process and needs sustainable investment. As Professor Vijayaraghavan mentioned, that it's easy to envision a response to a pandemic, but can we sustain this as a predictive response? Where do you really bring the funds to say, I'm going to maintain this facility irrespective of uncertainty in when we will have to respond? And I believe that capacity building has to be multidisciplinary, and THSTI actually has really kept this in mind and has focused on a multidisciplinary capacity building. And I'm happy to be part of that multidisciplinary capacity building. And it has to be a concerted global effort, because thinking only about our country probably is not going to be good when we think about a pandemic response. So I believe THSTI can take a lead to play a bigger role in the world. And we are aligning with the vision of CEPI. We just need to recalibrate whether the challenges of CEPI is what we really should be considered as a challenge, or should we really look at tailor-made challenges that is specific to India. And as I said, we are very keen to partner with industry and to understand their needs and to really build our capacity as per the requirements of the industry. And I hope the capacity we have can also become a reference lab for emerging pathogens. And this can also be a capacity that I want to actually also take forward as an antiviral platform. So this capacity building is not really a one-man's job. There was always a team behind this, and they have worked tirelessly for this capacity building. And the executive director of THSTI, both past director Dr. Kang and present director Dr. Garg, have been exemplary in their leadership and have promoted capacity building day in and out. So their support has been instrumental in actually maintaining this capacity and taking things forward. And thanks to all these collaborators who have helped us in establishment of assays and validation of assays. And of course, DBT has always been very supportive with generous funding through various programs. And thanks to CEPI and all of you for your time. I'm happy to take any questions. Thank you.
Thank you very much, Guru. THTI has shown how you have transformed the vaccine R&D scenario in the country during last 3 years or so by building this capacity, as well as then providing services and collaboration across the country. So, and that is what I think is the key, that both capacity building and collaboration, both are equally important. And the word sustainability you have said, collaboration will give us both regional as well as global role as well. Questions, please. Yes, madam.
Hi, Guru.
Hi, Rama.
Thanks for the nice talk, and congratulations on the truly impressive work.
Thank you.
I have two questions. First one, both are just for my knowledge. The first one is, so you did about 200, I think 250,000 samples you mentioned. You think that was on, so first part is, do you think that was on par or with the rest of the world, or, was it enough, so to speak, for the, or do you think it could have been more? It's, that is one thing. And the other thing you said was that you could double it easily. Now, what double it meaning, do you now have the infrastructure so that it's PCR, so the infrastructure is there, or doubling means you quickly train people and hire more people? Is it more personnel oriented? Or is it more infrastructure oriented?
So coming to the first question, we were the first lab to initiate RT-PCR testing in this region. And we were probably the only DBT institute which was functional during the initial stages of the pandemic in the NCR. So our partnership with ESI Hospital was what actually initiated this RT-PCR, because initially it was supposed to be a hospital-based diagnostic lab. Then later on, we became an independent testing lab. So on any day during the peak demand, we were processing about 400 samples every day with just 7 to 8 people. So you should think that at that time, you should realize that during that time, nobody was even coming to work.
No, that was the whole point of asking the question,
Yeah.
that it was impressive. I think you were very humble and just saying that we did these many samples. So the question was to just impress on everyone that it must have been difficult, and you pulled off something truly exceptional. Yeah.
Yeah, so we were able to do that because of the people who actually were willing to come and do this work. The later part of testing, even Dr. Bhaktosh has chipped in the last 6 to 8 months. Even he was part of the testing job. So I think after ILS, THST has probably processed maximum number of samples. And coming to the capacity, see, the capacity we have now is what we can do. So about 25,000 samples can easily be processed in a year. We have trained people. So suppose tomorrow if Serum Institute says we have a large trial and we need, let's say, 4,000 samples to be tested in 2 months, we can ramp up the capacity. So we have the infrastructure. What we need would be really getting more trained people. So if we can get heads up on when the timelines and the demand, we can easily ramp up.
Thank you.
Welcome.
Yes. Dr. Guru
Priya.
Prasad, right here in the front, right here, right in the front.
Oh, yes, ma'am.
Congratulations. I think it's
Thank
a fantastic effort. Your multidisciplinary GLP lab is something I think should be the country's pride. So I just want to ask you, are you going to now align yourself with any of the WHO's blueprint priority pathogens, since you have such a diverse capacity built over these last few years? Are you focusing? I mean, there are many people after dengue, many people after TB. Are you thinking of any of the priority pathogens?
Well, at my level, I have discussed multiple times with WHO representatives during meetings. And also we have had discussions that we should really now partner with the WHO and maybe take this lab to a level where WHO can recognize this as a reference lab. And also partnering with ICMR is key, I believe. So I again would emphasize that we need to actually work together with ICMR and make this as a national laboratory. I don't think there has to be any boundaries saying this is a DBT lab or this is ICMR lab. So we are doing this for the nation, and we should really make this as a reference lab, irrespective of DBT or ICMR. So I would really need help in that direction. I had briefly discussed this with Dr. Swaminathan when she had visited here. And we have to take this forward, and I don't have direct contacts with WHO. Perhaps at DBT level and at directors level, we can have these discussions.
Great job.
Thank you. Thank you. Thank you, Guru. It was excellent.
Yes, sir.
No, wonderful. This was what a vision me and Dr. Ban thought of 2007 to set up THSTI. So really you are fulfilling this type of translational research milieu to other things what we are doing about it. I'm really happy that you are facilitating and also preparing for the next pandemic which coming, and TH is the right thing, and all in the industries to connect to you to facilitate all of it. Excellent. Congrats. and entire team of THSA. Thank you, Guru.
Thank you, sir.
Thank you. Yes, sir. Let Ramarao speak. Yeah.
Yeah, it's really amazing effort. Congratulations. I have a question about your neutralization assays. Are those pseudovirus-based assays or live virus neutralization?
We are doing both. We have live virus assays where we do focus reduction neutralization titers. So these are high throughput assays. We also have plaque reduction neutralization assays. And we have VSV platform pseudovirus assays. But we didn't make the pseudovirus ourselves. So this is as part of CEPI network, there is a centralized lab which makes these reagents. So that way there is no discrepancy between lab to lab, lot to lot variations. So all the labs use the same reagent.
Okay.
Yes, sir.
So my question is related. It really take efforts to have a lab which is, ISO 17025 GLP lab. So congratulations for that.
Thank you.
My question is the assays which you have apart from the neutralization assays for the live virus or the pseudovirus, is it specific for the COVID or do you have any other assays which are validated for other pathogens? What is like, the number of assays you have, one, and are they available for the other pathogens as well or the disease as well?
So as I said, when we started this, it was really a pathogen agnostic facility. I was working on flaviviruses, so we thought we will have dengue as our priority and set this up because at that time, phase 3 trials were planned for dengue, phase 2, phase 3 trials in the country. So the need was to have a lab which can actually do validated assays for dengue virus vaccine development. So we have ELISAs for dengue virus assays, the molecular assays for dengue virus. We have molecular assays for chikungunya, RSV, Zika virus. We have neutralization assays for all the pathogens that I showed. And of course, quantitative assays for SARS-CoV-2 and IgM, IgG, NS1 assays for dengue also. And I don't think it takes a lot of time now for us to really establish an assay and validate an assay if there is a need by the industry. So the only thing we need is really partnership and discussion from day one, where you decide that maybe 3 months down the lane, these are the assays you need, and we can ramp up and get those ready.
Thank you.
Yeah. Guru, one question.
Sure.
As I understand, during the pandemic, there were some delays in getting reagents. As a result, some delays occurred for the task you were trying to do. So is there an effort to encourage industry or somebody to manufacture these reagents locally? Because I think this became an issue with vaccine manufacturers and some of the others also. So what is your view on that? That it should not become a rate limiting factor despite capacity and collaborations.
So we had multiple discussions during that period with DBT. Actually, our biggest constraint, even for RT-PCR, was getting those enzymes, primers, and probes, which was absolutely, not available in India.
I'm aware of that.
Yes. So we supported, product validations, but the core raw materials at that time, we really did not have that expertise. But I believe over the last 1.5 years, the C camp at the NCBS campus, at least Tasleem says that they have supported more than a dozen companies to manufacture these reagents in-house now. So now India, that is the reason why the RT-PCR testing costs have come down to about 250 rupees per test now, because the reagents are now available in India, made by Indian companies. So that capacity has been built.
Okay. Thank you very much. Thank you.
Thank you.
Our next presenter is Dr. Ankur Mutreja, who is leading the CEPI's LMIC engagement portfolio, including India engagement, and he's responsible for designing and implementing the strategic roadmap to engage with Global South, and also making effort to integrate R&D, manufacturing, and access plans of CEPI. Ankur.
Okay, thanks very much. And thank you, Chair, for the introduction, and thank you to the organizers for having CEPI here and for asking me to present what the CEPI 100-day challenge means for the real world. I think a lot has been covered since morning through the introduction talks and the opening remarks as well about the CEPI approach and what exactly the 100-day challenge is and what are the challenges to achieve that 100-day challenge. So I'm from CEPI, so I think it would be worth getting an introduction to global CEPI. I know you're all aware of what IND-CEPI is and what IND-CEPI's mission is, which is very closely aligned to the global CEPI mission. So we are a global partnership between various different governments, civil society organizations, philanthropies, industries, regulators, academia. So we work with all those entities. Our vision really is to have a world ready where epidemics are no longer a threat to humanity. Our mission is to really accelerate everything that's needed to achieve the vision, to achieve the vision. And that is primarily around designing vaccines, producing vaccines, and enabling equitable access to these vaccines so that affected populations during those outbreaks do not struggle. And one of those examples is COVAX, as has been mentioned in some of the previous talks. We were one of the co-leads of the COVAX program. So CEPI has a pathogen priority portfolio. We work on a set number of pathogens just to ensure that our programs are very streamlined and our approach is very focused. So these are the pathogens, as you're seeing on your screen, from MERS to disease X, and COVID-19 was added as per the requirement when the pandemic was declared as a health emergency by the WHO. So far we have funded several vaccine candidates for all of those pathogens, but last year we launched our CEPI next 5-year strategy, which is a CEPI 2.0 strategy, as Guru mentioned. In CEPI is aligning very closely with the CEPI 2.0 strategy, and this is what the strategy really is. So we have 3 pillars under the strategy, which is prepare, transform, and connect. Under the prepare pillar, we really want to work to minimize, or let's say get rid of, the pandemic COVID-19 threat. And by doing that, we really want to get to the broadly protective coronavirus vaccines so that upcoming coronavirus threats are also neutralized. We also want to develop vaccines for known threats. Some of the known threats, as I mentioned, are our priority pathogens. And then moving on to the transform pillar, we want to create a library of vaccines so that those platforms are available, those prototype libraries are available to really help us create vaccines for that crucial 100-day challenge that we all have been discussing today. So that's 100-day challenge is part of our transform pillar. And then connect pillar is really about connecting all the pieces of the ecosystem, as it has been mentioned again since morning, that there are quite a few efforts everywhere from academia to industry, from research institutions to public health, but all of those efforts mostly tend to work in a very siloed manner. So the idea of connect pillar is that we would really connect the dots, play a role of a facilitator, bring all the stakeholders onto a common platform, and really arrange those discussions. And then finally, supporting the efforts of Epidemics to really take the full ownership of their own national health security. So this is very, very important, because we don't really want to just invest or help out the world so that the high-income countries benefit. And my role in particular is to look after the Epidemic engagement portfolio of CEPI. So coming on to the 100-day challenge or 100-day aspiration, we really call it an aspiration because it is really a moonshot effort. It is not something that we should think of achieving, overnight or just, by installing systems, because again, as it has been mentioned, every pathogen is different. Disease X could truly be any disease. mRNA vaccine platform, which has worked very, very well for coronavirus vaccines, may not be the appropriate platform for the next disease X. So the idea of 100-day mission or challenge or aspiration or moonshot is that from the day of alert trigger, Now, the word alert trigger is really, really important, because if you do get the alert trigger wrong, then you actually set yourself on the wrong course. So you have to get that trigger right. And for that, institutions like THSTI, ICMR, DBT, all those institutions have to play a critical role in really establishing the disease burden and sending the signal through the right sources to the right agencies, so that we can pick up that alert trigger and really set the course running. And then what we really want to achieve in those 100 days is first vaccine available for use for people who need it the most. So this is not really a vaccine ready for mass distribution. This is really first vaccine dose available for human injection or delivery. Now, this is an example of how it took only 326 days for the development of emergency use authorized vaccines for COVID-19. As you would see, through preclinical all the way to registration, previously it used to take anywhere between 5 to 10 years. But in COVID, it was exemplary that the world actually came together, and all those silos that used to work in a very disconnected manner came together and started working in parallel simultaneously, and really delivered a coronavirus vaccine approved by a stringent regulatory agency in 326 days. There were vaccines. The Russian vaccine was even approved before then, but this is an example of approval by a stringent regulatory authority, which is European authority to start with. Now, coming to some of the examples, how the sort of median number of days that it took to develop the first coronavirus vaccine. Here are all the vaccines that got approval or were on the path of approval in the first 2 years of coronavirus vaccine development. And you would see that some of the vaccines like BioNTech, Pfizer, Moderna, AstraZeneca, Johnson & Johnson, and others were really able to achieve that within a time frame of almost just 1 year time. And all of this took a lot of coordination, simultaneous work, parallel activities, and you would notice that, you know, from sequence availability to first in human trials, and then on to the phase 3 trials, some of the efforts actually took just about 300 days. But we've been discussing 100 days because 326 days itself is also not enough. You would notice in this graph that when WHO received the genetic sequence of the novel coronavirus, or SARS-CoV-2 as we call it now, and the WHO declared it a public health emergency, if we had a vaccine available within 100 days of that declaration, which if you can call as a trigger, then only 0.2 million lives would have been lost. But by the end of 326th day, globally, almost 1.6 million lives were lost. So that's the reason why we are all really coming together here and working towards a common goal of achieving that moonshot of delivering vaccine in 100 days. Now, it is a moonshot because it will require a paradigm shift. It will require front loading of a lot of activities, which would primarily be around preparedness and breaking the firewall between development and intervention. So if you see, the 100-day challenge here is really sort of divided in sort of 3 pillars of its own. Or first, you have to have readiness, then you can get into the response, and then you can get into the rollout and the review of the process. The 100-day itself, how it would look like would be the immediate reaction phase, in which the candidate development using the prototype vaccines, which would have been already developed beforehand, would become very pathogen specific. All the immunological assays that Guru presented today would come into practice, and the CEPI Central Laboratory Network that CEPI has funded so far, which is further expanding from 11 to approximately 20 labs in coming months, is going to also help prepare for that immunological assay development in a very harmonized manner at an international scale. And also to further help, we are also putting together a manufacturing facility network for large-scale clinical batch preparation. So these hot facilities that we are bringing on board, very similar to the clinical CEPI clinical laboratory network, would really become activated as soon as a worthwhile trigger is approved by CEPI. Then you enter into the 60-day phase, which is the evidence generation phase, in that you really use the data that you've generated from the prototype vaccines and the libraries and the candidate generation, and you really use that safety and dosing information to get straight into the immunobridging phase 2, phase 3 trials as well. And then finally, a rolling review of all the work that would be done in the 30 and 60-day period would be done in the filing stage. Now, it's worth remembering that all throughout this period, you have to continuously monitor or assess the outbreak, because it may be that the trigger threat has changed. It may be that you need to ramp off, which is what I'm showing here in this slide. 100-day is really our draft starting position. But as you continuously evaluate your outbreak threat, or your change of scenario, you really start ramping off the 100-day mission, and through that you can then get to the 150 to 180-day window, or 200 to 230-day window. The key thing here is, despite all of that acceleration that we are trying to bring in, we would not minimize our efforts around safety and risk benefit. And also we would like to ensure that everything that is developed, even in those 100-day periods, should be equitably accessible. And for that, primarily, there would be investment needed in 10 initiatives to enable the overall goal. And the first one would be under the leveraging of the R&D toolbox. So really, as I said, front loading of all the activities. So this is really all the work which would start before the day 0 of the 100-day period. And then rapidly characterize the outbreak and trigger alert, and then disseminate information. As Dr. Raghavan this morning mentioned, the flow of information is absolutely key for all the facilities to really start working in a connected manner. And then scaling of the manufacturing by establishing activating pre-established infrastructure through things like CEPI manufacturing facility network would also come into play. And then gathering evidence according to predefined response pathway, scale, development, and rollout. So this is all information that would eventually become very, very useful for the speeding up of the review process as well. So CEPI is trying to work on every single piece of the value chain to really push the boundary, to really see how we can achieve this 100-day moonshot to the best level possible. So all the way from problem or gap identification, where we are running technical workshops at country, region, and continental levels. We're also doing manufacturing landscape surveys to really evaluate where every region globally sits today in terms of their manufacturing capability and capacity. Then we are also generating a lot of data for convincing case building, because in a lot of countries and a lot of regions, they have problems, they have diseases, but they're not able to really highlight those at a level so that they can be declared a valuable trigger to design something at a global level, which requires a lot of global advocacy. So CEPI is really part of that. And as I said, as part of our Connect pillar, we are trying to achieve that. And then capacity and infrastructure development. One of the examples that has been shown since morning is centralized laboratory network. Similar work CEPI is doing on animal network, manufacturing facility network, regulatory network, all of those pieces connected. And then all the way into system centering, from regulatory to COVAX marketplace as part of the ACT-A pillar, and then offering technical guidance and technical support to all the partners who we work with to really ensure the equitable access bit. So in reality, this is what CEPI's model is. We work with governments, regulators, philanthropies, civil societies, pharmaceutical industry, academia, and other stakeholders especially from the community side as well, to ensure that everything that is generally siloed comes together to really believe in the vision and mission. And I'm really glad to see this organization of the meeting is accelerating what the CEPI mission launched as a 100-day mission. And with that, I'll stop and take questions. Thank you.
Thank you very much, Dr. Mutreja, for making us aware the whole philosophy behind this 100-day challenge. Any questions? Yes.
So since you mentioned about AI as well during your talk, so what is the progress on AI? Can AI be used as a tool for rapid development of vaccines or what therapeutics or preventive measures?
No, I think that's a great question. One of the reasons why we are thinking of AI is because often the scale of the disease, when you're trying to start a 100-day mission, and when you really want to have the alert trigger as early as possible, instead of having real incidence data or mortality data or hospitalization data, you want to have modeled disease outbreaks. And there are AI systems these days that CEPI is funding, and also CEPI has recently funded some capacity building in that space from ICMR, getting training for chikungunya disease modeling, for example. So there is promise there that you can really use that modeling data to get onto that critical alert trigger that you need to decide whether you want to start the 100-day program or not.
Yes, ma'am.
Yeah, excuse me. Here.
Thank you, Dr. Mutreja. This is a very fascinating talk. I would like to have your opinion regarding, like, if the disease is known, then it is possible we do have some kind of information about the disease, and it is possible that we can have and come to an achievement after within 100 days. But if the outbreak is caused by any unknown pathogen, whose we are not aware of any of the information, we do not have any of the information about the pathogen, how feasible it is to come up with this 100 days challenge of CEPI, what you have talked about. So your opinion, please.
No, absolutely, Dr. Datta. I think that's a great question. I think that's why you work on prototype libraries of vaccines instead of working on specific known pathogens. And you are really working with the hope that any new pathogen that comes either fits in one of those library categories or it is closely associated with one of those. So to give you an example, SARS-CoV-2 vaccine design, AstraZeneca vaccine design was very much based on the MERS vaccine design. So a lot of effort was put into that, and therefore it was very, very quickly translated into SARS-CoV-2. So you really work on a similarity model here instead of finding something absolutely identical. Having said that, there are potential viral families that are likely to cause pandemic in coming decades. And so the idea is to create vaccine libraries for all those viral families so that you are covered in the best possible manner.
Yeah.
Yeah, great talk. This is Sujata here from ICGEB. So with respect to your AI, the previous question, I'm just curious, are you also collecting these data points yourself, or are you using the existing data points? Because definitely it's going to be a disparity.
Yeah, no, we are not collecting the data points ourselves. We do have some epidemiological networks for our priority pathogen diseases, but for disease X, we are really going to rely on existing other networks.
So, hi, Ankur, this is Dr. Atulya. My question is basically, are you focusing on pathogen surveillance and post-immune surveillance initiatives by CEPI to identify that pathogen X well in advance, because these pathogens are evolving in environment, and if we can identify well before emergence of any pandemic?
Yeah, no, again, I think similar to the previous answer, I think, for priority pathogens of CEPI, we are investing into epidemiology of those things as well. But for other disease X or other viral families, we are dependent on, early warning systems that other people are putting in place, such as, Johns Hopkins in the US, or there is a Berlin hub for surveillance. There's a hub that is coming up from Saudi Arabia for the Middle Eastern region, and so on. So we really are thinking that those would be able to provide us that crucial alert trigger if required. Thank you.
Yeah, Dr. Ankur. Hi, this side.
Yeah.
This side. Right. So this is Mihir Chhaya from Bharat Biotech. This is relating to a statement you made at this, and I think in the third or fourth slide, where you said that the mRNA platform technologies worked very well for COVID, but they may not be appropriate for disease X. So that was a very interesting statement coming from CEPI. So could you please enumerate more on that?
Yeah, no, I said that because I think, again, it has been mentioned since morning, whether we've been lucky or I think whether spike protein was something that came up to be very, immunogenic and protective as well. And that may not be the case for another pathogen where, a simple sequence of a particular protein or a particular gene would guide you into a proper vaccine. And that's where multivalent vaccines might be needed to start with. Or they might be viruses that have multiple genomes inside them. SARS-CoV-2 had only one. So, there could be other platforms which might become more useful as compared to mRNA in those cases.
Yeah. Thank you very much. We'll come back. Okay.
It's not a question, it's just an information for the young people in the audience. CEPI was set up by 5 members, founding members, and Department of Biotechnology was one of the founding members. And I'm saying this because I feel the historical perspective, institutional perspective is lost. And Department of Biotechnology represented Government of India, along with the Government of Norway, Gates Foundation, Wellcome Trust, and World Economic Forum. And that was a very futuristic vision for Dr. Vijayaraghavan was the secretary at that time. I think should be given enough credit for that, and it should be mentioned in all forums.
Thank you. Again, another example of visionary collaboration, and the repeated underpinning coming is in this is collaboration and going beyond the boundaries and the traditional partnership exercises. May I now invite Professor Priya Abraham from CMC Vellore. In fact, she was at the forefront of country's fight against COVID as director of ICMR, National Institute of Virology, Pune. And she has been on various WHO committees, working groups, and has been leading infectious disease related agenda across the globe. Professor Priya Abraham.
Thank you, Dr. Arora. And I must say, I'm really happy to be here at this really good meeting. And the topic that I've chosen to take is vaccines for low incidence, high consequence viral agents, which we know is an unmet need, not only in other parts of the world, but in our own country. So you are all very familiar with the WHO blueprint list of priority diseases. I'm not going to go through all of them in the interest of time. I'm going to focus a little on the examples of these diseases that we have seen in our own country, maybe deviate to yet another a forgotten agent, and probably wind off with where we stand today after listening to the wonderful lectures thus far. So start with Nipah virus, and this is particularly for the younger members in the audience. This is an exotic virus. It's a zoonotic agent
today believed to have its reservoir most likely in fruit bats. First known outbreak was in Malaysia in 1999 and in Singapore with a mortality of 40%. It was then seen in pig farmers, and it was believed to come from pigs who were probably the amplifying hosts, which picked up the virus probably from bats, and then transmission happened into humans. The next outbreak came closer to home. It was in Bangladesh in 1999, and right up to last year, they have been having repeated outbreaks, small ones, larger ones. Around 322 individuals have gone down with Nipah infection with a mortality of 70%, which brings it up to an even higher level. And the possible source is believed to be from consuming date palm sap. It's a tradition, particularly in the colder months of the year. And we believe that this date palm sap may be contaminated with the saliva of fruit bats. And therefore, when people consume this date palm sap, which is not cooked, it's done neat, so to say, the chances of transmission to humans does occur. Now, India also has been home to Nipah virus infection. And if you see right from 2001, right up to last year, we have had 5 outbreaks. And in the far right of your table, take a look at the percentage mortality. Of course, in one particular case, it was just a single individual, but you can see the mortality case fatality rate is rather daunting. And it's interesting to see that the cases in India have shown a kind of clustering. It's seen in Eastern India, in West Bengal, and also in the neighboring country of Bangladesh. And all the subsequent outbreaks have been from the state of Kerala. Now, if you look at vaccines under development, you know, sometimes I think when there are many answers to a question, there really is not a clear answer. And there are many candidate vaccines that have come right up to phase 1 clinical trials of different genres, different platforms, live attenuated recombinant vesicular stomatitis virus, VSV vector vaccines, mRNA vaccines, subunit vaccines that take care of not only Nipah, but Hendra, which is a cousin of Nipah in the Henipah virus group. And again, another subunit vaccine. Then we have also examples of other recombinant VSV vectored vaccines, even in the adenovirus vectored vaccine, and again, recombinant vaccine genes put into an attenuated measles virus vector. These are all the different varieties of Nipah virus vaccines on the anvil, but none have gone past, or we've got the results of phase 1 clinical trials. At this juncture, I hope Dr. Shaligram is listening to me. I would think one of the quick fixes, if we were to have an outbreak, is to be able to actually have monoclonal antibodies. As you know, that has been historically used earlier. In one of our first outbreaks in Kerala, it was used. And we do not have a steady supply of monoclonals from overseas. In fact, in the last outbreak, we scrambled hard to get it. We didn't get it. So, Dr. Shaligram, the challenge is back to you. We have spoken about this earlier. I think we need to make our own monoclonals, humanized monoclonals. So we have a small stockpile of this, given the rate of fatality with a virus such as this. We go on to another fairly deadly pathogen, Crimean Congo hemorrhagic fever. It was detected way back in 1944 in the Crimean Peninsula. But today, all that's colored in orange is where this virus has been picked up from. So mainly in Southern and Eastern Europe, Africa, Middle East, Asia, including Western India, the states of Gujarat and Rajasthan. It is seen basically in regions where we have the distribution of hyaloma ticks, and it is infection is typically picked up when individuals handle infected livestock. These happily infest the goats, the cows, and the other livestock that farmers and people in a rural setting would rear. And in the healthcare situation, as we know of a case in Gujarat where a surgeon went down with succumbed to CCHF, the healthcare providers are also extremely at risk. It's a viral hemorrhagic fever with a case fatality of 5 to 30%. The first confirmation of CCHF happened in Gujarat about 10 years ago, little over 10 years ago. And one thing I would say, I would proudly say, ICMR NIV has been able to do is quickly develop diagnostics. So we have developed diagnostics for all the high consequence pathogens, be it Nipah, be it CCHF, be it even Ebola, yellow fever. I'm no more part of ICMR NIV, but even having left it to join back my alma mater at CMC Vellore, I look back with pride that we were very good at developing diagnostics and picking up the agent in good time. Again, with CCHF, we have about 4 candidates, a DNA-based vaccine using the electroporation technique of delivery, formalin, the standard formalin inactivated vaccine, replicating mRNA vaccines, and a multi-epitope subunit vaccine is based on a immunoinformatic approach that is a little more, very early in its development. But these are options that are, on the anvil, but nowhere near being available if we were to have an outbreak even in our own country. This is a virus nobody talks about. It's not on the WHO blueprint list, but many of you heard about it. It's Cyasunur forest disease. It's a flavivirus which had its origins in the forests of Shimoga. There was occasional spillover into neighboring areas initially. This virus breeds in ticks, and they actually can get into other rodents, monkeys, and eventually into humans. The first isolation of the virus was actually successful way back in 1957, again by ICMR NIV Pune, and it has a case fatality of about 3.4 to 3.5%. Now, how does this virus actually get into humans? Yes, we contribute by the extensive deforestation and all the other animal interactions that go with it. So when we encroach into forests, people's dwellings are on the fringe of the forest, cattle are grazing there, monkeys are happily, roaming in and out of human dwellings and back into the forest. And then people go in there to pick up fruit. For instance, in Goa, people would go to get the cashew fruit in its season, okay, between May to January or so. And when people go in there, they are picking up these ticks on their bodies, and the ticks, bite them. And if the ticks actually carry the virus, that's how infection can transmit. And of course, people would go there for fodder and firewood or what have you. The humble agricultural workers and farm dwellers would walk into the forest that's so close by, and that's how it gets into human dwellings and can infect humans. And what you see is what started in the forests of Shimoga today is you can see evidence of either virus itself or in humans or in monkeys or in rodents, or evidence of antibodies in monkeys or in humans. And today, what is colored in a yellow color depicts the areas where we have been able to pick up evidence of KFD activity. So it's really a hotbed on the western coast of India, going right up to Gujarat, bit of Madhya Pradesh, Maharashtra, Goa, Karnataka, Kerala, and even into Tamil Nadu. What are the efforts that were made for this, ill-known virus? Way back in the 90s, ICMR NIV had developed a formalin inactivated vaccine. This was handed over to the Shimoga-based government virus diagnostic lab. And then they handed it over to an Animal Health and Veterinary Biologicals Institute in Bengaluru, and they started making the vaccine. Initially, this vaccine seemed to provide some protection to forest and agricultural workers. But today we know, when we look at its pathetic potency tests, when we are seeing infections despite even 2 doses of vaccine, this vaccine has now been declared as stocked out due to serious regulatory and quality issues. So here is a window of opportunity. Maybe it's only a niche population, but in those who live on the fringe of the population and where we continue to allow zoonotic spillover, this is a vaccine that could very easily be developed in our country and is not really the concern of people in other parts of the world, at least not yet. Zika virus, much has been spoken about it. We all know that our first case occurred in 2018, and luckily, as per genetic sequencing, we do not have that critical mutation that is associated with microencephaly. One might say it causes a very mild asymptomatic infection, but for the woman who picks up that infection during a pregnancy, and we saw the image that Professor Raghavan had showed, the image of a child with microencephaly, that is devastating for the child and the mother and the parents. Sero surveys subsequently, this was the first case was in Gujarat. We then had reports from Rajasthan, Madhya Pradesh, Tamil Nadu. Last year, Kerala, Maharashtra, and Uttar Pradesh joined the string of states where we're actually picking up evidence of infection. And then this gave an impetus to ICMR via its VRDL network to do a wider sero survey. And today we have activity of Zika virus sero prevalence in 17 states in India. There are again efforts to make the Zika virus vaccine. And again, there's a DNA-based vaccine, purified inactivated version, a live attenuated Zika virus, which is a chimeric virus, which contains the dengue type 4 serotype as its backbone. And then we also have an mRNA vaccine, which is directed against structural proteins of the virus. So that there are different phases of development, but we still do not have a vaccine for use in countries where seroprevalence has been clearly shown. There are more examples of candidate Zika virus vaccines, including genetically engineered VSV vaccine, a measles virus-based vaccine, and also a novel vaccine that targets the mosquito salivary proteins. We are hoping that this would help to, prevent mount an immune response when an infected mosquito bites. Now, Dr. Shaligram very hearteningly mentioned the Serum Institute of India's efforts towards sending out doses, which I presume is for the Sudan Ebola virus. We all know the Sudan Ebola virus yields to about 40 to 60% mortality, maybe less than the Zika virus. But we also know that the currently available Zika virus vaccine and the therapeutics that are available are not effective against this Sudan Ebola variant. There are at least 24 investigational vaccine candidates if one were to look into literature, and they are also targeting not just the Ebola virus, but the Sudan Ebola virus, but the Zika Ebola variant, as well as the Marburg virus, which is a very good initiative because they are all equally deadly pathogens. So we hope that we'll be hearing something good about the efforts of Serum Institute of India. A Lassa fever vaccine. This is another viral hemorrhagic fever, which every year causes about 100,000 to 300,000 cases annually, particularly in the western part of the African continent. You can see the number of deaths that occur each year. There is one candidate vaccine using the recombinant VSV vector backbone model, and this has been also supported by IAVI and CEPI. I wanted to also say that with the Nipah virus vaccine initiatives, I must acknowledge CEPI's very gracious efforts in supporting at least 4 of that slew of vaccines against new candidate Nipah vaccines. Now, we heard a lot about monkeypox. By the way, young people, we don't call it monkeypox anymore, we call it Mpox. Okay, so for Mpox, we do know that this year round, we have seen, in contrast to the sporadic cases that we have seen in Africa and a few exported out of Africa, this year we have seen almost 80,000 cases the world over. Increased transmissibility surely is the case. Altered clinical presentation, and in many cases, it must have been subclinical for the way it spread. It has affected certain specific groups. We know this virus is evolving in contrast to our understanding of DNA viruses in general, but it's a big genome and it's doing a lot of different things. So it is evolving. And very recently, one of the WHO meetings I was attending alluded to the fact that there is evidence that some of these mutations have led to resistance to tecovirimat, which one of the key antiviral agents that we have in the antiviral armamentarium. Second and third generation vaccines are there in developing countries. They keep it all to themselves. They also have therapeutic options. There is non-availability of either vaccines or antivirals in our part of the world. Think of our healthcare workers. They might be seeing cases that are mildly symptomatic, presenting with an altered clinical presentation. They are not protected. We don't even have something to treat them with. And of greater risk would be if this monkeypox, though thankfully it seems to be going down a bit, but if this were to get into young children, pregnant women, and immunosuppressed, our options would be more limited. Definitely, we need to find indigenous solutions. And in response to this, ICMR did put out an expression of interest. There were 5 major companies that have come forward with making the monkeypox vaccine. And I would really like to see that becoming reality very soon. Why is the urgency? The good news is that in terms of diagnostics, surveillance, and training, we are in a good position. Even to handle the most deadly of viruses. We in India have a BSL-4 facility that can handle even the nastiest of pathogens. So we can work with diagnostics. We can train people to handle in a higher biosafety level. We can do surveillance. One of the good things that ICMR did was to establish the virus research diagnostic laboratory, which is over 130 labs across the country. And COVID taught them all to do molecular testing. We all spoke about some of the good spin-offs of COVID. So diagnostics, training, and surveillance is something we can handle. We can even make new novel diagnostics. But right now, for many of the WHO blueprint organisms, there's no therapeutics, and vaccine is not in the at least close on the horizon. These agents not only have epidemic potential, but many of them have possibly pandemic potential. It may be used for bioterrorism. That's not often spoken about, but we need to keep that in mind. And we also know that some viruses are slowly and stealthily evolving, which might hamper our sustained efforts in surveillance and diagnostic product development, and even in our therapeutics and vaccine domain. But I do not want to close off on this very somber note. We listened to some of the earlier speakers. We listened to Dr. Guru Prasad. We listened to Dr. Dr. Shaligram and Dr. Guru Prasad. They both have something good to offer. And I think this is the time of interinstitutional hand-holding, breaking down those, iron curtains between institutions. And I think COVID has taught us a lot that we can be self-dependent to a great extent. So I think we may not, when I was preparing this talk, I said, no way, 100 days, 365 is possible. But now I'm bringing it down, 326, maybe even less than 326. So I think we should think positive, and we need to harness all that we have in our country to make some of these vaccine products a reality. Thank you for your patient listening.
Thank you, Priya, for refocusing on the statement that contextual realities have to be taken into account, and that's important. And for some of these, we have to take the initiative rather than wait for somebody else. So we have about 20 minutes, and Pramod, how do we go about? Should we call everybody at the podium? I think all the speakers, if they can come over to the podium, and for next 20 minutes, we can have a question answer session. We have our online speakers, Professor Bernard and Dr. Umesh Saligram are there. Rest four of, and give us 1 or 2 mics so that we can take up questions from everybody.
I would request all the speakers to come on the stage for the panel discussion, please.
Take this one. ये वाला भी लो. Please. इधर. So we will invite questions.
Yeah.
So anybody would like to? Yes, sir.
So yeah. So
please address your question
Yeah,
to
so,
the particular speaker if possible.
Yeah, this
Otherwise-
question is to Dr. Priya, and so myself is Dr. Noor from University of Hyderabad. So this is regarding the monoclonal antibodies. So and therapeutics you talked about. So requirement of monoclonal antibodies, and we have seen that how monoclonal antibodies in COVID, they are not so potent. So rather than morning Serum Institute, they talked about equine anti-serums. And I think that's one of the quick and very good therapeutics. And why I'm saying, because we have been associated with developing therapeutic antibodies, FAB2, raised in equines against COVID-19 in collaboration with Vince Biotech. And it has already completed a phase 2 trial. And the success rate, if you compare, because I was one of the part of these trials. So why I'm saying this could be a really it's groundbreaking, and it could be quick because we have from the development to clinical trials, it was done
Thank
in
you.
India.
I think you have made the point, Priya.
So
would you like to answer?
Can it be used for other diseases?
Historically, we do know that high titer equine anti-serum is an option if we're really stuck But more and more people are moving away from non-human sources of hyperimmunoglobulin. But since it's good you asked me that question, because we worked with 5 companies, including Serum Institute of India and Vince Biotech. We handed them the inactivated virus and actually tested the titers, because we did PRNTs. So we checked out the titers of the antibodies that these equines did put up. They put up enormous, the Serum Institute of India, it went into lakhs. The titers were in lakhs. But it fell out of popularity because after the, the Placid trial, the human convalescent plasma trial, which did not yield very good results, this use of hyperimmune or ready-made antibodies somehow didn't take off. But it's there as an option in the future.
Ma'am, my question, see, because I have been associated with these trials, and we have seen itself like within 2 days, the people who are admitted, the chronic people, they were discharged. So I think it has a little potential, and a lot of companies like Serum and who have a bigger animals, they can take it forward to fight against the disease X.
Sure, it is an option if we are stuck with no antidote or not a vaccine.
One of
not
the
developed.
options, certainly, yeah. Yes, sir. Yeah, in the center. This way.
So my questions are for Dr. Rama Rao. First is regarding your prime boost strategy of vaccination. So which, a memory T cell dominate in this case? Is it against the primary antigen or the secondary? And how does the B cell in this case will get help? And my second question to you is regarding your there's vaccines against tuberculosis. So why do you think that this coronavirus vaccine could be quite successful in limited period, but BCG vaccine has failed?
So in the prime boost, the prime is very important. The prime basically set the stage for the direction for the immune response. So it really dictates a lot of things. And then the boost is basically there to really amplify the signal. So it is important to use the right combination of the prime and the boost. And the boost always should be a stronger vaccine than the prime. So for example, you always start with the DNA followed by a viral vector, but you don't do the other way. You give the viral vector and you don't come in with the DNA, because DNA is not a strong boost compared to the viral vector. So the order of the usage is also important. And coming to the TB vaccine. So what was the question?
Question was that whereas for SARS-CoV-2, the vaccine could be developed easily.
Yeah, so for the TB,
But for
basically
TB
it is an intracellular pathogen, and it is a complex pathogen, and it is antibodies. We still don't know what is the role of the antibodies in protection against TB. So dependent on the T cell based protection. So all these make it complicated. Just like any chronic infection, it's hard to develop a vaccine against.
So do you think like in 100 days, a vaccine going to generate a bona fide memory T cells, which is one of the long-term T cell memory is a cardinal feature of the success of the vaccine, whereas in 100 days, we may not get bona fide memory.
And the issue with the TB would also be the exposure to the lungs, the dormancy, the slow growth, and you could definitely develop a vaccine, but I'm not sure we know for sure what are all the antigens that we need to be targeting. So you need to have more antigens to be targeted to get a better protection. So there is more complexity involved with the TB.
So Professor Amra, one question I had, you prepared this MVA COVID vaccine, single dose, and you went up to NHPs. What is the durability of antibody after a single dose? Because you said the durability is a very important component with MVA-based vaccines, and the T cell responses are also very good. Can you give an idea? Because both are primary characteristics of MVA as a pox virus, vaccinia virus. But when you have this vectored vaccine, does the vector the other antigen also behave similarly?
Yeah, that's a good question. We haven't checked the durability for the COVID vaccines that much because there was a big need for showing protection. So we typically challenge the animals either after 4 weeks or 8 weeks after the final vaccination. So we don't know specifically for that.
Yeah, because that was one of the key characteristics you said with MVA as a vector, the durability enhances tremendously.
Yeah, we can at least we can say in the first 8 weeks or so, there is very little contraction. But I think for when we talk about durability, we should really think about months.
Years, months and years. Yeah. Okay. Thank you. Yes. Please, one of the hands here.
Hello, good afternoon, everyone. I'm Dr. Shazina Saeed, and I'm a public health geneticist by training. So thank you to THISTI for organizing this amazing meet. And I think my best answer could be given by Dr. Ankur. Traditionally, we know vaccine development, which goes through various phases of the trial before it goes out into the population. So when you have this very short duration, because it's a pandemic or an epidemic that the population, can you explain from the public health perspective of the pros and the cons of the population?
Sorry.
Yeah, that's okay. I can repeat. So traditionally, we know that vaccine development goes through various phases of the trial, etc. But when it is, when you have to put out something into the population from a very short period, what are the public health perspectives of, the pros and the cons? Just for my understanding. Thank you.
Yeah, no, I think that's a very relevant question. I think safety would never be minimized, and that's why those prototype libraries and the candidates, the platforms and everything, all the information that we would collect beforehand would be important in deciding what size of trial should be done, what is the dose that you should go for to start with, and that is going to be the likely final vaccine dose as well. So all of those factors would have to come from that preparedness work that you have done beforehand. So you're absolutely right. This is not something that you can squeeze in every case. And here, as Dr. Arora just mentioned, we are really talking about pandemic vaccines where threat is really high and you need to come up with something to save millions of lives very, very quickly. So.
Yeah. So I have two questions.
Yeah, please go ahead.
One is pertaining to the regulatory, maybe Dr. Shaligram can say because of his vast experience in this. So what has been historically the various roadblocks, regulatory roadblocks that had occurred in our country, and how things have changed since COVID? And what are the action points we can take in the future pandemics, preparedness? That's one question. The second question is, all the talks boils down to how important is surveillance. So what are the efforts taken by maybe CEPI or the other institutes involved with respect to the surveillance of the ill-known outbreaks that occur, like Chandipura or other things that are generally there, but it's not coming up. So what are those efforts being taken?
If I may answer from a laboratory.
Let Dr. Saligram respond to the regulatory response.
Yeah. So thank you very much. It's a very good question. So certainly, in COVID, what has happened was there was a very nice coordination complex which got built, and that's why in the early my talk, I thank Professor Vijay Raghavan. So that coordination complex which used to drive, because there are several regulation one has to cross through, and it's complex and it involves a lot of steps you have to get through when product has to go there, which is obvious needed because one has to scrutinize every other data which is generated from a larger exposure perspective. That has to happen. So generally this procedure takes years together for a classical vaccine. In COVID, all that happened was this coordination complex of moving things faster, and regulators work day and night. There were times when they used to be there, switch on at night 2 o'clock. I remember Dr. Somani calling me night 2 o'clock and 3 o'clock also while reviewing the files. And so it's just all that everything was done due to the pandemic nature, due to the people. There are so many deaths were occurring. So everything was done on a fast track without, I think, much of the what they would do classically. But classically, they would take their own time. Here they had to put extra time and energy, and though lockdowns were there, so everybody came together and experts came together, made themselves available very quickly in a just calls time or emails time. So this is what has happened.
What is the status now?
Now the status is obviously, one must give a little relaxation time to everyone. It is not you cannot run this stress system continuously. Actually, you have to give a people sufficient time to really review data, because a lot of things and a lot of understanding do not happen just like you submit something and then immediately you have an approval. It's all about communication. It's all about understanding. So you have to give a time. So, again, for regular diseases, it is taking its own time, which is okay, which I personally, if you ask me, we don't find any issues around that. But certainly, if there are some improvements which can happen, will be great. Basically aligning of all global regulatory system to one way of doing the things would really make a lot of sense, because if the pandemics spread across, then one regulator has approved, then you have approval everywhere. Otherwise, amount of stress a manufacturer has to go through in terms of GMP audits after audits, and then you have to keep manufacturing and supplying and all sorts of things. It's too much. But therefore, bringing this with our regulatory system with a global regulatory alignment would be the ideal one. So our approval would be considered as good as a US FDA approval or EMA approval would be the most ideal way of looking at it, actually, provided all that happens in a sufficiently short period of time, so that there is no shortcuts. There is a sufficient review time for the data. That's how it should happen. So it's a discussion which will take a bit longer time. So I have concised it in a short answer.
Tomorrow we have a session also, and maybe some other things. Priya.
Sorry, I forgot about the first question and was answering the second question. So with regard to surveillance, again, trainings happen imagining, like right now, diagnostics for the Sudan Ebola virus are happening. The government has started looking into that, because anytime a disease happening elsewhere can easily get into another space. And so we've trained people for Ebola, for yellow fever. We're developing diagnostics for the Sudan Ebola. So I think that because of that preparedness from the laboratory side, and having a wider network that's trained, at least detection, the first alarm trigger that was referred to earlier is almost there.
Yeah, I would just kind of share the information that DBT has started a program for novel pathogen discovery program, where known and unknown pathogens would be looked at some key points across the country, Northeast, Western Ghats, and some of the plateau area where KFT and other things are there. So there is a effort going on. Yes.
Sorry, I just wanted to add that for diagnostics also, FIND has its own 100-day mission. So, here we are talking of vaccines from a CEPI perspective. And I think, again, it all sort of boils down to that preparedness, that risk taking during peacetime, so that you are ready in the crisis time. And so that 100-day period is really the crisis period. In fact, even if you go back to when CEPI was established, it was really a risk taken in establishing CEPI because at that time there was no risk of pandemic in sight. So, when Government of India, Norwegian government, Gates Foundation and Wellcome came together to establish something like CEPI, and then saw really the benefit of that when really COVID struck. I think it sort of highlights how important preparedness is.
Yeah.
So can I ask the next
Yeah,
question?
please go ahead. Yeah. So we will just take two more questions.
Yeah. So I am Anamika Gambhir. I'm from Department of Biotechnology. So government is preparing its roadmap, technology roadmap for 2047, and DBT happens to be leading the effort alongside 12 other government departments. So my question is basically to the industry, Serum Institute, as well as to the academic partners. To Serum Institute in particular, one of my questions has already been asked by the previous speaker as to, because we have been asked to list out the challenges that the industry or the academicians are facing in terms of one, technology transfer. And my specific question to Serum and other representatives from industry would be how better we can incentivize industry in terms of their R&D spend. We keep talking about government to increase its percent GDP, spent in S&T R&D. But how best can we do to incentivize industry? And a very candid admission, because these recommendations are going to be, given out to the cabinet as well as to the PM, and they are going to, look at it for very early interventions. Thank you.
Yeah. Umesh, would you like to answer that?
Yeah, thank you. So very, very relevant question, actually. Yes, in fact, we always feel that incentivization to innovation is not only about India, but probably India is way better, but it's a big challenge globally. We have had several experiences, including our rotavirus vaccine, which was thermostable, and everybody wants a thermostable vaccine. But when time came, everybody was going with a classical vaccine. So certainly innovation needs an incentivization, which is always the case. There are several models which are already being used by the Western world, like a BARDA and other models. But I think each model has to fit to the country's need. You can't have those kind of models running for a country like India, which is LMIC. I think the idea of really creating a little different translational machinery for early development is a key thing, because more de-risk program, if it is proof of concept, clinical proof of concept of a product in terms of method of manufacturing and in terms of a clinical outcome with a short and concise trial, if that is available, and that's where what we are talking with Dr. Garg also on THTSI, if we can come out with that early translated product and a very well characterized and or good enough method of manufacturing, then scaling up by industry and providing that would be really a focus of industry. So that work, if that can be done, that will be a great help. And in that one also, when regulators are already integrated, then regulators knows, oh, this is an innovation and this is how it is getting translated. It is done by an independent agencies like THTSI or ICM or whatever it is, whichever is this one. This would have a real good acceptance, and then scaling it up and taking it to phase 2 by 3 by industry, and obviously then making it a cost effectively available product to the public is certainly industry would be very happy to do that. Now, I think more is de-risking strategy in early science is a very important aspect from regulatory standpoint as well as from the translational standpoint is a very important strategy for a country like India. Instead of trying to really fund and pump in too much of money is not the right way. In fact, translating in a short possible time and giving an asset which is really well proven asset or kind of going to be successful asset, then industry feels comfortable. Oh, this is going to be a success story. Then they are ready to take a further risk and translate it further. So that's the very important aspect which needs to be driven in country like India, actually. That's what is my opinion. Thank you. But I will send you a email with that respect, giving our views on this.
Thank you. You want to make some comment? No.
Also,
There is one last question. I think.
What Dr. Shaligram is saying. So you are.
Your mic is not working. Yeah.
It's working, sir. When you look at your Oxford partnership, it's a very successful partnership, Serum and Oxford University partnership. What is it that Oxford University is doing right, and what is it? What are the challenges you face with the Indian Academy?
Same question.
Difficult. Yeah. So I'll
Like-
tell you. To be very frank, I don't know, but all that I'll tell you, it is not about doing right or it's just a chance event. It's just an interaction. And probably that's what when we talked first, Dr. Shailaja, with Professor Garg, I personally felt that it is more about getting to know what all science is going on, where that science is going on. It's more about a marketing of innovation and reachability of innovation happening in the institutes in India to the minds, decision makers, internal scientific champions within the industry is a very important aspect, which is a missing aspect, what we called as a business development function. You have to sell what you're doing actually. When you sell it, you push it, then people come to know. And that's how it clicks actually. And that's what happened in case
Can
of.
Can I make one comment?
Yes, please.
Actually,
That's
I
your
was
last
about to ask whatever you have asked. So we came to know about your take on vaccine production or vaccine development. That is in terms of the commercial agency, you are vaccine producing agency. I would like to know from academicians, academic institute like NIV and THTSI from Dr. Guru and Dr. Priya, what roadblock you face. You have many of the academic institutes, they have several candidate vaccines. And even I come from one academic institute, we find it very difficult to translate it to, and what Dr. Shaligram says, to translate it to the commercial level and to the commercial agency should accept whatever he is telling, should accept our candidate vaccine. So what is your roadblock, if you can say in personal level, then, and I think I understand that there is from the initiation of the development of vaccine, there should be some collaborative effect, collaborating activities among the commercial agency as well as the academic. But why the foreign academics will be successful and not the Indian academics? That is our great question.
In fact, you have given the answer. The answer is that right when we start working academics, at that time collaboration is required so that industry and the the requirement of each side are different. The speed of working of each side is different. The imperatives are also different. But if they come together, then, and that is why some kind of a marriage has to take place right in the beginning. It may be arranged marriage or love marriage, but I think academics and industry has to come together. And I think example, Priya probably can give the example of Bharat Biotech and ICMR, that they came together pretty early and decided to work together. And that's why translation of science into product becomes feasible. One last question to Dr. Mutreja, that one of the key issue which is emerging is people getting together and working together. So for regional networks and collaborations, what is CEPI going to, what is the CEPI's strategy? I think this would be the final question.
So no, I think that's absolutely important. And that's why out of the 3 CEPI 2.0 pillars, we have one full pillar just for this connect side of work. And that connect side of work, doesn't just work on the technology or the pieces of the vaccine development value chain. That also connects various stakeholders like governments from advocacy perspectives, from community leaders, so that vaccine acceptance or vaccine awareness becomes more. And also, of course, from technology perspective, when we think of central laboratory network, when we think of epidemiology network, regulatory network, manufacturing facility network, I think having all of these, as I think it has been discussed at length, keeping all of these activities warm during peacetime is actually a risk-taking process. And that's exactly what CEPI is investing into and facilitating. So that's how we're trying to keep those networks ongoing.
Thank you very much. I think all of our speakers deserve a big round of applause. Thank you, everybody. Thank you.
To hand over a memento to all the speakers as a token of appreciation. Thank you so much, sir. So with this, we end our first session. We request you all to now proceed for lunch outside the auditorium, and we will begin our next session at. So can we have a memento for our chair, Dr. N.K. Williams? So we have another one for you. So I would request Dr. Shelja to please thank our session chair by gifting him a memento. Thank you, ma'am. Thank you, sir. So we have lunch outside the auditorium, and we will begin our next session at 2:45 p.m. So I would also request all the speakers of the second session to hand over their presentations to Dr. Shankar, Dr. Priyanka, or the technical staff on the dais. Thank you.
hello, 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 mic check, hello, hello, hello.
Welcome back. To chair our second session on academic preparedness and industry readiness, I would request Dr. Sudhanshu Vrati, the Executive Director at Regional Center for Biotech, to come on the stage. Dr. Vrati is known for his studies in the field of RNA virus replication and vaccine development. He is an elected fellow of the National Academy of Sciences and Indian Academy of Sciences. The Department of Biotech awarded him the National Bioscience Award for Career Development, one of the highest Indian science awards, for his contribution to biosciences in 2003. Sir, I request you to please come on the stage.
I don't think. Hello? Good afternoon, everybody. Welcome to session 2 of this important meeting. The session is about academic preparedness and industry readiness. And this is very important area in the context of the limited academia and industry interaction that we have in this country. It's also important from the point of view that our academic laboratories, are they ready to work in a GLP environment which is required for conducting research that could then be passed on to industry for commercialization. Similarly, is our industry ready and meets the expectations of our academic laboratories to absorb the innovation and the quality of research that goes on in these labs. So in this context, it is very important to have a dialogue to understand how well prepared our academic labs are to conduct research that can be commercialized, as well as industry, are they ready to absorb what is being produced in our academic labs. And the interaction between these two is very, very important for success of the mission that we are talking about, whether we can produce a vaccine in 100 days or 365 days, as was said in the morning. So we have several speakers which are drawn out from academia as well as industry who would be giving you talking about their work, and maybe they will allude to the issue that I have raised. So we are looking forward to hearing from them. There are 7 speakers in this session, and you know that everybody is given 15 minutes time so that we have some time left for discussion. Please stick to time. And is there a timer, or should I time it myself? I'll be timing it. So I will let you know 2 minutes before your scheduled time for completing your presentation. So at around 13 minutes, I will somehow draw your attention to the time that is running out, and then request you to please finish quickly so that we have some time left for some interaction. So I would like to invite the first speaker for the second session, Dr. Sweety Samal. She's from THSTI, and she'll be talking about a novel vaccine platform for the development of pan-beta coronavirus vaccine. Sweety has over 15 years of research experience in the area of infectious disease biology, with a focus on viruses such as paramyxovirus, influenza, HIV, dengue, and SARS coronavirus 2. Her lab has developed a multi-domain nano-cage-based vaccine platform, and this platform has recently been awarded $20 million by the CEPI for the development of a broadly effective beta coronavirus vaccine. So we look forward to hearing you. Sweety, please go ahead.
Thank you, sir, for your kind introduction. Good afternoon, everyone, and welcome back. Our lab is mainly focusing on the influenza and respiratory viruses. And today I will briefly walk you through one of the novel vaccine platform that we have developed after this corona pandemic, which are currently we are using not only for the development of the pan-beta corona vaccine, but it might be useful for the development of the other vaccines of different pathogens. So COVID-19 is caused by the SARS-CoV-2 virus, and it belongs to the Coronaviridae family, which contains a large number of viruses, which are mainly responsible for causing common cold to severe respiratory diseases. So these coronavirus family has been long back neglected because mainly most of the viruses from this family cause the common cold, which is a very self-limiting disease. However, the first pandemic of the 21st century was by the SARS virus in 2002, which has occurred in the China and later on spread to more than 25 countries. And this is the first hit to the scientists that these coronaviruses can easily spill over from the animals. 10 years down the lane, another virus emerged known as the Middle East respiratory virus from the infected dromedary camels, and the human-to-human transmission has been established. Even though this virus is not similar to the COVID-19, which has a very high transmissibility rate, but currently also the MERS outbreak is there in the Middle East region and some many parts of the Southeast Asia. And if you look at the disease severity, the case fatality is around 35% as compared to the COVID-19. And as you can look at the timeline, 7 years after the emergence of the MERS, we all have witnessed the COVID-19 pandemic. And you can see that these coronaviruses now rapidly emerging and mutating, and it can cross the host barrier, and we don't know what will be the next pandemic from this virus family. So this is a snapshot which has been taken from the early when the Wuhan outbreak occurred. And if you look at the difference between the SARS-CoV-2 as compared to the SARS and MERS, the SARS and MERS, even from the same family, the transmission has been restricted to minimum country. Maybe during the 2002 or 12, the international travels were less than COVID-19. But if you look at the case fatality rate, however, COVID-19 has known later on has high transmission rate and high infectivity as compared to the SARS-CoV-1, in which it more than 90% similarity is there both in the phenotypical and biological behavior. But if you look at the case fatality, MERS still hits the top between the SARS and COVID-19, and yet we do not have any vaccine available for the MERS, and there are different reasons. There is also a scientific reason, and another reason, as we heard earlier, that the acceptance or the industry willingness to develop a vaccine which will be useful and have the market value. So we started working on the vaccine in early 2021, where as a virologist, we understood it is the SARS-CoV-2 is very different from the SARS and MERS virus. If you look at the SARS and MERS virus, it is highly stable, and unlike other RNA viruses such as influenza or HIV-1, the mutation rate is quite low, and the proofreading mechanism is very much evolved in SARS and MERS. But this is not the case of the COVID-19. After the Wuhan outbreak in 2019 December, the first mutation appeared D614G, and later on we have encountered the Alpha, and in the early 2021, everybody has witnessed the Delta surge, which has pushed the healthcare system up to the brink, and lakhs of people have succumbed to this Delta virus, which is still considered the highest pathogenic virus in the SARS-CoV-2 family. So that is the time we talked and we started brainstorming whether there is a need of a pan-beta corona vaccine against emerging SARS-CoV-2 variants of concern. And by that time, I think all over the world, in India, all the people, I mean, every scientist has been started working on the COVID-19, and there are many people from the, every vaccine industry is also working for the SARS-CoV-2. So we do not want it to duplicate the efforts. Rather we anticipated that the vaccine, which has been made of the Wuhan 1 spike protein, might not be able to give the full protection to the emerging variants. So taking into that account, we have taken a very high risk approach, whether we can have a novel all-in-one vaccine developed to tackle this future coronavirus threats. And from the morning session, we have heard there are a lot of aspects we need to take it into account where we are developing as a vaccine. That's for that first. It is also very necessary for the academic institute to understand what the industry needs and whether this novel approach can be translated to the market. So we strictly put some of the consideration that we need a platform where we can plug and play, where there will be a flexibility to display or express more than single antigen, epitopes or domain of interest. We should use a very convenient and robust expression system. As we heard from Professor Rama, one of the bottleneck for MVA vector to go into the clinic to the market is unavailability of the cell line. So we also wanted to have a very robust expression system to express this vaccine construct. And also with that time, we know that mRNA vaccine, even though has been proven very good for COVID-19, but there is a stability issue and storage issue. And for the country like India, where in the summer, the temperature goes up to 50 degrees. You need a vaccine which can be stored, if not at the room temperature, but at least in the 4 degree. And there should not be any antibody-dependent enhancement. Even for the COVID-19, SARS-CoV-2, AD is not a major phenomena, but one of this aspect has been seen in MERS, where in the early emergence of the MERS, the inactivated virus vaccine has been used, which has been shown to cause antibody-dependent enhancement. So these are the factors which we have strictly incorporated to think that whether we can develop a vaccine platform which can tackle and which will have all these characteristics. So one of the platform that interested us is the self-assembling protein nanocage platforms for nanoparticle vaccine. And this is a very emerging technology, and many research groups are started working it, and Dr. Raghavan is also working on this platform. So protein nanocages, they self-assemble and multiple antigens can be displayed in the nanoparticle cages. Another positivity, another advantage of this nanoparticle-based vaccine, that it has been shown that they can induce very robust and high magnitude of both the humoral and T-cell responses. And one of the widely used nanoparticle is the ferritin, which is the iron nanocage, and it is found in all living organisms, and that is widely used for development of the many nanocage-based vaccine platform, and similar for the SARS-CoV-2 and influenza. But once again, we didn't want to duplicate the efforts which has already been done by others. So we kept on digging into the literature to find that whether we can have another kind of this nanocase system which we can use for the development of our vaccine platform. So we have come because that time, I guess everybody started reading about the SARS-CoV-1, MERS, and the coronavirus. So we have come across the paper and we found that the coronavirus contains one non-structural protein which has the self-assembled property, and it can form nanocage. So the N-terminal and C-terminal domain of the nanocage is open, and it usually interacts with other non-structural proteins and help for the viral replication. So we wanted to exploit this phenomena of this NSP protein for the development of a vaccine. So this NSP self-assembled to form globular nanocage with antigenic epitopes which can be displayed on the surface. And you have the flexibility to staple the epitopes, and we wanted to express it in E. coli. And as you know, so far for viral antigens, it is very difficult to express in E. coli because most of the viral antigens, they need a native-like structure, or for the influenza and HIV, the glycosylation also plays an important role of the neutralizing response. But for the COVID, SARS-CoV-2, again, we are lucky for the spike and RBD, even though monomer and glycosylation doesn't play that much important role on elicitation of neutralizing antibody. So we have taken the antigens from the antigenic region. It can range from 20 mer to 200 amino acids. So today I will not show you the failures, but 2 success stories that led us in the belief that this vaccine platform can be used for other vaccines too. Okay. So a little bit about that non-structural proteins. It forms a dodecahedral structure. That means you can stitch 12 epitopes on the N terminal and 12 epitopes on the C terminal. So if you want to have one epitope for RBD, so if it is displayed properly, then you will have a 24 epitopes of the RBD. That means your antigen is more, yield is more, so definitely your cost of production will go less. Another also important aspect of having this non-structural protein, because later on lot of paper has come up that the non-structural NSP protein, which is not only highly conserved across the coronaviruses, but it can induce the T cell responses. So you have two arms you can induce through this method. You can have your display antigens, which could induce the humoral and neutralizing responses. And you also have the NSP, which can also elicit the T cell responses. Here you can play around the number of epitopes, type of epitope or domain, and the length of epitope. So even though we started with the spike protein, but here you can also include another protein of interest. It might be from the nucleoprotein, it can be the matrix protein, it can be the envelope protein. And you can also stitch epitopes from different viruses. So here I'm showing one data, which is a proof of concept, where we have developed an indigenous multivalent nanocage-based vaccine against SARS-CoV-2 variant of concern, where we have taken conserved 20 more epitopes from SARS-CoV-2 Delta and Omicron, which is highly conserved in the N terminal. And in the C terminal, we have taken the conserved fusion peptide and peptide repeat domain from the spike 2 domain, which is highly conserved across this variant of concern. So that forms nearly 80 Na multimeric particles. It is stable at 4 degrees for more than 2 months. And these are all tagless, which has been purified in the E. coli at the lab scale up to 50 to 80 mg per liter. So it is definitely interest for the industry. And if you give intramuscular immunization, along with mainly squalene-based adjuvant, it can induce robust antibody and significant T cell response. So we have also conducted one prime boost challenge study with this drug substance, and we found significant protection with squalene-based adjuvant. And here I'm showing the data where it has neutralizes the Omicron variant. So based on the concept, we have, it has taken by the CEPI for development of the pan-beta corona vaccine, which will consist of SARS-CoV-1, MERS, and MERS epitope. These also form 80 to 100 multimeric structure. It is highly homogeneous and the yield is around 80 to 100 mg. And here you can see an ELISA humoral response, and you can see that the antibody can bind to all the MERS spike RBD, Wuhan spike, SARS-CoV-1 RBD, Wuhan RBD, Delta, Alpha, and Omicron RBD. And all this RBD also prepared in the mammalian expression system in our lab. It also produces very good T cell response and also neutralizes the Alpha and Omicron virus. And we are taking few of the construct for the preclinical challenge study. And we are also looking at the neutralization response against the SARS-CoV-1 and MERS. So definitely this concept, at least for the coronaviruses, fall into 100 days vaccine production, because it can be rapidly and easily tweaked for new variants and novel coronavirus. And because the expression system is quite handy and feasible, you can very easily and with a short span, you can screen a large number of antigen. So, and also being on the CEPI vaccine network, it also helps us to understand what other consortiums are doing, and we can refine this novel vaccine platform. And definitely it's a very high risk project, and it started from the lab note of THSTA, and within the less than 365 days, we have taken for the formulation development, and it would not have possible without the team from the top leader who has believed on the concept, scientifically guided us, and all the team members who have relentlessly worked to have this vaccine come forward. So this platform also could be used for the other vaccines because where you would like to display multiple antigens. And with this, I would like to thank you all for your attention. I'll be happy to take your questions. Thank you.
Thank you. Thank you, sweetie. We'll take a few questions now. Yeah, Rama.
So which NSP did you use?
We are still on the PCT filing, so I can't disclose right now.
Oh, no problem.
So, I have a question. When you said pan beta coronavirus vaccine, the way your vaccine is designed now is against the known beta coronaviruses. Okay?
Yes, sir.
I thought that you would come up with a vaccine that if there's any beta coronavirus
Yes, sir.
that emerges from anywhere, this
Yes.
vaccine would work.
No, the thing is that right now we are doing for the SARS, like suppose right now the Omicron 2.75 is in the circulation. So we can take that epitope and fix it there. So anytime you can just take out and plug it.
Yeah, but is it possible to make a vaccine which will work against any future emerging variant?
No, sir, I think it will be very difficult unless. Because, so the mutation, the virus knows also how to survive. That's why it puts the mutation.
No, so virus is actually mutating mostly around the receptor binding site to, give its advantage. But I'm sure there are enough proteins which are highly immunogenic and are, not mutating.
So,
can we not use them?
Yeah, so we have put conserved domains, but if you look at the MERS is a little bit distance variant than the SARS-CoV-1 and SARS-CoV-2. But if you take only the SARS-CoV-2 variant I'm concerned, that's what I'm saying, that we have taken the conserved reason. That's why you are seeing we have taken the Omicron epitope, but we see the protection against Wuhan, Delta, Omicron, Alpha. So in the variant of concern, it can be possible, but if you are taking the virus from the same family, different viruses, unless and until they are distance, they are closely related, it will be a little bit difficult to put the conserved domains. So there are some conserved domains are available, and this one is working with the conserved domains well, at least for the coronaviruses, but for the other viruses, it may or may not work.
Yeah, Priya.
Sweetie, I just wanted to ask you, you have not, the question is along the lines of Dr. Vrati's. You have not included any bat coronavirus or any animal source coronavirus?
That's what our next strategy, like to include virus. Right now we are working on virus. We'll include more the virus. As I told you, it's, this is a Nobel platform, and in the academic matching with the industry pace. So we have prioritized, we have first completed with the VOC, then we are completing with the pan beta corona vaccine, and then we will include the MBECO and bat epitopes.
Right.
So how much can be taken up in this platform? Like how many epitopes or how many?
Right now, yeah. So because this nano case has two terminal, N terminal and C terminal, we started with 20 amino acid. Now we have reached up to 200 amino acids. So in one of the construct, we have able to purify with RBD alpha in one part and RBD Omicron in one hand. So it can take up to 200 amino acid. And we are also working with the monkeypox, where we are able to stitch all the 4 protein of the monkeypox.
Right.
Mahesh Murthy, CMC Fellow. This is really great work, sweetie. So the question I had is, there is a wide amount of variation between MERS and SARS-CoV in terms of their receptor binding domains and the receptors they use. So how prudent is this strategy to if you have a novel virus, you don't know, only if it is, for example, binding to ACE2, you will then use it in this
No.
platform. Is that how it works?
No, here, this is a platform. You can put any kind of antigen. So, and also we have seen that if you put the N-terminal, the expression is more. What we do, we took, suppose SARS and SARS-CoV-1, they use the AC2, but still there is a difference between the RBD. So you will link it with a different, you take the MERS, which has the antigen binding site with DPP4. So we use a lot of bioinformatic analysis. Then we take in the epitopes, which can be 20 more, 30 more, or 40 more. Then we linked it with the linkers, and then we stitch it with the nano cage.
So the second question I had is, you're using the heptad repeat region.
Yes.
Now it's really conserved under SARS-CoV-2, but it's slightly different when you want to look at between MERS and SARS.
Yes.
So do you do that individually for each one?
No,
or
that's the beauty of this platform. You can take from 20 to 200 amino acids. This HR2 domains are only 20 mer to 29 mer peptide repeat. So you can just fuse it together.
So the strategy of including this was, now see, heptad repeat, and these are all basically T cell epitopes. They're not really neutralizing as such, right?
Yeah, but that is interesting in one.
Yeah, no, so I'm curious about the mechanism by which this actually happens.
No, these are not T cell epitopes. If you look at the fusion strategy of the spike protein, it starts with the pre-fusion and then ends with the post-fusion conformation. HR2, mainly the heptad repeat opens up when the virus attached, then opens up into the fusion process. So if you have a antibody against the fusion peptide and it binds to the fusion, then the virus cannot enter. So it can also have the antibody, and I mean, this works for other viruses in the similar way. So that's why recently, and we take this epitopes in a reverse vaccinology approach. Like a lot of paper, recently a science paper has identified a S2 domain from a monoclonal antibody. So we take that epitope and stitch it here. And then we see that whether we are seeing the neutralizing response. So far, the coronaviruses, it's working very nice, but we don't know for the other viruses.
And you also tried it for seasonal? Because when you say pan beta, you want to look at others as well.
At least we will go for the Embeko viruses. Sure. Beta coronavirus, again, it will be the animal vaccine perspective. Maybe slowly we'll go into that. Right now, we are concentrating on the beta coronaviruses.
Thank you.
Thank you, sweetie. We will now conclude.
Thank you. Thank you.
And we move to the next presentation by Dr. Harish Chandra from Zydus Life Sciences Limited. Dr. Chandra is currently working as General Manager, Vaccines and Diagnostics at Zydus Cadila, India. He has over 18 years of experience in the field of vaccines, biologicals and diagnostics, and has led many teams in analytical development, quality control, and process development for the development of various vaccines for human and veterinary use for both domestic and global markets. Dr. Chandra is giving a talk which is titled as Emergence of New Technologies in the Era of COVID-19, Paving the Way to Combat Future Pandemics. Dr. Chandra.
Yeah, good afternoon, everyone. First of all, I thank the organizing committee for inviting me for talk in this forum. My topic of talk is emergence of new technologies in the era of COVID-19, paving the way for future preparedness. But before I talk on these technological advances, I will introduce my organization. Zydus Life Sciences is a primarily pharmaceutical company, and they are relatively emerging player in the vaccines. Though we have been working for last 20 years in the vaccine space, but initially it was 1 or 2 products. Currently we have 15 products in our pipeline, and we have 6 manufacturing and 2 R&D sites. And right now we are going major upgradation of our facilities and scaling up different greenfield facilities. Also, we have dedicated manufacturing facility for our ZyCoV-D vaccine, which we have developed. So on Zydus capability front, we can have capability to conduct research from concept to first in man trial. So all the preclinical toxicology we do in-house. And phase 1 trial also we do in-house. We have wonderful facilities, and only phase 2 and phase 3 onward we outsource to third party. On Gyrus capability of different vaccine platforms, so we have capability for live attenuated viral vaccines, inactivated viral vaccines, polysaccharide and conjugate vaccines, recombinant subunit and VLP vaccine, toxoid vaccine, combination vaccine, and now newly added portfolio of DNA vaccines. In fact, we were the first company in India to launch H1N1 vaccine during the 2009 pandemic. Gyrus is also there in the diagnostic space, and as Dr. Priya Brahm specified in the morning that there are certain diseases which are rare, but they may emerge as a next pandemic. So we are already prepared, and we have in-licensed this technology from NIB Pune and already have manufacturing license. And if need arises, then we can manufacture the same in very short period of time for zero surveys. During COVID time, we have created rapid diagnostic facility also and developed SARS-CoV-2 rapid antigen manufacturing. And now we have 3 products already manufacturing license, and there are 5 to 6 going to add up. I want to mention about anti-SARS-CoV-2 antigen ELISA. We were the first company who launched this ELISA kit, and ICMR used our kit for the first zero survey in the country. Now, if we talk about COVID-19 vaccines, so when this COVID struck in December 2019 and first sequence was out in 2020, various companies rushed to develop the different vaccines and tried new vaccine platforms. But this time, new technologies have took center stage, maybe because of speed of development as well as scalability of the product. And we all know success of adeno-based vaccines. Others are the nucleic acid vaccines, where mRNA and DNA both were developed and commercialized. We have taken also the lead from the last MERS and SARS outbreak and research, and we have seen that in these diseases, DNA vaccine candidate were advanced into phase 2 clinical trials. So we decided to develop DNA vaccine against SARS-CoV-2. If you see our ZyCoV-D vaccine journey, after completion of preclinical toxicology, we have done phase 1/2 adaptive clinical trial, and then went on to do largest clinical phase 3 clinical trial in 28,000 subjects. And it was evaluated in various demographic in 12-year and older population. It was found to be safe and efficacious. But our vaccine was 3-dose vaccine. So we have done again clinical trial with 2-dose regimen with adjusted dose. And again, it was found equally effective. We have also done booster dose studies, but result analysis is under progress. Our effort came with the world's first plasmid DNA vaccines. It was truly indigenous. And I thank to DBT Bharat in believing this technology for funding, and IRSA Pune for analyzing the clinical samples. There are some unique attributes of our vaccines. It is thermostable, we can say. It is stable at 25 degrees for 2 months, and multiple freeze thaw did not impact the vaccine quality. We have also done 28-day open vial studies, and vaccine was found to be usable after 28 days. It is given through delivery device because only syringe and injection will not work in case of BNA because of poor uptake. So, but it works with this delivery device beautifully, and it is needle-free device, so it is also painless. It is a plug and play technology. In case of any variant, we can immediately synthesize the gene and replace the gene. We have already made so many variants and done the animal studies and have the data. It comes with digital support also to avoid the counterfeit, and these can be integrated in COVID software. During COVID pandemic, because this DNA vaccine requires large amount of dose, so it needs large infrastructure to build up. So in COVID period, we have created very huge infrastructure, and we can see here that we have installed one of the Asia's largest chromatography system and one of the world's largest cross-flow systems. Our whole facility is automated network of process tanks, so that with one button only it can operate entire process up from downstream to tangential flow, final filtration, everything. We have also set up syringe and adapter manufacturing facility in India, so that if need arises, we can provide the same. So in the end, I will say that we have done a lot, but still lot to be done. Like disease surveillance, it was discussed in the morning session that we don't have structured disease surveillance for animals, human or environment, because we have seen that a lot of human infection happens from animals. So until unless we have lead, it will be difficult to act. Definitely more vaccines and technologies are required, and we can get newer vaccine using these plug and play technologies to fulfill unmet needs, new adjuvant and formulation to increase potency as well as stability. In one of the CEPI reports, I have read that there are 260 viruses across 25 viral families known to infect human. So it is really hard that how to choose that which could be the potential virus. So CEPI is also advocating to make prototype against each virus family and keep the data ready, so that in case of any pandemic, we can directly switch over. We need more large scale production facility across the geographies to cater the demand rapidly. We need more BSL-3 facility for clinical and potency assays. During this COVID time, we have seen that there were only 2 laboratories. One was THSTI, one IRSA Pune was providing the PRNT services, and there was huge backlog. We also need local gene synthesis facility. Getting gene identified and synthesized, it takes around 30-40 days currently. We don't have any facility in India right now. For everything, we have to send outside, and then gene comes to us, and then work starts. So if you have local facility, then we can save these times during crisis. Also, we don't have GMP cell and virus bank characterization facility in India. For anything and everything, we need to outsource, and it takes 8 to 10 months before we get the data. So with all these effort, though we cannot stop the outbreak, but we can act early to prevent it becoming larger epidemic and pandemic. With this, I close my presentation and happy to take questions. Thank you.
Thank you, Dr. Chandra. Are there any questions? Yeah, please go ahead.
Govardhan Das from JNU, Jawaharlal Nehru University.
Yeah.
I think a DNA vaccine been introduced during the pandemic period only, right? So DNA vaccine, the safety, especially the long-term safety, we really do not know much about it. Still do you think we need to carry on all the DNA vaccine thing? And as my second question is, everybody is doing all this vaccine research, but I did not hear anybody talk about actually the longevity or durability. So is there any way to measure durability? If I understand it's not possible for such a short period of time, but one can really measure the, the memory T cells, especially central memory T cells, or stem cell like memory T cells. I did not see anybody measure. Do you do that kind of thing?
So similar question was asked by regulators also when we presented our data to them. And they asked to generate some more data. So we have done distribution studies in animals. and showed them that within 14 days, it disappears, and we could not detect any portion of DNA. We have done through RT-PCRs. Number two, we have generated 6-month antibody longevity data and submitted to the regulators. Though since it is new vaccines, so we don't know what will happen after 2 years or 10 years. Because data is not there in public domain. Until unless it is administered in large population, we will not know that what impact it will give.
I have a question myself. These DNA vaccines in general, they have had low immunogenicity
low
or
uptake,
efficacy.
yes, because of such low uptake.
So, what kind of research is being done to improve the uptake and therefore the immune responses?
So two things, sir. You can say merit or demerit, because DNA vaccine always require in large quantity to be administered. It will be in milligram quantity. So, and that too, it need some device, injector device. People have tried many devices, but thankfully this device, PharmaJet device, worked for us. And with 2 milligram and 3 milligram dose, we got good immunogenicity data.
Yeah, but that's about 60% or so, right?
66.6%.
Yeah.
Efficacy.
Yeah. So, by packaging DNA in some sort of lipid particles, would that improve immunogenicity?
Actually, initially we tried, during initial development, we tried to add different lipids, added various formulations, but we found that naked DNA worked the best. So that is why we did not move to those directions.
Okay, are there any? Yeah, over there.
Thank you. Thanks for the nice talk. I'm here.
Yeah, hi.
I just want to know, have you explored the possibility of uptake of these DNA, which is used as vaccine by microflora in gut or human microbiome? And what could be the possible implications if they get uptaken by these bacteria, which are known to uptake DNA from environment? And how could it affect
microflora in our gut?
Yes, because they are known to uptake DNA from environment, and that's how they.
No, we have not done any such study.
Rama.
Are you delivering intramuscularly or intradermal?
Intradermal.
Okay, so that seems to be like a very high dose, right?
Yeah.
Anyway, congratulations on the licensure.
Yeah, thank you.
Okay, just one over there at the back.
Yeah, excuse me, sir.
Yeah, sure.
Did you try it with any adjuvants in your vaccine formulation? Because as a construct, you can clone your genes. In addition to your gene which you have cloned, did you try it with any adjuvants, genetic adjuvants? Because if you add genetic adjuvants, it could help you to increase your immunogenicity instead of electroporation, PharmaJet, these kind of devices.
Which adjuvant?
Genetic adjuvants. Big.
Genetic
Adjuvants, yeah. This additional gene, you can say. Did you try it with any adjuvants in your vaccine?
Yeah, we tried various adjuvants known to induce the immunogenicity, and we could find that naked DNA works the best. Initial part of development, we have tried lot of adjuvants.
Expressing along with your construct, along with your gene which you used. In addition to your gene or separate formulation?
In addition to our gene, yes.
So was there any antibiotic resistance marker in the DNA plasmids, or were they free of any marker? And if so, how did you propagate them?
Yeah, to grow the DNA in bacteria, you need to have some marker. It is with kanamycin marker.
But that can also then, as one of the questions if it can spread to the gut microflora or to the environment in general. Of course, better than ampicillin, but still, I mean, is that a concern or this is not a
Because
concern?
ampicillin is not advisable for manufacturing drugs to be injected in human. That is why we have used kanamycin.
Okay, we will now conclude. Thank you very much, Dr. Chandra, and move to the next presentation by Dr. Sudeep Kumar from Biological E Private Limited. He'll be talking about manufacturing preparedness. Dr. Kumar has more than 20 years of experience in biopharma, from large-scale fermentation-based products to recombinant therapeutic vaccines, proteins, diagnostics. He has worked from R&D to tech transfer and manufacturing of different molecules. He also established the VLP technology platform for different vaccines in India in collaboration with Novavax. He is also actively involved in the development of novel therapeutic proteins for cancer therapy. Sudeep.
So good afternoon to all of you. And first of all, I would like to thanks to the organizing committee and to the chairperson regarding giving this opportunity to make the presentation. Since morning, I am hearing a lot in terms of the development of vaccine to minimize the timeline in terms of the development. But I'm not going to talk anything which is related to the development. I'm more on to the manufacturing side, and I will take you through that even if everything has been developed on time on a fast track, what extra precaution, what extra care has to be taken in the manufacturing, because as we all know that vaccine is a large scale game. It's not like a therapeutic protein or vaccines, which require a huge quantity in terms of producing and supplying to the different countries or at different place. And in the morning, Dr. Raghavan has well said, couple of my slide will be representing that also, because there is a lot of things which need to be worked out in R&D, even if you have developed the molecule. But finally and ultimately, the manufacturing plays a big role to reach to the different patient and to the population. Now, if we look in terms of the last century, there are 5 global pandemics, but out of that, maybe 4 global pandemic was restricted either to some country or maybe to some region of the world. Only the COVID-19 vaccine was worldwide. And that is the problem in terms of supplying the material and producing in large scale. Given the major burden to the society of individual, international organization, government, and to the research and pharmaceutical industry, which has invested a lot of money in terms of developing the molecule, and also the industry is ready to keep on investing as long as we can protect the human life. The novel vaccine technology, which right from the morning we are hearing, that which claim to be developed faster than the existing vaccine. Most of the existing vaccine is taking more than 5 years in terms of the development and finally to the commercialization. Only in case of pandemic, we have seen that due to the effort of the scientists, and also there is a lot of support from a different regulatory agencies, this particular vaccine has come to the market very fast. Although there is a lot of things need to be still review and work out in terms of the mass production of this vaccine. Now, the pandemic illuminated long-standing challenges in globally, and respond to the pandemic biopharmaceutical manufacturing ecosystem, which was unprepared till now. Only in the last couple of years, most of the manufacturing companies, they have started building the facility in such a way that any such type of pandemic situation will come, we are able to handle it in a proper way. The coronavirus disease pandemic has prompted rapid investigation and deployment of vaccine platform, which has never been used. We have heard about the mRNA. We have also just now heard about the DNA vaccine, which was not at all there before the COVID vaccine. The COVID-19 vaccine success has generated interest in emerging vaccine technology for potential application. Now, how can epidemic disease be controlled in future? Everybody knows there is nothing new in this, that a vaccine which, while it cannot prevent outbreak, but can stop from spreading if it is given in time. The outbreak must be detected rapidly. So from the morning, we are hearing a lot in terms of the diagnostic kit, in terms of sequencing the gene and all those things. So the first and the foremost important point is at the initial stage, we have to detect rapidly, and so that the risk group can be targeted for vaccination and availability of vaccine in time. The manufacturing network have shown how effective manufacturing and supply chain network can drive down the response time on everything from PP kit development and delivery. Now in the morning, Dr. Raghavan has well elaborated this point, but again to recap all the people who were there in the morning, who are not there in the morning, is that if we look across the global COVID-19 created development from scratch to manufacturing, which include the testing in animal and human and brought to the public, and considering that this was a total new disease, many first place possibilities are very clear. Now, it is possible to design a vaccine candidate within 2 days of genetic sequence of a new virus being published. So that we have already seen during this COVID period. It is possible to move into the first human trial in 66 days from the release of genetic sequence, which Moderna has did. It is also possible to publish the first safety data in 63 days after phase 1 clinical trial, which is Moderna did. And it is possible to go from first human clinical trial to vaccine registration in about 7 months, which Pfizer BioNTech did. It is possible to get emergency use approval within a week after filling required data from the regulator. Now, till that point, we have seen in the COVID vaccine that all these are possibility is to bring the vaccine fast into the market. But the actual problem is start after this when you go for the mass production. The first vaccine which was approved, the quantity was not available in such a large quantity where the people have got the vaccinated. It took a little bit more longer time in terms of building the facility, in terms of the manufacturing. The current pandemic has highlighted the exposure of low and middle income countries to global vaccine supply shortage, despite effort by industry to increase manufacturing capacity, including in large developing countries. This challenge extends well beyond COVID-19. Now, what is possible now? No, sorry. Now, I will take you in terms of what is the hurdle we have faced since we have made millions and millions of doses in biological E for COVID vaccine. We know what are the challenges we have faced and how we have overcome about that. Now, if you look in terms of the manufacturing, there are two different clear-cut paths. One is a drug substance and one is a drug product. Now, drug product, if you look, most of the vaccine manufacturing company and maybe some of the pharma company who's making injectable, they are quite equipped in terms of doing the blending and filling in vial or in PFS. To expand the drug product facility is not a very big challenge. It is slightly easier than the drug substance side. Now on the drug substance side, to scale up and extend the manufacturing facility, it takes a very long time because you need to have all GMP requirement, regulatory approval, and also if you want to build a new facility, it is not going to take less than 1.5 or 2 years. So what is the possibility in terms of any pandemic outbreak? What is the best possible thing we can do is that we can increase the existing facility to an extent where there is a certain possibility of moving disposable equipment, fermenter or disposable column in terms of doing the manufacturing. However, on the drug product side, it is very easy in terms of bringing another high-speed filling line and place it into the controlled environment or class B, because normally it is required class A supported by class B and get the approval. Most of the vaccine manufacturing company, they have already a huge facility in terms of the filling capacity. And like what we did is during pandemic situation, somehow we have slowed down the other vaccine manufacturing and we have continued the COVID vaccine manufacturing. Now, the lack of manufacturing capacity for vaccine and therapeutic poses a public health risk to the world and prevent new medicine from reaching to the patient as fast as possible. As a global community looks to prevent, prepare for, and respond to future pandemic, it must commit to a new future for biotherapeutics manufacturing. And a public-private partnership plays a vital role in bringing together the innovation, infrastructure, and human capital that make this new ecosystem possible. From the morning, and also what Dr. Sudhanshu was mentioning, that there is a lot of partnership is required in terms of the academic institute and also to the private company. That is only limited up to the development stage. But beyond that, once it comes into the clinical material preparation and also the mass production of vaccine, that the whole thing lies onto the industry. For that, there should not be any partnership because that require a special infrastructure and manufacturing capacity. Now, there are two key considerations. The future preparedness required truly remain the global pharmaceutical manufacturing ecosystem to better support rapid response. One option is to extend the existing facility. That is the fastest way to move in. So the vaccine company who are already is in this field, for them it is very easy to extend or add some more equipment and continue the manufacturing. The second thing which comes is the new manufacturing line, which high demand output and substantial physical requirement, including an extremely skilled workforce and large scale bioreactor. That has two implications. What we have observed is that when we have tried to expand the existing manufacturing facility or to bring the new equipment for making the new facility, since it was a global pandemic, so the supply chain got badly affected, and that is the reason where we need to work it out in terms of the global supply chain for equipment and also for raw material and consumables. Most of the consumables product like tubing, bag, sterile vials, filters, all those things has taken much longer time to bring it to the facility. The delay for delivery of some of the material forecast to be 12 to 15 months. So you can imagine that even if you place the order now, it was taking 12 to 15 months, whereas in the normal condition, you may get in 3 to 4 months. So the global supply was badly affected during the pandemic reason. There are two reasons behind it. The first, there was a mass requirement from all over the place. The second, due to the transportation, due to the flight delay, due to the non-movement of the flight, this has happened. So finally, building an integrated approach for biomanufacturing is important. It is not only up to the development and clinical trial, it is beyond that, and where the supply chain plays a very big role along with the manufacturing, so that the vaccine can be made in large quantity and should be available to each and everyone in terms of preventing the spread. And these are the basically three pillars which from the morning itself we are talking of. And I think Dr. Sudhanshu has also indicated to me my time is getting over. So I'm closing it here, and I'm happy to take any question from your side. Thank you very much.
Thank you, Sudeep, for keeping to time. We'll take questions now, or queries or comments. Yeah.
As far as is concerned, as compared to normal vaccine production, how much extra cost the company has to invest during COVID like pandemics?
See, that depends upon what you require. The fortunate part was that we at Biologically, we were already having a large scale manufacturing facility, and we are quite well equipped in producing in Pichia. So somehow this technology was matching with our system, and we have not much invested onto the drug substance side. On the drug product side, yes, to enhance the capacity, we have purchased one high-speed filling line. That filling line was also, we have invested because we got a confirmation from the WHO that because of this COVID vaccine, your pentavalent should not get affected. Otherwise, if we would have delayed the pentavalent, probably we would have not spent it. But just to answer your question, if it is a drug substance and if you are going to build purely new facility, it will cost more than 100 to 200 crores. And the similar goes on to the drug product side. The complete GMP, because everything has to be as per the regulatory guideline, because the audit is going to come from the WHO, the US FDA, and from the EMA. So definitely the manufacturing is totally different setup, what you will see. Each and everything need to be get audited by the regulatory agency. And most of the equipment and consumables, what we are using or what the other company is using, is we are highly dependent on US or in Europe. So that was the major hurdle during the COVID vaccine mass scale production. Till it is in R&D, it is easy to get a small quantity and do the work.
Right, Varadarajan, just over here. Yeah, you go ahead.
So my question is something similar to what I asked in the morning to the industry representative. So again, you highlighted that it is important that academia and industry collaborate, right? So what to you is lacking at this moment? Where can we improve as far as the academics are concerned? Because academic research at times is driven by the inquisitiveness of the scientist, which may not be, taken up by the industry later on. I'm talking about the translational aspects of it. So when the industry is talking about more academy industry interaction, what is it that you exactly want from the academic institutions?
See, normally, I mean, Dr. Sudhanshu has also mentioned about that. If the collaboration is at the very beginning stage, then it is fine. Otherwise, after that, in industry, suppose if the molecule or the vaccine is successful, the audit will start right from the R&D. So the academic institute should be well equipped and they should be well trained in terms of working and doing everything as per the GLP guideline. Because once we go to the submit to the document, the audit starts from the beginning. So like in institute or research institute, if you just have clone developed and you will transfer, if you have developed the clone and you have transferred and rest of the thing has been done in the industry, because industry R&D is also GLP is in place and the audit is in place. So the audit is quite regularly getting audited by different agencies, so we are well tuned to it. So this is the only lacuna which I am seeing, is that everything in the institute, research institute, I am also coming from the research institute. I have worked more than 10 years in Tata Energy Research Institute. So I know that normally the setup of R&D in industry and the setup of R&D in the institute is different in terms of the mindset for working onto the GMP guideline, doing all the documentation as per the GLP guideline.
So again, I'm re-emphasizing that my question stems from the fact that government is now making the technology roadmap for 2047, and we've been asked to flag challenges. So you said that academics should be, GLP compliant and all that. In what way can industry come? In what way does government need to incentivize the industry to make these facilities possible in academic institutions? What are the incentives that the industry is looking for?
See, many of the industries, even biological, is also investing a lot of the money in different research institute. So once the collaboration is right at the beginning stage, the industry will help and also support financial, and also scientifically is the institutes are well equipped in terms of doing the scientific work. But onto the GLP part, and also in terms of investing whatever the money is required, that is there, provided if it is starting right from the beginning, not maybe after certain period of time.
In continuation to what Anamika said and what you said, you keep talking about we're starting right at the beginning. And when Anamika asked you to define what is the problem which the academics can think of working together with the industry, there's no definition of is there a priority of biologically, this is where we would like to work, and can the academia help us working on this problem?
No, there is absolutely no problem. Probably you are...
no problem, the priority.
No. I'm just telling the priority is also there. The biological E is working with many of the research institute. Both in terms of the development of the product, and in the morning itself, you have heard a lot in terms of doing a lot of analytical development for our COVID vaccine. So we support, we finance the people in terms of the academic as and when required, and wherever it is required, we are doing it. So we are not limited that we will not go to the research institute for any collaboration.
Which means that everything is all right, whatever way it is, that's the best case scenario. And there is nothing better that the government can do in terms of refining or reforming its policies to make the industry more inclusive of the academic institutions, or to incentivize R&D, setting up R&D basis in the academic institutions.
I don't think the government
there must be things which the government can do to incentivize. That's the basic question. What is it that we can do?
I think the government has already, everything is in place. It's between the government institute and the industry to come together.
So let me answer this. I have had also experience of working in both environment. Government has to do nothing really. It is the academia that has to train its people to record their data, to conduct their laboratory experiments under GLP. Unfortunately, there is no training for GLP in our academic setting. And if you try to implement any GLP of any level in academic lab, it becomes very difficult when you have students, trainees, people walking in, people walking out. So, in academic setting, it's not something that can be easily done, but we can certainly apprise or make our researchers sensitive to this, because the quality of data that we produce is good for publication, but when it goes to regulatory approval, thousands of things are missing. I mean, it's just that, people jot down only in telegraphic language, and that is not acceptable for these purposes. So government has to do nothing. It's just the academic training and the mindset has to change. Yeah, Varadarajan, this will be the last question.
Yeah, so I have the general impression that for mRNA manufacturing, the footprint is a bit smaller. And since can people not hear me? So I have the impression that for mRNA manufacturing, the footprint is a bit smaller, and the process development is less varied. So can that therefore be more distributed instead of having single large facilities as you were talking about?
See, it depends upon the yield. When I'm talking about the large manufacturing facility that require at least millions of doses to become in a batch. Because once it comes into the pandemic side, your facility, irrespective of the size of the facility, that should produce close to 1 to 2 million of dose per batch. Then you will be able to meet some of the requirement what in the morning we are talking. So it's irrespective whether it's a 500 liter bioreactor or 2 KL bioreactor. Each reactor should give you close to around 5 to 6 million doses to cater the full requirement if in case of the pandemic situation.
Okay, I think we'll stop here in the interest of time. Thank you very much, Sudeep.
Thank you, sir.
And we'll go to the next speaker, Professor Raghavan Varadarajan from Indian Institute of Science. His title of talk is IISc MINVAX Efforts on COVID-19 Vaccine Development During the Pandemic. Raghavan's laboratory works on understanding the relationship between protein sequence, stability, and structure, the application of high throughput mutagenesis to guide protein structure prediction, and on immunogen design for HIV, influenza, and SARS-CoV-2. In 2017, he co-founded MINVAX to take forward the influenza immunogen design work to the clinic. In early 2020, his lab initiated immunogen design efforts for COVID-19 as well, and candidate vaccine formulations for influenza and COVID-19 will be tested by MINVAX in clinical trials in 2023. So he's going to give us a glimpse of his work at IISc and MINVAX on COVID-19 vaccine.
Thanks for the opportunity to talk here. I just have a few slides. I think they were a bit surprised how few I had, but hopefully it'll be of some interest. So I'll just give you the timelines of the, does this work? Or is there a pointer here? Okay. Yeah, so this is the timeline of our, vaccine development efforts. So in late January, we had some discussion with the Bill and Melinda Gates Foundation because they had visited us a little while ago. And then by the middle of February, we had our constructs, placed orders for gene synthesis. Fortunately, we got them in before the lockdown happened. We immunized animals in March, and we probably had the first immunogenicity data in the country. Then the lockdown happened, and with great difficulty, we got police permission to ship sera across 3 states to Stalin's lab at ICER Trivandrum for NEWT assays. And then we did, analysis of the boost sera and got neutralizing antibodies. And then, it's taken a while to get funded to do clinical development. So we finally got funding released in April 2022 for various reasons which are too complicated to explain. So our candidate antigen was the receptor binding domain of the spike protein of SARS-CoV-2. And we showed very early on that this had exceptional thermotolerance. So we could take the lyophilized protein and expose it to 100 degrees for 1.5 hours. And at 37 degrees, we could keep the lyophilized protein for over a month without any change in the antigenicity. And also, more recently, we've shown the immunogenicity is also unaffected. We have subsequently shown that even we then made trimeric versions in this was, and that was ready a few months later, and we finally published it in 2021. And these also showed neutralization of all the VOCs. There was a lot of interest in our work. And then later on, we actually further stabilized the RBD to enhance the immunogenicity. Okay. So I'll just summarize, all the scientific stuff. So the wild type RBD is highly expressible in cell culture, and but it's a poor immunogen. And stabilizing the protein through mutation, which we did, results in about 100-fold increase in the neutralization titers. And this I've already mentioned. And so we think that these vaccine formulations likely do not require a cold chain. So the sera neutralizes all the variants of concern we've tested, including the Omicron variants, and the formulations protect transgenic mice and hamsters from high-dose pathogenic challenge. We've made permanent cell lines, and the clinical development is ongoing. Very early on, we also made the proteins in Pichia, and we found that they were, for reasons that we don't fully understand, much less immunogenic than the mammalian cell expressed protein. And so all of these factors, glycan identity, location, and protein stability, all play very key roles in immunogenicity. Okay. So this is, the science bit of things. Many of the things that I want to say, the previous two speakers have mentioned some of these aspects. So how can we improve our future responses to pandemic threats? Now, the most important is that we need timely, adequate, and broad-based funding. So there's a sufficient pool of expertise. In any given area, in the country, there are just very few people working. And, rather than have this 100 days of producing a vaccine, I really think we should just, we could set ourselves the modest challenge of within 100 days of your, technical committee approving funding, if you actually get 100% of the funding, that'd be a big win, okay? And you've seen how long it's taken us for some of the things. And, we really, I cannot emphasize the importance of getting funding on time, every time, okay? And I do not understand why this cannot happen. Okay, we are all just struggling. We have to write reports to describe funded progress at the end of the year, and the whole year we haven't got funding. Now, with this situation, how do we expect anything to happen? Okay, so specifically now in the case, and this doesn't apply to industry, but to the academics, it surely applies, the first bit. We need things within our borders, okay? We need, again, this has all been said before. I don't want to say too much about it, but some of these are complicated, okay? Getting all the media, all the adjuvants, they're all very closely held, in many of them in multinational companies, and they, they don't give them out. We have been very fortunate to work with the Vaccine Formulation Institute in Lausanne, and they have put adjuvants in the public domain in GMP grade, which is what we are using, but that's only one thing, other than alum and SWE, there is no adjuvant in the public domain. And gene synthesis, right? So we were lucky, we put in an order to GenScript early on in the pandemic, and we got antigens. And the pandemic came, all the flights were shut down, nobody had any DNA. And there's, as Dr. Harishchandra said, there is no company in this country that can make a gene for you. They can only make oligonucleotides, okay? And then you have to assemble the gene, which is a total waste of time. We should have people doing gene synthesis, and if it's not profitable, the government should pay them so that gene synthesis is readily available very quickly to anybody who wants it in a matter of 1 week, instead of, not being there because all the flights have shut down. Okay, we need quick IEC approvals, many more BSL-3 facilities, including for NHP challenge. It's virtually impossible to do NHP challenges. We also need a revised IP licensing and funding policies to facilitate academia industry collaboration. There is some work being done, But right now we have a situation where sometimes the funding agencies are owners in the IP, which makes it impossible to license it out. Exclusive licensing is also not permitted. And these are things hopefully that are in the process of being changed, but they have to change, because otherwise you cannot have a proper collaboration, because academia work is early stage and industry needs to take it forward. They're putting in a huge amount of money and they need, certain assurances on the IP from the very get-go. And we need faster access to reagents, virus, isolates, etc., and exceptions for carrying out work during a lockdown. Even sitting in IISc with a BSL-3 facility, it was like 6-8 months into the pandemic before we had access to the virus. And this is a virus which is in 100 million people in the country. Now you can't insist that you be worried about whether it's going to escape a BSL-3 facility. Okay, we need faster regulatory pathways. The current pathways are totally unsuited to innovator molecules and they're not in harmony with international practices, right? So you require a stable cell line, tag-free material, and 3 large batches just to get material for a GLP safety tox study. Abroad, you can just make material in shake flasks with a histag and put it into a phase 1 if it's properly documented. So we were stuck, right? We made plenty of protein, enough to do a phase 3 within a month or 2 of getting a clone. But because of all these requirements, and we couldn't do process development, only a large company could do that. And they were not interested. So, and really these are a huge barrier to rapidly testing new formulations because it costs crores to do all this, which it shouldn't. And these should be waived up to and including a phase 1-2 trial. The problem is all the regulatory policies are suited to not innovator molecules, but biosimilars, for which that the molecule is going to work. So people can invest huge amount of money upfront. What's the point of investing 10 crores in a molecule which is going to say, fail a GLP safety tox study, right? So this is also going to change. Okay, so that's it. And I must thank all the people who did the work at IISc. We had collaborations with THSTI, CSR, IMTech, and some of our material was validated in Australia with ICER Trivandrum, in CBS for developing the ACE2 mice, and Binvax, which is the startup that I co-founded and closely work with. And I must thank the Bill and Melinda Gates Foundation for giving us this early money. And I must thank the PSA's office, Dr. Shailaja and Vijay, for very early on supporting us to do some of this work when we had no funding. Citibank gave us a little bit of money, CSR money, and BIRAC. Finally, we need large amounts of money, and this is only possible with larger things like through the NBM, BIRAC, DBT, and DST, and they are really the backbone of everything we do, and we are deeply grateful to them. So thank you.
Thank you, Raghavan, for raising some very important questions regarding the funding, but that we will not discuss here. Is there anything else that you would like to add to his list of things that are required to be done? Or any query for him? Yes.
Hi, Dr. Raghavan. I think very pertinent and very relevant presentation. My name is Saurabh Sopti from CEPI. I just want to build upon in terms of, I don't want to gloat over the challenges you face trying to raise funding from the government. Just curious, what were your expectations or experiences while you were trying to raise private venture funding?
So venture funding, so we did raise venture funding, but Minvax works on flu and COVID vaccines. So, the market for a COVID vaccine long-term, because we have all these regulatory hurdles to go through, was uncertain. So venture funding here was not willing to give finally, we got initial funding early on before the pandemic started. When the pandemic started, though we had good material, venture investors were unsure of, what the long-term outlook would be. And it was only when the Delta wave started and lots of people died that actually some investors, and then we didn't really do anything. Somebody read a newspaper which said this vaccine was lying without funding, and then we got some venture funding to move things forward. But in general, not specifically for a COVID vaccine. And, for a small company, it's risky to put money, venture money into this kind of thing. It really has to come from, to be de-risked from a grant.
Right.
Yeah, you raised very pertinent points. One of them I really want to touch upon is the animal models. Like, animal models. So animal models are really required for some of the diseases. Maybe they are there for the COVID, and another is the NHP, which are not there. We don't have many facilities in India, and particularly what is required in the containment for these pathogens are badly required for when we want to accelerate the innovations and the testing of these molecules.
Yeah, I 100% agree. And it's extraordinarily difficult to get permission to do studies that will, that will then result in euthanization of the NHPs. And so, the facilities are not there. It's difficult to get those permissions. And then the result of all this is that if you do an NHP study, then those animals are there for the life of the facility. And what you do. So that's another problem which has to be dealt with. And some of the NHP facilities are not available to industry users also, although they're in publicly funded institutions.
Sudeep.
So Dr. Raghavan, a very nice presentation. And in one point you have mentioned that the regulatory required 3 large-scale batches to go for animal toxicity study. But the regulatory requirement is not for a large scale. One batch should be sufficient enough to produce a quantity which is required for the preclinical study.
It's not the quantity, but they needed to see batch to batch.
Batch to batch, there are 3 consistency
Batch
batches.
to batch, yeah, 3 consistency batches.
But the batch size should be not very large. It should be sufficient to produce toxicity material.
But what is small for biological E, it costs a startup quite a bit to do, because we have to do it in a CRO. And so just those 3 batches, even if you do at 50-liter scale or whatever, to run a whole phase 3 trial, probably we need at good yields, we may need 10 liters of a batch, right? But anyway.
Okay.
Yes.
Yeah. It's just a comment, because I think Gaurav raised a question of private equity, and being from the industry and being a founder, I'll tell you how difficult it is, especially if you're from India and your company is registered in India. Because if your IP is registered in India, it has very little global retail. That's the first question. So the first thing an investor would want you to do is put your IP in US or in the country that you're raising capital from. Second, the founder, after setting up the company and going through a whole host of things, will have to at least give away 40 to 50% of his equity. At least. So he's going to be on the minority side. Ask me. So now the question is, how can you raise non-dilutive equity? Either the government can give it, or we can create an ecosystem which I call stage gate. And that I have used very well with certain foreign companies, especially with the COVID, that if I hit this milestone, this is what you're going to give me. And this is the amount of shares you will give me. Now, let me educate you a little bit on this. Money you will get because you have invoiced. For RBI to get the shares of a NASDAQ listed company, you will need a TIN, a DIN, and another number. That took 18 months. The share of that company, which I got $5 million against, fell from $7.50 to $1.50. Now, why would I go through all this pain to raise capital? You understand? So if we need to fix the system, we have to understand 3 parts of the system: science, development, and capital, which unfortunately, as a scientist, It took me 17 years.
Okay. Thank you very much, Raghavan. We will now move on to next speaker, Dr. Syed Khalid Ali, Chief Scientific Officer, Panacea Biotech. Dr. Ali is a medical scientist with expertise in vaccinology and infectious diseases. He is responsible for overall development of vaccines and biologicals, including a broadly protected beta coronavirus vaccine at Panacea. The title of his talk is Industry Pandemic Preparedness, Meeting the Challenge, Sustainability. Dr. Ali.
Thank you, Dr. Vriti, for the introduction. And I would like to start by thanking the organizers for giving me an opportunity to speak on this occasion in such an important forum. I will also thank Dr. Raghavan Varadarajan for the energizing talk. I think it was like a cup of coffee for a lot of us. So thank you for that. The topic of my talk today is going to be industry pandemic preparedness, meeting the challenge sustainably. So this is another and highly simplistic view of vaccine development. There are multiple steps involved. And if we look at where both industry and academia excel at, you can clearly see that especially in the early phase, in the antigen discovery, lead identification, lead optimization, preclinical proof of concept, and even up to early clinical stages, academia is really good. And we have a collaboration with THSTI where a candidate that went through this step, that went through these steps has been taken up by us. Similarly, industry is really good in process and analytical development as well as formulation development. So I think we have heard through the day that Indian vaccine industry provides most number of doses globally. There is no other country that makes more number of vaccine doses than India. And we do it in an affordable manner. Dr. Shaligram's presentation this morning talks about the large number of doses that are made and the kind of manufacturing capacity that we have. And so we are really good at manufacturing at scale, late stage development of the product, getting them registered, WHO pre-qualified, and then supplying them globally, not just for our own domestic needs, but for needs outside of India. And so today, I am going to focus, in order to address the topic of the day, which is preparedness for future pandemics. Is India ready for 100-day challenge? I will focus on one aspect, which is manufacturing of vaccines. So I think both academia and industry have their own set of strengths. Industry is good at certain things and academia is good at certain things. I think even for pandemic preparedness, even to meet the CEPI's 100-day challenge, what we should do is we should harness these strengths as well as we should build upon them. We should further strengthen these approaches or these core expertise that we have. So coming to manufacturing, I think there was a publication which came out recently, and I have shared certain statistics here, which clearly emphasize the need or the value of having vaccine manufacturing in the country. So 5 countries which had vaccine manufacturing capacity, they could produce certain number of doses, they could vaccinate. I mean, in terms of vaccine supply during COVID pandemic, and we are talking about COVID vaccines, they could vaccinate more than, they could provide more than 100 doses per 100 people. So 100 individuals got more than 100 vaccine more than 100 vaccine doses. And so this emphasizes the value of having vaccine manufacturing in-house. The other 2 points that I have highlighted here, they address a behavior that was noticed and which is called as vaccine nationalism. So I will elaborate. I think if you look at vaccine purchases, countries which are the high income countries with economic muscle, they entered into one-on-one deals with vaccine manufacturers. And US, Europe, Canada, France, and other countries, which represent 16% of global adult population, they procured 54% of available vaccine doses. In contrast, LMIC accounts for 81% of global adult population, but they could only purchase 33% of vaccine doses. This was by 15th of March, 2022. Also, if you look at figures of how many vaccine doses were given, and this figure is from 14th of April, 2022, high income countries, 200 doses were given per 100 individuals. In low income countries, 22.3 doses could be administered to 100 individuals. And if you look at the 50 least wealthy countries by GDP, 8.2% of their population had at least one dose. So it was very obvious that vaccine nationalism, every country for themselves. And so I think this data clearly emphasizes the need of having vaccine manufacturing capacity. We, India, is quite lucky in that regard that we do have sufficient vaccine manufacturing capacity. But as I mentioned, we need to further build upon it and harness it. Okay, so to look at few things about vaccine manufacturing, and Dr. Sudeep gave an excellent presentation regarding GMP manufacturing or manufacturing in commercial setup for regulatory submissions and for sales and marketing. He mentioned, and I will re-emphasize, is these vaccine facilities, they take several years or months. And oftentimes vaccine manufacturing facilities are in short supply. If you approach a CRO for a GMP manufacturing kind of a requirement, you will be told that they are booked several months in advance. You will get a slot. If you approach someone today, most likely you will get a slot sometimes by the end of 2023. So these facilities are in short supply, difficult to make, and they are quite resource intensive to run. I mean, Dr. Sudeep again mentioned these are classified facilities, meaning these facilities, they meet very high standards of containment, sterility, cleanliness, to be called them as, CNC, grade D, grade C, grade B, or grade E. These are the various grades of good manufacturing facilities. And so you need skilled manpower, you need a lot of resources, electricity, water, and all of that. So, and then if you are to meet a 100-day vaccine challenge, then the only thing that needs to happen is you should have a vaccine facility which is up and running on standby, because other modalities may not happen. And to have such facilities on standby may not be very sustainable. Well, at least for a country like us, but I'll further elaborate on that. And then to add to this, different vaccine manufacturing platforms, they have different manufacturing requirements. For example, a whole inactivated COVID vaccine, you need a BSL-3 kind of a facility. A DNA vaccine or a RNA vaccine, messenger RNA based vaccine, it's a different facility. So it's not as easy, and having a vaccine manufacturing facility ready to meet the 100-day challenge could be one of the bottlenecks. But that being said, there are countries like Germany. They have invested close to $3 billion, billion with a B, to have 5 vaccine manufacturers, on standby. So these 5 vaccine manufacturers will keep certain vaccine manufacturing capacity on standby to meet need of COVID or any other pandemic or epidemic which may hit. But is this something that we can do? Country like us, that is 25,000 crores till 2029. So it's a lot of money. And even this does not guarantee that the facilities that you had kept on standby will serve the purpose. So the example here is Emergent, a similar arrangement in US. Emergent was contracted with making adenovirus-based COVID vaccine. They failed, and there was another report recently. I mean, they had made 400 million vaccine doses, which had to be destroyed because they did not meet the quality standard. So manufacturing is one area to have a dedicated vaccine manufacturing setup that could cater to different vaccine platforms and is ready on standby. It is difficult. Another approach that can be taken is repurposing of existing facilities. We have a lot of vaccine manufacturers in India, Zydus, AstraZeneca, Serum, Bharat. But during COVID, they had to, kind of stop their regular vaccine production. And these are essential vaccines. What this could result is shortage in supply of essential vaccine. This can cause outbreaks. We are currently, seeing the news of measles outbreak all over India. This is not due to shortage of measles vaccine. It's probably due to less coverage during the COVID times. But this could very well be another scenario. So we are in this situation where if we have to meet the 100-day challenge, we have to have a facility. And if we can't spend $3 billion like Germany, then what is an option for us? Because clearly that is not a very sustainable option. So there are various models that have been suggested during the day. One of the models that us at Panacea, me along with our BD team, we came up with is something like this, to have a setup with split capacity. For example, 25% of capacity to cater to internal needs, 25% for contract manufacturing, and then 50% or whatever proportion could be kept for pandemic preparedness. Yeah, 2 more slides. So, and these, it will be best if these facilities are, located in academic institute or any biotechnology cluster, because that is what is needed. Also, we will need multiple such facilities across the countries because they have to be equipped for 2 different platforms, or there could be any other model. And point here is that the revenue that is generated from continuous use, from internal use or from CMO activities, that can be used to support the manufacturing facility. Okay, and having this in an academic setup will also provide the trained manpower that, as pointed out, is critical. Also, final point, this has been discussed quite in details by previous speakers. During vaccine, like in non-clinical side, there was shortage of remdesivir and oxygen. We had shortage of raw material. Panacea, we were quoted lead time of 42 weeks for one of the components for a media. So if we order today, we will get the media in another year. So you can imagine this is not a very rosy scenario. And the point about which Dr. Raghavan mentioned about having all the essential supply chain inside the borders, this is being discussed. I think this is a press release fact sheet. So the President Joe Biden is going to invest in national biotechnology and biomanufacturing so that they don't have to rely on sources outside of the US border. They consider this as a risk to their vaccine and health security. I think we need this in India as well. So this was the second point that I wanted to make with regards to vaccine manufacturing. And final point is, I think we need more early interaction between academia and industry. This will help with the process and analytical development. And whenever research work or activities or product development is being carried out in an academic state, in an academic institute, if this is done with late stage in mind, I think that will be extremely helpful, and we will be able to cut down on process and analytical development timelines. And we have a good example of this. I think we have collaboration with THSTI, Dr. Sweetie, Dr. Amit, and this is working out really well. So with that, I'll stop, and thank you for your time and attention.
Thank you, Dr. Ali. We will take now questions, comments.
Thank you, Dr. Khalid. Very nice talk. You raised the issue of 50%, 50 lowest income countries having only 8% vaccination, but the mortality there was less than some of the other countries.
Yes.
Any thoughts on that?
Yes. So I had raised that point to emphasize on the need of having vaccine manufacturing capacity in-house, and not just the manufacturing capacity, the entire supply chain. So the idea is to be Atmanirbhar or independent in every aspect of vaccine manufacturing. Agreed. I think you rightly pointed out the mortality in those 50% of the poorest country was low. But that does not mean we should hope same thing will happen to us if there is a pandemic. So we should be prepared to be totally independent and having the manufacturing aspect at least covered in-house.
No, I agree with you. I'm just raising that point because this has been raised time and again. So
yes,
it needs further thought, and
exactly
why is that happening?
Yeah.
Right over there, just at your back.
Hi, hi. This is Harshit Jain, also from Panacea Biotech. Dr. Syed, could you help us understand how the Indian academia, especially the startup community, colleges, as increase our supply chains?
You were breaking in between. I couldn't hear the entire
Please
question. Can you
repeat
please
your
repeat?
question.
Hi, this is Harshit Jain from Panacea Biotech. So Dr. Syed, you mentioned there's a national biotechnology mission by the US government, and you also mentioned the need to be more Atmanirbhar. Could you also give a message for the Indian academia on how they can focus on other things apart from just vaccines to increase our overall capability in the country and develop the skill set that is required so that we can do it fast when time is not on our hands?
Agreed. Yeah, that is a very good point, actually. So in terms of biopharmaceutical research, vaccine is like the star that everybody is looking at. But in order to make that star, there are multiple smaller stars or planets or moons or whatever you want to call them. So you need good manufacturing systems. For example, the manufacturing equipment that we use are all imported, right? Sartorius, bioengineering, biotree, very little is happening in India. Media, very little is happening in India. We rely on Thermo, Gipco. Enzymes, that was discussed during the morning. Something simple, things as simple as yeast extract, right? So, and all of these individual components, they are an excellent opportunity for every country or any setup to spend in. They are equally, they are as important as the vaccine itself. And if the academia or biopharmaceutical setup is to invest and then develop something that will have market not just in India, it will have a global market. The other point is, I think, which I mentioned already, is that I think industry exposure to the fresh graduate students who are working, I think it is excellent, because this point was already discussed a few times. we, industry and academia kind of speaks different languages. And having that bridge or having someone that has seen both the sides will be extremely valuable.
Right, last question.
The data that you showed about the procurement and consumption of vaccines in India, it's very early 2021. And at that point of time, only the healthcare workers were provided with the data. Do you have any data in 2022? I mean, when the general population and precautionary dose have come, I think that the data, scenario has changed.
So the vaccine administration data I showed, where 200 doses in high income country were given to 100 individuals. So that is 2 doses per individual. That is from 14th of April 2022. So it is only 6 months old. The procurement data is from 2021, but the vaccine administration data and also the 22.3 doses per 100 population in LMIC, this is again from 14th of April. So it's old, not most recent, but it is from 6 months from now.
Okay, thank you, Dr. Ali. We will now move to the next speaker. That is Professor Amit Awasthi from THSTI. He will be talking about vaccines and T-cell response. Amit has been at the forefront of the nation's fight against COVID-19 pandemic. He contributed by establishing the animal models to study pathogenesis of SARS-CoV-2 infection and to support academic and non-academic partners to test their vaccine candidates and antiviral drugs. He has also contributed to cellular assays for Sputnik 5. He has received various prestigious awards like CDRI Award, G.N. Ramchandran Bioscience Award, etc. Amit, so over to you.
Thank you very much for a kind introduction. So actually, I'm one of the organizers. So I thanks all the speakers because I keep hearing a very good talk and it's really mind-blowing. So I'm going to really talk about cellular immune response in COVID-19. And this is actually what Guru has said, that we keep building the capacity, and that's what happened at the beginning when we joined THSTI in Gurgaon, and we keep building. And this is what we built in August, actually, 2020, just before the pandemic, right? And this is the immunology core. So this is something like, we wanted to start this facility, and we had all the infrastructure. We built all the infrastructure, and then we started, doing this antigen-specific T-cell assays, which actually came handy for the work what we have done for various industries. In addition to these cellular assays, we also built, established animal models, and these animal models were become handy for these various stages in the vaccine development. And these are the 2 stages what we actually contributed very significantly. One is the preclinical stage. So we tested the various vaccines of, came from industrial partners as well as academic, institutions. So we provided the support using this preclinical model what we had established. And then we also, supported the clinical trials where we measure the T-cell response particularly, because T-cell response were actually initially was not thought to be very important, but subsequently data keep emerging that the role of T-cells in COVID-19 is as much as antibody response. So these are the 2 critical platforms what we actually set it up. And using this platform, we could actually did preclinical, trials, antiviral drug testing, animal models we generated. And these animal models not only we use for this vaccine development or efficacy testing, we also use them to understand the immunopathology of the disease. So some of the things which I'll briefly mention in my next slide, and then obviously the cellular assay what we have established for clinical trials support. So these are the 2 important assays. One is the hamster challenge study what we have established in THSTI, and these we know that hamster is a kind of it's a natural model. It's like a kind of human in a way that the way the virus actually gets into the, animals and they get actually sick, losing weight, around 4 to 5 days and they recover, right? And around 14 days, they recover. And that's how we also have a very similar kind of, pattern in case of human. But we also established another animal model, which is very acute because that's actually dies in 7 to 8 days. So we have these two different models to ask very different questions. One is we can use for testing the vaccine, but also we can use them for antiviral testing. And these are the various, partners, biopharma companies and the academic collaboration, what we have done using these models to support, COVID-19 research and the vaccine development in the country. So, I mean, two notable vaccines which got, approval. One is the Zycov-D, world's first DNA vaccine, and the second one is Corbevax. Both of these vaccines we have tested in the hamster model in THSTI, and we provided them the data, and these data they have subsequently used for further approvals. So one of the things which I mentioned that, in using these animal models, we are also very much interested to understand the pathophysiology of the disease or the infection. And one of the things what we have used using this hamster model, that, SARS coronavirus not only actually infecting the lungs, but it actually goes into various other organs, and that's what causes like extrapulmonary disease. And one of the notable things what we could actually showed and published subsequently that, the SARS coronavirus gets into the heart, and actually using this various integrated metabolomics, lipidomics technique, we could clearly see there is a cardiovascular complication. There is a kind of that causes by SARS coronavirus, and that causes fibrosis in the heart. So in addition to this animal model, the T cell assay, which was actually very, very important, came out in various studies that T cells actually playing a role. Although these T cells are, is not as measurable as like, you can measure the antibody response. So therefore, what we quickly did, we quickly established these 5 different antigen-specific T cell assay in THSTI, and these assay we actually used for our own study, as well as we supported, various biopharma companies, and we provided them the data, which subsequently they have used for, DCGI and emergency use authorization of these vaccines. And these are the various, clinical trials we supported using these T cells assay. And some of these, BioE, which is a Corbevax, and Sputnik Light and Sputnik, we actually adapted the same protocol what they have published in Russian, this Lancet journal in Russia from Russia, and we use the same protocol. We established the same protocol, exactly the same parameter, and we provided and we got exactly the same result. So basically, we have done the 2 different trial for the Sputnik. One was actually the vaccine was imported from the Russia, and then the clinical trial was conducted in India. And then we provided the T cell assay data. And the second one, they actually transferred the technology to India, and Hetero Pharma actually, generated this vaccine in India. And then we have done bridging. We were part of the bridging trial where we also provide the, clinical, this T cell data for their clinical trials. And this is when you see this kind of thing, when your data being used for, various, approvals, you feel happy that, contributed something which is important, which is having a national importance. And that's what our lab actually, and THSTI has done in these various, trials. So, we know that T cells actually is very, very important. And one of the things which is very confusing about the T cell, because there is no universal, T cell assays. So various lab actually does different T cell assays. So everybody have their own protocols. There is no universality. So, and we know that various publications that, when you see that, severe disease, you have less functional T cells. And when you have a milder disease, you have more functional T cells. So that clearly indicate that you have, T cells is kind of, related to the disease severity. And this is also from various data sets. Actually, we made this, using various data sets. You can clearly see when you have a mild infection, you have very robust T cell response here. But when you have a severe infection, your T cell response is very, very poor. So this is also further indicating the importance of T cells. So the problem is the T cell is not really, and we also shown in the THSTI in a vaccine effectiveness study that, when you have, this is Covishield, around 63% vaccine effectiveness for the Delta, and which is around 81% for, Wuhan strain. But one of the interesting thing from this study came out that although during Delta surge, the Delta is not being neutralized good using this, Covishield, but at the same time, the T cell response remained preserved between ancestral and the Delta, spike antigen. So these studies really clearly indicating the role of T cell, and we wanted to really make some sort of universality or uniform or unification of the T cell assays. So what we have done, we have selected 3 different assays which has been done worldwide in various studies: activation induced marker, ELISpot, intracellular cytokine release assay. So we designed a study, and where we actually taken this around 1725 articles downloaded over 2, which published over 2 years using different keywords in the PubMed, and then you downloading this, and based on the exclusion and inclusion criteria, we come down to around 135 articles shortlisted. And from 135 articles, we actually downloaded all different data set for these 3 assays, which is AIM assay, ELISpot, and intracellular cytokine staining. And then we try to define the range of, each of these assays using the meta-analysis, systemic review, and meta-analysis. So you can clearly see that activation-induced marker, when actually the T cells get activated, the peptide antigen, you can say they actually express different surface marker, as you can see on the screen, CD40 ligand, oxide. These are the various different markers people used in different study, as that you can see here. These are the different study sets. So basically, one of the marker which normally used around is OX40 was CD137. Around 56 study use this marker. And then we further define the range of each of these markers from this data set, and we come up with a range for CD4 AIM assay. So we define the range. If your data comes in this range, that means your T cell response is actually going to be good. And the similar thing what we have done for the CD8 assays, and very similarly, we also define the range for CD8 antigen-specific response. We also did the ELISpots because we selected 3 different assays. So ELISpot, we also defined the range. You have different studies. We have vaccinated, active COVID-19, immunocompromised, and the convalescent. And taking all these things, we come up with the range, and this range come around, if you use like 100 to 500 spots, that means your T cell, antigen-specific T cell response, making interferon gamma is sufficient and is going to be good. So using these assays, then we further actually did the validation. We did the validation cohort. We around taken the cohort of 80 vaccinated or convalescent into hybrid immunity individuals, and then we did these ranges. We actually reactivated these the cells, and then we actually defined what is the range what we had defined. Our validation cohort also pinpointed those range. So that means whatever we actually did the meta-analysis, defined the range, is also actually true for the validation cohort. And then finally, what we have done, we did this correlation analysis. So we wanted to make sure that interchangeability of each of these assays, what we were actually mentioned during in this study, and what we actually could conclude from this, that if you actually do interferon gamma ELISpot assay, which is significantly correlate with the CD4 AIM, if you use these markers, or you can also use the interferon gamma, TNF alpha, and CD8 AIM assay, which is used by C25, CD69, and co-expression. So basically, either you can go for ELISpot, or you can pick any of these assays, which is kind of correlating and tell you that your antigen-specific T cell response is actually very good. So with this, we have made some publications on COVID-19 for various vaccine and various other animal models, and these were the people actually who worked day and night and supported this whole work during COVID-19. And obviously, last but not the least, these are the very important contributor to our study, and they were very generous during this COVID-19 to provide the financial support. And thank you very much. I will be happy to answer any question.
Thank you, Amit. Thank you. We'll take questions now. Yeah. ये इधर दीजिए.
First question is, were these antigen specific T cells you were measuring, or they were stimulated through anti-CD3 or CD28?
No, so we haven't used anti-CD328. These are activated by peptide pools. So we have taken this, overlapping peptide pools of spike antigen. We have taken pH as a positive control, and we also use a negative control, which is DMSO. So obviously, I think the best thing you could do is using the tetramer to pick these antigen-specific T cells. But I think in case of human, have different MHC, so I think it's very challenging to use the tetramer for this kind of thing. Therefore, I think this is perhaps the best way to measure the antigen-specific T cell response.
Could you see unresponsiveness or exhaustion in these T cells?
This is very, very, very valid question. I think Dr. Garg is here and he keep asking me the same. So I think we haven't really gone into those markers. We were focusing only on the activation marker. I think we could have included those panel as well to see the activation versus exhaustion together. So then the ratio between activation versus exhaustion could be more important to have that ratio. But I think we are doing that study now.
Amma.
Amit, here. You are seeing nice correlation between the ICS assays as well as the ELISpot. Were you not?
So we are seeing very good correlation with the ELISpot and the AIM, as well as interferon gamma, the Luminex based assay, not the ICS, because ICS is having a bit tricky because you have to reactivate these cells, you have to activate them, put Golgi stops. It's kind of a bit tricky. So that's what we did not go there. We try, but we don't see the correlation. So we see the correlation with the CD4 AIM, CD8 AIM, ELISpot, and the interferon gamma released by the Luminex.
Okay, so
the advantage of you can go for ELISpot assay, which is around 72. It takes 72 hours. So what you can do, you can go for CD4 AIM assay, which is 24-hour assay. And what you can do, you can have CD4 and CD8 together. So you have the two subset together, which is difficult to do in ELISpot.
Yeah. So just for your information, for the ICS assays, what they found is if you go for 18-hour stimulations, then you can score better.
Okay. We haven't tried. We tried to do the protocol what was actually used widely. So we did not see that kind of correlation.
Right, question over there.
Yeah, hi, thank you. This is Harshit Jain from Panacea Biotech. So Dr. Awasthi, we've seen THSTI work during the entire impressive work that you've just shown in the last 2 years. But could you elaborate on how experience in dealing with COVID-19 in an pandemic situation, how it prepares THSTI and the overall team better to respond to such epidemics or pandemics, and also how, what kind of capabilities that you have developed that can help industry develop better assays faster and also support them in clinical. So if you could overall summarize on a strategic level, the learnings that you had in the pandemic and how you have shaped THSTI to become a pioneer in India's response to epidemics and pandemics. Thank you.
Thank you. Very long question. So basically, I myself was kind of bit hesitant to take up this challenge before pandemic because I was kind of doing happily good in my own, corner in autoimmune diseases. But I think now that the COVID-19 pandemic gave us an opportunity, and now we are very confident. And what actually we could do, utilize this knowledge, what we had, gathered over the year. Animal models, the T cell response assay, and this is now we are expanding to other things. For example, monkeypox, right, or mpox, right? So basically we are doing the similar thing. We are activating this, the individuals who received the vaccine who born before 1977 or 79 or after 80 to see what kind of, so the assay remains same, right? But only thing is you have to keep changing the antigen and everything. So I think overall we are kind of in a much better position because we have these platforms, and these platforms can be quickly switched to address the question or hour of the need.
Yeah, last question.
Thank you, Dr. Amit. Any correlation with memory B cells? That's first question. And the T cells, have you looked at the subsets of memories?
I haven't checked the memory B cells, but what I did not show the data here, we have tested the follicular T helper cells, and we tested the circulating follicular T helper cells, which is having a positive correlation with the B cell response. So what we could see that, that follicular T helper cells is also correlating with the ELISpot as well as the AIM assay. So therefore we are extrapolating it. I'm not saying memory B cell is going to be same. I'm trying to extrapolate because follicular T helper cell is kind of going to give you the same B cell response.
All right, Amit, thank you very much.
Thank you.
We will now move to the next speaker. Delivering a VLP candidate in less than 100 days. Dr. Kundu has over 23 years of experience in various companies and positions, including establishment of production facilities for therapeutics and biologicals, vaccines and blood-derived products. As an entrepreneur, he co-founded Premas Biotech 17 years ago. Premas has become one of the top protein research and manufacturing companies in India and globally. Dr. Kundu.
Good evening, and sir, thank you so much. I'm really thankful to the organizers for giving us this opportunity. And I'll talk a little bit about and focus on the VLP vaccine that we sort of put together right from the scratch. And before I do, this is my agenda of my talk. I'll give you a little bit of, because many of you would not know of us, but we are practically your neighbors here. We are in Gurgaon, Manesar. I would like to give you a little bit of introduction to us before going. So we are a company that's in functional proteins. And based on that, a lot of our expertise over the years has been, we've manufactured over 800 proteins. We've got a phase 3 molecule along with Oramed, oral insulin in the US. We have a couple of other phase 1 molecules. And now the oral VLP vaccine for COVID is perhaps moving into phase 2 in Mexico and South Africa. All this has been possible by the ability for us to manufacture or develop 3 platforms. And the first one, which we call Decrypt, has been instrumental. It's a engineered yeast platform. It has been instrumental in making a lot of the membrane proteins. It is very dear to me because I worked a lot on membrane proteins. And it is this coming together of this platform, which is the engineered yeast, the vectors that we made, which was able to make this, the VLP vaccine that I'm going to speak to you about. Of course, we've made another novel India-based antibody library, which we are very, we've got now an asset moving into preclinical and perhaps phase 1 in immuno-oncology. And the last platform is called Axtec 4D, where we can take a human patient tumor, grow it outside, now dose and do a lot of omics on it and figure out the sensitivity. So these are the 3 platforms our company has typically developed, and we are moving ahead. When we look at the various speakers today and everyone, this is perhaps towards the end of the day, I'll make it a little light. One is you're looking for speed. The other thing you're looking for what we call roughly a definition of a platform. So from both a regulatory and a research side, perhaps from a research side, it's far more easier to understand what a platform could be. But what would be the definition from a regulator perspective? Well, how would a regulator look at a platform? The other thing we have sort of touched upon a little bit is cost of goods. Cost of goods have different connotations across the industry across platforms, across continents. Then would come scale. One is the inherent scale that you may have. The other one is the scale that you would like to perhaps build. So how did we get onto our triple antigen? So this decrypt platform, as we call, is an engineered Cerevisia strain. We've knocked out a few proteases. We've re-engineered certain energy pathways. We've made little bit changes in our vectors. We have optimized expression, upstream elements, and so on and so forth. The person who actually designed this is sitting in the audience. So if any of you have questions, we can get her to answer that. Oh, sorry. The other thing is, when you're looking at Saccharomyces cerevisiae, it's really easy to manufacture. The biggest issue that we have is one unit operation is, how do you break it open without cracking the contents inside? And sometimes I call, how would you break open eggs on an industrial scale where you break the outside and not break the yolk? So that is something we developed. And to be honest with you, this single unit operation took us about 10 months to develop. It's not easy, but once you know it, these are the things that actually allow you to convince a regulator that perhaps you have something which is reproducible, repeatable, and scalable. And another, on a lighter side, once the Belgium television was actually interviewing me very early days of the pandemic, and I said, you know what, Cerevisia, just to make it easier, and I'll tell you how sometimes similes or synonyms can backfire. So I said, I was trying to convince this lady, the reporter, that Cerevisia is very easy. And I said, it's like making beer. You know, in beer, they use yeast. Now, that was transliterated into Belgium. And next day, I got a call from the Budweiser CEO saying, we have large fermenters. You are using yeast for, as you said in your synonym about alcohol, would you like to use our fermenters? This is the typical typogram of our triple antigen VLP. We took spike, membrane, and envelope. We had worked with a lot of sort of similar viruses in the animal world. So we knew spike mutated. So therefore, we designed as early as January 2020 to have two other antigens that would not perhaps mutate so much. So we took membrane and envelope. Even as we speak, the mechanism of action of membrane and envelope is being worked out. There are multiple sort of, what do you call, indications that membrane and envelope are involved in some parts of the disease spread. And this is a cryo-electron microscopy picture of the VLP that we made. Very thankful to Monideepa at IIT Delhi for helping us get this. Now, how does this happen? The mechanism of action is that we overcrowd the endoplasmic reticulum because the highest real estate in any of these early eukaryotes is in Cerevisia. Cerevisia has naturally very large endoplasmic reticulums. So which means you can stuff it with a lot of proteins. We overproduce there. Then naturally, these proteins are now shifted to the Golgi. Once you produce furthermore, they are shifted into the ergic space. This is the space that these proteins, all three of them, come together to form a natural agglomeration of these proteins, which naturally have a very, what you call, static and constant ratio amongst them. Of course, we did a lot of genetic engineering to figure out that we should have more of spike, a lot of M, and very little E. So that sort of mechanism we had, and that's scalable, which resulted in the picture that I showed to you. Then came the question is, how quickly can we do it and replicate it? And this is just a summary slide of a lot of work that went on. We can develop the recombinant clone in about 45 to 50 days. We can then scale that up in about 21 days. And then in the next 20 days, we can give you material which is fully qualified. So that adds up to about 90 days. Then we put our entire system in making the Wuhan, the alpha, the beta, the gamma, the delta, the epsilon. And that's okay. But the question is, how does this qualify as a platform? So one of the things I tried finding out is, what is the definition of a platform from a regulatory point of view? A regulator, EMEA, has at least taken the first steps to define what a platform should look like. If the platform technology utilized to manufacture a licensed vaccine or other investigational vaccines is well characterized, it is possible to use the toxicology data and the clinical data accrued from the preclinical and clinical studies using that platform for other products to support a first in human clinical trials for newer threats. And therefore, now you start putting the Lego blocks of what not only would look like a platform from an R&D point of view, but also would qualify to allow the acceptance of a regulator. Furthermore, EMA has come out with something called a PTMF. Like you have heard of a DMF, which is a drug master file. They have come up with a PTMF. It's a platform technologies master file, the principle on which is integrated in the EU's new regulation on veterinary products, which means now, before we go into humans, at least on the veterinary side, if we can establish a platform which A, meets the definition on the top, B, can prove that it is well characterized, we will not require you to go and run the wheels of development over and over again. Has risk? Of course. Has logic? Sure. The question is, can we now extrapolate this logic to human science? So what does the data demonstrate? I'm very thankful to Dr. Amit and his lab for the data that he has generated here. We got some excellent data in terms of challenge studies. Then furthermore, if I go on, we were able to show that all the three antigens elicited a response. We also got elevated IF and gamma, which means T cells were being tickled as well. Then because of the regulatory issues, we had to, because this is novel and we were applying in India, we did huge amount of animal studies to show that it is safe. What was the scale up all about? And this is the simplicity of the platform that I talked about. Pretty much in about 120 hours, you could generate your drug substance, where about 48 hours was going to be your production time, irrespective of scale. Many of you know that Cerevisia doubles in about 2 hours, especially with the larger volume you put in. You could do about a 5 KL run or a 5,000 liter run in about a 120-hour cycle. Some of the data that I'm showing you again is at 550 liter, just to show you the replication in terms of scale up. Now let's look at the size. Let's examine the size. We get a polydispersity of this is a pleomorphic virus. The VLP is also going to not be of standardized shape. You get a distribution between 124 nanometers to around 300 nanometers. Only problem is when you put a 0.2 micron sterile filter, you lose 50% of your drug substance because it clogs the filter. Part of the learning. Then you look at an HPLC. When we initially started doing HPLCs, there was no HPLC to be found. So we partnered with Epidemics to make an HPLC for such a large molecule. Then we looked at a functional assay that could this bind to a ACE2 receptor, because on HepG2, there is high endogenous ACE2 receptor versus a MCF7, which is a low endogenous receptor. And we found, using fluorescence studies, it was there. And we found that the fluorescence was indeed internalized. Then a question came from the regulators. Out of the 18 batches that you have made, what is the ratio of the S, M, and E? Many times the antibodies were not available, but we had to develop. The person who developed this very interesting assay is actually also sitting in the audience. We found out that we got a very consistent S100, M about 1,050, E about 65. This allows a very good correlation to some of the data that is out there. Unfortunately, this video is not running. We then said, can we visualize our VLPs? So we undertook a nano tracking experience at 2 different dilutions, 0.5 milligram per ml, 0.25. And if the scatter graph looked well, then we are not imaging anything which is like an exosome, but VLP particles. The video is actually impressive, but due to technical issues, I'm not able to show you. So when we look at the distribution of the particles, we get a very uniform median particle distribution. And from this, we calculated the molecular weight of this particle. The resultant average molecular weight is about 100 megadaltons. Now, when you're looking at 100 megadalton particle, all the equipment that is designed per se breaks down and falls. None of the resins, none of the tubings, none of the hollow fibers is designed to withstand a particle this big. Either it will break it or it will not go through. It was a very interesting phase for us. And therefore, we had to develop a purification cycle which only included a very interesting size exclusion based selection. Now let's look at, maybe I have 2 minutes. All right, so I'll end very quickly. Now let's look at the DLS data. I've given you data which says, which I built my story, that there is a size, there is a shape, there is a molecular weight, there is purity, and there is a definition of the size. Now let's look at what the other particles fared. As we did the variance, you will see it's a very interesting development. As the spike changed, M and E remained the same. However, when it came to the India variant that we did, suddenly we see, and with the Brazil variant, we see there is a drop in the size of the VLP. The M and the E is the same. Just by changing the spike, we are seeing consistent changes. Certain things are very consistent, and somewhere we see certain drops. This is very interesting. All other physical phenomena, including the RT and the ACC, does not change. However, the size, hydrodynamic radius changes. When it comes to pre-formulation, we are typically between 3.8 milligram to about 4.2 milligrams. So coming to this, to get into the strictest definition of an R&D platform, this qualifies. To get into the strictest definition of a regulatory viable platform, I think this does qualify, but has to meet certain other regulatory needs. However, what we were also able to do is get about 300 milligrams per liter. At 10 micrograms, that's 30,000 doses per liter. So in a 1,000 liter, you are making about 30 million doses in 120 hours. That's not bad. So it is meeting most of your speed, platform, cost of goods, and scale. Cost of goods is about 60 rupees a dose or even less. We are now taking this platform and to show, because we've also cloned and expressed, perhaps for the first time, the full-length neuraminidase in this platform. So now we are adding HA and some other proteins to make the influenza VLP. We are now trying to work on the RSV and so on and so forth. Of course, we have taken this and formed a new company in US called Oravax. We have lyophilized the VLP. We take Oramed's oral formulation, make a capsule. This has gone and passed phase 1 in South Africa, and we are now being allowed to go into phase 2 and phase 3, hopefully in Mexico and South Africa as well. Now, none of this would be possible without a team. I'm really grateful to my team. I stand on their shoulders, the management, the board, BIRAC for its funding, to a subsidiary company, and our advisory board, which consists of these three eminent people, Tony, Gautam Sanyal, and Professor Suman Das. Suman is a student of ICGB of Dr. Shahid Jamil. With that, thank you so much, sir. Thank you. Happy to take questions.
Raghavan.
So with cerevisiae, normally hyperglycosylation is an issue. So is that a problem? For the spike has many glycosylation sites.
In many ways, yes. We had to find new antibodies for ourselves, because when we were using the spike, which is hyperglycosylated, the way you said, many of the commercial antibody kits were not working. So we had to A, raise some of our own, and B, find the one that worked. But that is true. But we did not find any issues with the neutralization, neither the immunogenicity. And both in the oral, we got a strong IgA and an IgG response.
Nice.
Yeah,
nice work. Did you have the stabilizing mutations in your spike?
Sorry?
The proline mutations to stabilize the spike. Did you use those?
No, this is actually whatever the natural sequence was. We just optimized it for East, the codon optimized it, but it was the full length sequence. East, on the other hand, has X2 sites. So it actually broke it into S1, S2, and that's how it formed the VLPs.
And what kind of neutralization titers did you get? I might have missed that slide.
When we did the oral one, actually it was done here. We were getting 1:680, 1:1280 and things in the PRNT assay.
Yeah.
Hello, yeah. So this is Priyanka from Panacea Biotech. So Prabhuda, I have a very interesting work. So I have a just question, like, because your VLP sizing is varying from 150 to 300. So how do you see in the consistency batches you are getting your yield, and is it affecting any of your immunogenicity?
That's a very good question. Priyanka, that's true. You do get, it's a pleomorphic virus in nature. So it is going to vary in size. There's nothing I can do. However, what we did is we found out hour by hour that if we harvest, say at 96 hours, the size, say 88, 92, like this, we kept on optimizing till we got to a situation where we got fairly consistent bulk. So we were able to identify that induction hour.
And through that, the yield is consistent from batch to batch.
Sure, of course.
Good evening. Representing a company called, here, sir, in the back side. We are representing a company called Postnova, who are manufacturing field flow fractionation. In the VLP separation, you have shown the polydispersity. So have you tried any microfluidic based technology, like a hyphenated technology, before detecting to NAPAC analysis, something like that, to get a monodispersed separation, then collection, and then downstream you have worked something like that?
No, we didn't. We went to DLS. But if you want to talk to my analytical head, he's here.
Yeah, what I am talking about, the hyphenated DLS, like you would first separate those the polydispersed viruses before characterizing these viruses in the DLS.
So we purify it first.
Yeah.
Absolutely. We have to purify it. We purify it. There is a 0.2 micron filter.
Yeah, so that is what have you done any process without any matrix? Like when you are purifying, then you are actually wasting all the, like you have stated in your presentation, like most of the things are damaged in the purification.
So we cannot use any normal resin. We have to use a mixed mode resin. We cannot use it. So in a mixed mode resin, what happens is the VLP goes in, everything else binds, the VLP just comes out in the void volume. So we did not use a bind and elute way. We bound all the contaminants.
Okay, sir. But what we wanted to say that there are systems available where you can actually separate without any specific reagents, what you have said. You can separate without any matrix. You can actually collect a monodispersed range of the viruses.
Correct. Because what you do is you do it based on size. If you think 100 megadaltons, you take a hollow fiber, which is say about 1 kilodalton, 1,000 kilodalton, 1 megadalton hollow fiber. All your proteins will go through, your VLP is retained on the lumen side.
Yes, sir. But there are different kind of the cutoff size of different hollow fibers are there. So you can actually optimize.
Yeah, you absolutely can do that.
Yeah. Thank you, sir.
Okay. Thank you, Dr. Kundu. We
Thank
will
you,
now
sir.
move to the last talk of the session by Dr. Vinay Kumar Elani from Bharat Biotech. The title of his talk is Vaccine Platform Technologies and Product Development Strategies to Address Infectious Diseases. Dr. Elani is working as a clinical research scientist at Bharat Biotech and is part of development team of COVID-19 vaccines. Dr. Vinay has done his master's in molecular biology from Skövde University in Sweden. But who is speaking? Sure, anybody can join. So we have to wait? And completed his PhD from Oslo University, Norway. Sai, are you there? I don't think he's there. Oh, there he is. Okay, Sai, can you please switch on your camera so that we are sure that you're there? Oh, good to see you, Sai. We can now, do you want to say something before we start? We can't hear you. No, I still can't hear you. I think you, yeah. Can somebody unmute him?
Hello, Sai sir. Hello. Hello. Hello, Sai sir.
No, no, you have to unmute him for here, from here. Okay, I guess let's go ahead with the presentation and we can hear from Sai. In the meantime, we can sort this out. Please go ahead.
Good evening, everyone. Yeah, I would like to thank you, the chairperson and the organizing committee, for giving this opportunity for us to talk here. And in this presentation, I would like to talk about our company, BBL Biotech, and its group of companies. And then I will talk about our vaccine portfolio, what we are having. And what are the technologies to face this, future epidemic challenges. We will talk about what we are developing and what we are adapting here. And then I will talk about the adjuvant technologies and clinical development strategies to accelerate the vaccine licensure. And then we'll conclude by saying that whether biotech is ready to take up this 100 days vaccine challenge or not. Bharat Biotech is established in 1996 by Dr. Krishna Ella and Mr. Suchitra Ella with a vision to develop the vaccines against neglected diseases and to make them available for everyone with an affordable price. And this is the Bharat Biotech is a Hyderabad-based company in Telangana, and we have other sister companies several across these states in India, with working 3,000 personnel and with 5 cities in these states. This is our inline Bharat Biotech vaccine portfolio. We have started in 1996 with this hepatitis. With this hepatitis B vaccine drug B in 1996, and by 2 years we got this marketing authorization in 1998. And thereafter, every year and after, we have developed one new vaccine or one new vaccine formulation for this existed vaccines. And most recently, we have developed these 2 COVID-19 vaccines. One is the intramuscular COVID-19 vaccine, that is Covaxin, which can be administered intramuscularly, which is whole virion inactivated vaccine. And another one is the Incovac, international COVID-19 vaccine, which is based on this adenovirus vector. And this is the world's second international vaccine which got this national regulatory authorization to vaccinate. And this is our pipeline portfolio of Bharat vaccine. We are developing several vaccines against these neglected diseases like cholera, chikungunya, Zika, and this TB vaccine, and these vaccines we are developing now. And the technologies for vaccine manufacturing platforms, what Bharat Biotech is having. We are having these various vaccine manufacturing facilities, technologies, and the cell culture technology we are using, and we are using several cell lines we are using in this platform, Vero cell lines and MCK and Hex cell lines. And by using this Vero cell line platform, we have made several vaccines like rotavirus vaccine, Rotavac, and killed vaccines like Japanese encephalitis vaccine, and the most recent one, the COVID-19 vaccine. Covaxin was also developed by using this cell culture vaccine platform, manufacturing platform. And Bharat Biotech also having these recombinant technologies. We have, in fact, our first vaccine, hepatitis B vaccine, was developed by using this technology. And initially we have done in the cloning in the bacterial, and now we moved to the yeast. We are cloning this in the yeast. And we have also established polysaccharide conjugate vaccine technology, where we have developed this typhoid conjugate vaccine by conjugating this VI polysaccharide of typhoid bacteria with tetanus toxoid. And this is the world's first typhoid conjugate vaccine, which got WHO pre-qualification. And we need to make, develop such technologies where also because it may, because of due to this emerging antimicrobial drug resistance problem, these bacterial diseases also become an epidemic in future. We may face such kind of things also. So we need to be develop these kind of technologies to make such vaccines polysaccharide. And Borg Biotech is, developing and collaborating with several universities in USA like Wisconsin, Washington University, and Thomas Jefferson universities. We are developing these vector-based vaccine platforms for these COVID-19 vaccines. Flu and this rabies vector were using to develop these COVID-19 vaccines. We have tested in phase 1 trial as well, CoroRab, with by using this platform, and we are also using it with one more vaccine platform, flu vaccine platform, for to develop the COVID-19 vaccine as well. And most interesting, this adenovirus vaccine we have already tested, and we got the marketing authorization for this international vaccine COVAX. And we also trying to engineer this platform to insert any kind of antigens what we found, to make ourselves available for to face these future epidemic or pandemic situations. And the most important thing is the facilities, what we need to have. Recent pandemic situation, we all came to know the importance of this BSL-3 facility, and Borg Biotech has this BSL-3 facilities. Because of that, we have made this whole virion inactivated vaccine. And for the future, maybe we need to develop this BSL-4 also. We never know what kind of challenge we are going to face in future. So we need to be prepared in that way. And we are working on various adjuvants to test, to make, and we are testing these analytical and formulation technologies by using these different kind of adjuvant, different novel adjuvants. And most recently, we have used these two novel adjuvants. One is the Algel IMDG, which is TLR7 by AI agonist, which we have used in our Covaxin vaccine. And because of this adjuvant, this Covaxin vaccine has shown the high cell-mediated immune response when compared with the traditional Algel. And we have also used another adjuvant like CEPI, which is an oil water emulsion, for our CoroRab vaccine. Just before I have shown that rabies vector we have used to develop this vaccine, and we have conducted the phase 1 clinical trials. And we have seen that these two novel adjuvants have shown high immunogenicity compared to this traditional Algel, and as well these adjuvants are well safe to administer for the human beings. We need to be prepared ourselves to test, to face these future epidemic or pandemic situations at the time. By the time we will develop all the safety and things for these novel adjuvants, and we can use in future to prepare the vaccines in a short period. And now I'm going to talk about these approaches to accelerate this vaccine licensure. The correlation of protection, establishment of this correlation of protection is an important thing to accelerate this vaccine licensure. And one more thing, we can conduct an immunobridging studies with these previously WHO pre-qualified vaccines to accelerate this vaccine development speed, and we can supply to other countries. And we have seen in previous presentations that still there are several countries not received enough vaccines. By using these strategies, we can make them, we can improve this supply chain. And we can conduct these comparative clinical studies as well. By using the vaccine, we can compare with the vaccine which are already proven for their efficacy. And this correlation protection, if we establish this correlation of protection for the disease, then we can use it as a surrogative endpoint to assess the vaccine efficacy instead of this clinical endpoint. And this helps us to save the product development cost and time for us. And by this strategy kind of thing, here I'm showing some list of these diseases where we have already established the correlation of protection for these diseases. And we using this strategy, we have got this licensure for our rabies vaccine, where we have conducted immunogenicity study and have proven that our vaccine has induced immuno antibody titer more than this COP. And we have also conducted for, as I mentioned, this type of TCV vaccine, typhoid conjugate vaccine. When we have got the licensure and WHO pre-qualification, we have not studied any efficacy studies for this vaccine. And we have compared, we have conducted immunobridging study with the typhoid VI, which is the previously WHO pre-qualified. And based on this data, we have got the licensure as well as the WHO pre-qualification. Based on the later on, we have conducted, efficacy studies by using this human challenge model with the help of this Oxford University, and have shown that our typhoid conjugate vaccine have shown 87% of the efficacy. And later on with this we have collaborated with several other institutes, and this TIVAC has conducted several effectiveness and efficacy studies across the globe in various countries. The non-endemic regions and endemic regions, and the regions where the extreme drug resistance bacteria or typhoid bacteria was outbroken, like in the Pakistan. And those studies were shown our vaccine has efficacy more than 80% in all the regions. And especially the 97% of efficacy has shown for this typhoid conjugate vaccine in this in the Pakistan, where this extreme drug resistance bacteria where typhoid was outbroken. And we can also conduct these comparative studies with previously proven for their efficacy. And we have conducted an efficacy study to prove the efficacy of this Covaxin vaccine in 25,800 participants across India, and have shown that 78% of the vaccine efficacy against this symptomatic COVID-19, and it was published in Lancet. And based on this vaccine, for our next vaccine, what we have developed, the intranasal Incovac vaccine, we have conducted a comparative study and proven that non-inferior, and in fact, it is superior. The intranasal vaccine is superior to the intramuscular vaccine here. We have shown this high neutralizing antibody titer, as well as the, based on this immunogenicity titer, it was shown that the intranasal vaccines, intranasal vaccine Incovac is superior to the Covaxin. And based on this data, we got this licensure for these intranasal vaccines to vaccinate both. We have also conducted another heterologous booster study. Based on this data, we got the license for homologous as well as the heterologous vaccination for this intranasal vaccine. And this is our strategy. For inline portfolio vaccines, we have conducted the various clinical studies. Based on the vaccine type, for some of the vaccines, we have conducted efficacy studies. And for some of the vaccines, we have conducted these immunogenicity, immunobridging studies and comparative studies with the previously the vaccines which are having the proven efficacy, and all to get this licensure and accelerate this licensure speed. And this is our plan for the future vaccines which were in the pipeline portfolio. And we are planning to conduct these studies, and we are also planning to conduct these human challenge studies for this intranasal vaccine still under discussion. And in conclusion, I would like to conclude that the Bharat Biotech has the wide expertise in this product development and clinical development, the formulation or derivative strategies, and manufacturing for platforms and these adjuvants. Based on by using this expertise, we can develop a vaccine in this within short period. And we have shown this in this pandemic period. We have developed this Covaxin vaccine and manufactured. We have got this vaccine from the NIV. And after getting this vaccine, by the 100th day, we have completed our vaccine development process by production and preclinical studies. And by 240th day, we got this emergency use authorization from the DCGI. And it happened because of this help and support of all the national institutes, NIV, ICMR, and all these AIMS hospitals, which helped us to conduct the clinical trials. And we got this virus from the National Institute of Virology, and we are supported by a team from this NIV, especially Nivetha Madam and Priya Madam and Gajan and Dr. Gajan helped us to develop this vaccine in terms of this. While conducting our clinical studies, they have helped a lot in by supplying these RT-PCR kits as well as these ELISA kits to sites. And based on our expertise, I mean, we can, it's 100-day challenge, as Bernard said, like that it is cannot be guaranteed to develop the vaccine in 100 days, but we have this expertise to rapid development of the vaccines. We are still developing our technology, and we are adapting the new technologies to develop and to take up this challenge to develop our vaccines. Thank you. Just I'm happy to take questions.
Thank you, Dr. Eleni. We'll take questions if there are any. Yeah, please. Sai doesn't have a question for us. Sai, you want to add anything? We still can't hear you. No, it's not going to work. I think there is some technical issue.
I have a question.
Yeah, please go ahead.
Did Bharat Biotech have challenges in manufacturing? We heard a lot of talk about manufacturing capacity and that being one of the biggest roadblock in the 100-day challenge. Even if we have the vaccine, manufacturing would be a problem. And from some media reports, we did hear that Bharat Biotech had manufacturing challenges even for the Covaxin. What were they and how did Bharat Biotech overcome them?
Yes, actually, we are also having these challenges I mentioned previously. In the previous speakers also mentioned, I mean, during this COVID-19 pandemic situation, definitely, I mean, the general challenges we face several to manufacture this. I mean, compared to other these mRNA or DNA vaccines, we have developed this whole virion inactivated vaccines. That BSL-3 facility is one of the biggest challenge to maintain. This is the first time something we are handling this kind of situation in there. And readily, luckily, we are having this, I mean, readily available BSL-3 facility for us to develop this vaccine by the time, but it would have been difficult for us to, for any company to make ready this kind of facilities to develop these vaccines, whole variant kind of vaccines.
Well, there's no other question then. Thank you very much, Eleni.
Thank you.
I think that brings us to the end of this session. I would like to thank all the speakers for their very nice presentations and sticking to time, and also to the audience which actively participated in the deliberations and discussions. So thank you very much. We will close here and hand it over back to the organizers. Thank you.
So I would request our chair to hand over a memento to all the speakers as a token of appreciation. So we had a panel discussion. So do we have to we should have the panel discussion or we want to skip it? Okay, then we can just go ahead with the ceremony. So can we have the mementos, please? So I would also request Dr. Pramod Garg to thank our chairperson by gifting him a token of our gratitude. So with this, we end our first day of international meet on preparedness for future epidemics. We sincerely thank our chairpersons, speakers, and delegates for making this day so enriching for us.